behind him. And one of the highlights of his research career is that he had done research in very difficult areas, including research in the Antarctic region. So I would like to invite Nalin Mehta, sir, to kindly deliver his talks. Sir, please take the stage.
A very good morning. A very good morning, ladies and gentlemen. First of all, I'd like to thank the organizers for having me here. And I'm not sure if after those stupendous talks today, I'll be able to hold your attention as compared to the stalwarts who had batted yesterday. And being the opening batsman is not a very easy job, but let me do what? Everybody is fresh after a good breakfast. So to ensure that, like Dr. Pandey was fearing yesterday, that people tend to sleep, I shall not stick to the podium for too long. So I'll be discussing ethical issues in biomedical research, and the focus is on the word discussion. I will not necessarily stick to my slides. With the permission of the organizers, I will have a slightly different format of discussion for this particular talk. I'm not here to lecture you on ethics because most of you are probably already very well versed. But I'm here to discuss issues, if you have in particular, while you are undertaking research, the issues you have with your ethics committees, which are also known as institutional review boards. We call them IECs or institutional ethics committees. And if there are members of ethics committees who have issues in dealing with research proposals, we can have a discussion on that as well. So this is what I intend to do, but not necessarily in this order. And may I request you to please raise your hands and ask your doubt or clarification, whatever have you, the minute it strikes your head or brain. Because when we hold on to a question, we tend to forget it. Now, ethics is a very interesting thing. Everybody feels that we are very, very ethical because we do things our way, including the good deed for the day where people tend to do when they help somebody cross the road. So on a very busy intersection, people feel if they help somebody cross the road, it's a good deed, without realizing that that person may not have wanted to cross the road, a blind man. So when you help a blind man cross the road, you will not be helping him, you may be harming him or her. Because did you ask the person whether the person wanted to? We tend to believe that whatever we are doing is for the benefit of the research participant or for the benefit of the patient. So there comes the issue of consent. So I think it's very, very important to have your ethical guidelines and your ethics as part of your nature rather than getting into trouble, because life is very, very different. The perspective you get from behind bars is very different from what you get otherwise. So how do we define ethics? Campbell et al. Have said it's best thought of as a critical scrutiny of moral thought and morality as our idea of right conduct. Now the question is, what is right conduct? Half the world drives on the right, half the world drives on the right, and we are so equally divided, we do not know what is right or wrong, or what is correct. Meaning thereby that the ethical context varies from place to place. It may be a geographical terrain, it may be the ethnicity of the locals, and such a diverse nation as ours, we have a very, very, varied population, right from Kanyakumari to Kashmir, and you come down to the end, you come to Arunachal or Nagaland, go down to Chowpatty. Different races, different tribes, different people, different languages, different food, different customs, different cultures, and different traditions. Now the issue is that it is your responsibility as a physician or as a researcher to ensure that you respect people's cultures, languages, diversity, and you are not trained to do that. But you are expected to follow the guidelines, the ICMR guidelines. Now, when people talk of recommendations and guidelines, they tend to say, oh my God, these are just guidelines, I don't need to follow them. But please remember, the ICMR guidelines, ICMR being a regulatory authority, this is as good as de facto law. Because the Drugs and Cosmetics Act 1960 has very categorically stated that you shall follow the guidelines or recommendations of the national regulatory authorities as amended from time to time, etc. So these are not just guidelines. Any violation can at times be violation of law or infringement. Now you have PTND, you have Organ Transplant Act, you have a whole bunch of laws. So this also is de facto law because under the Drugs and Cosmetics Act there is a statement. So please be very, very careful with your dealing. The other issue is nobody teaches you ethics, especially resident. Now there is an ATCOM module in the competency-based medical education curriculum that has a few sessions for first-year students. So you tick your box that yes, we have taken a couple of sessions on ethics. But it does no good to anybody, is what I feel. Now when you're a postgraduate exposed to research or when you're a scientist getting into research, the onus of knowing, appreciating, and following ethics lies on you. And if there's any trouble, you are in trouble. So please feel free to ask questions if in your area of research you have had any issues or as members of ethics committees you have any issues. Because after the 2013 fiasco, the members of ethics committee are also culpable if things go down in research. You are accountable. You may have punishment or punitive actions against you. A slightly simpler one, it's a reasoned process for dealing with a problem concerning what ought or should be done in a situation. A reasonable thing, you talk of morals, you talk of values. Now, what do you think is reasonable in a specific situation? And what is that situation? I'll just touch upon morals and values. Morals are societal constructs. What we tend to do in society to ensure that there is a little bit of decorum. What you do or what you do not do in public is governed by our moral assiety. Values are your personal. You may have, or a small group of people may have some specific values. But when you talk of ethics, and well, let's take it in the context of healthcare. A patient comes to you for treatment. The patient has, say, an appendicitis and requires surgery. Acute appendicitis may rupture, blah, blah, blah. You tell the patient that you need to be operated. The patient says, I don't need to be. I don't want surgery. The patient is competent. You talk to the patient's relatives, they all say no surgery. And the patient dies. After you have recorded that there will be no surgery, the patient has refused life-saving surgery. You are safe. You save the patient's life by operating without the patient's consent, you are in trouble. So what you are right may not keep you safe. So there are serious issues with ethics. And apart from being an unusual topic, it grows with experience. As you do more and more complex things, through your experience, you start picking up the nuances of ethics. It's not just guidelines and regulations. You might have to do minor track corrections or changes. That again will come from your experiences. And as doctors and physicians and biomedical researchers, you are in a very, very unique. And a whole bunch of sterilized because people thought that they were too basic or too illiterate to bring up babies. So these kind of things happened on the planet. But the World War II was a life-defining moment. History is written by the victors. So the crimes committed by the German doctors in places which they had occupied were horrific. So the entire world woke up, the Nuremberg trials took place, and then there was the Helsinki Declaration. After the trial, people decided that we should do something to prevent harm to humans who are being treated or researched upon. Now, thalidomide. We are all aware of the thalidomide issue. It was a wonder drug. But because it caused a horrible phocomelia where there were, deformed limbs, the drug went into severe disrepute. But the problem was not that it caused phocomelia. The problem was that the manufacturers were aware that if pregnant women took this drug, they will get into trouble. They had not told anybody. So these kind of issues raised a lot of problems. Eventually, the court decided a few $1 million of compensation was paid in those days. So the drug companies became very careful that we need to research our product very carefully before we release it into the public. Otherwise, there were some people who were struggling with diseases like cancer, and they were ready to do anything, take any risk in order to try and increase their longevity. So if a drug manufacturer had a drug which had higher risk, patients were willing to take it, but the people who were protecting the patients, they said no patient may get harmed. So then there was a discussion that there should be all stakeholders deciding upon what kind of research should be permitted or what kind of research should not be permitted. And that is why Articulate Patients Group, they came together, and then they decided that we'll make ethics committees with all stakeholders. And that is how the ethics committees came into being, to ensure that this was on the Times cover, we do not use human beings as guinea pigs. How many of you here are members of ethics committees? Right. How easy is it for you to decide upon the risk or benefit of a research protocol? You remember? You raise your hand. Yes, so how do you decide risk and benefit of a trial when the proposal comes to you?
Sir, all the members are given the copy and then we discuss.
How do you decide? See, when the ball is in your court, it becomes very difficult.
Sir, we have a checklist.
My dear lady, there is no checklist for risk-benefit analysis. The risk and benefit to every trial may be different.
Sir, I was telling about the, like, when we have a ethical thing, that time we have some of the points we put and then we used it as.
Okay, so you check that this will cause no harm to the patient, etc., etc. If you think hourly blood sampling for a research protocol, 10 ml of blood drawn every 2 hours for 24 hours, once a month, is that acceptable or is it risky for a patient? Raise your hand if you think it should not. Every 2 hours, 10 ml of blood is drawn for research purposes for 24 hours, so 12 times. Is it all right or not? So as members of the ethics committee, you will not permit. It is perfectly all right. Drawing 150 to 200 ml of blood does no harm to the patient at all. All you need to make is sure that you have a Venflon or something in place where you can draw the blood without causing multiple pricks. Every time you donate blood, it is 350 cc approximately, 300 a pint and the anticoagulant. So what we feel may not necessarily always be risky. So the committee needs to sit and decide whether this proposal should go forward or not. So when these decisions are made, modern day bioethics is basically, staked on 4 pillars. The first is autonomy of the research participants. You will not carry out research without explaining to the research participant what will be done, what will their participation entail, what is the exact process, how long will it take, will there be any compensation, will there be any risk, and the fact that they are free to walk out of that research at their own will without any coercion, without any compulsion. So these are things the participants should know before they participate and you consent them into enrollment. You can't tell the participant that if you participate in my research, your treatment in this hospital will be free. If you don't participate, you fend for yourself. People do that. And have inducement. We'll give you so much money to come every day. Yes.
Sir, whatever you are saying, the ethics is absolutely fine, but the thing is, suppose if I am a patient, is it not duty of the doctors that I have been informed all the ethics? That is it not duty of a doctor that I should have been informed before going to operate at any charge or anything about this?
No, I didn't understand. You have to be informed about what?
About regarding that, suppose you are saying that it is safe that means blood of 150 to 200 can be extracted. Is it because if I'm a patient, I don't have any idea about that. So my question is not duty of a doctor to be informed to the patient?
Absolutely, that's what I'm saying. So everything that you do to the research participant should be explained to him or her. And you should tell them that these are the risks associated and these are the benefits from the research. The risk may be minimal or more than minimal, and it depends. The ethics committee may give even high risk consent to research which is extremely important. For example, cancer patient research, the end point is death. So the patient is going to die. That doesn't mean you can't try a new drug which may have very toxic effects, because the patient is dying in any case. So the committee has to connect the risk versus benefit. What can be the benefit of a research? It may simply be direct benefit to the participant or to the participant's relatives, or to people with a similar disease or disability, or it may just add to the scientific knowledge or the corpus of scientific knowledge. So you may have direct and indirect benefits. That is what the committee will. And a very, very quick touch upon risk benefit, we are not going into details. When you have inclusion and exclusion criteria, inclusion criteria help you narrow down the participants in such a manner that it is good for scientific validity. Age matching, sex matching, etc., etc., right? Exclusion criteria, on the other hand, mitigate the risk. You will exclude people. For example, some medicine you are trying and say diabetics should not be included. It may harm them. Pregnant women will be excluded. So people who may get harmed from a particular intervention may be excluded from the research. It mitigates the risk. That having been said, there is one very, very common thing that we see in most research protocols. Exclusion criteria: pregnant women, alcoholics, smokers. These three groups have done nothing to not benefit from research. So please exclude pregnant women, smokers, and drinkers only if their participation is going to cause trouble either to the validity of the research or they are going to get harmed. From every research, you don't have to, categorically remove them. Otherwise, there will be no research on them at all. They also deserve the benefit of research. The second one is the no-brainer, beneficence. You should do good to the patient as well as the participant. This applies to healthcare and to research. Now the third one, non-maleficence or primum non nocere, was the first ethical principle if you go down in history. First of all, do no harm. And how can you harm a research participant? It may be physical harm, it may be mental harm, it may be dignitary harm, it may be even financial harm. You get them to participate in a research, they get into a little trouble, they have to spend a lot of money getting themselves treated, etc. The resources that you have will be shared equally among the participants or patients if you are treating them. And more importantly, subject selection. Or if you have students, I will take only my students. The inclusion criteria should define what subject participants you will include in your study. So these are all things which are equitable. A lot of people do what? I'm going to pay 500 rupees per visit for some protocol. So I choose my friends and relatives, and that's my sample. And a far more pleasing voice than mine. Anyway, so I hope these are things that we have probably understood reasonably well. Now, out of these four basic pillars of bioethics, we have general ethical principles which are given in the ICMR guidelines for biomedical research on human subjects. Those of you who are working on pediatric age group, there is a separate guideline for pediatric age group. The 2017 modification that happened, we have a separate guideline. There is a separate guideline for genetic studies. There is a separate guideline for animal studies. There is a separate guideline for gene therapy. So if you go to the ICMR website, all these guidelines are there. may I request you to download them in a folder on your laptop and office computer and read the guidelines which are specific to your area of research over a period of days very thoroughly. There are a couple of them, social responsibility and environmental protection, which are highlighted in, I don't know, the ladies may call it brown, I call it red. We are bad at colors, guys. We just know black, yellow, green, white, etc. Ladies know magenta, burgundy, turquoise, indigo. See, I have one wife and one daughter, so now these colors are something which I also appreciate. Otherwise, my life was all black, white. Yours also, gray. Most men are gray trousers, black trousers. Anyway, so these have been added in the latest guidelines, which I think is very pertinent. Whenever you are doing research, you should have social responsibility that stems from one is informing the society of what you are doing, and your moral obligation to disseminate the results of your research to the public. If you have done a project, a funded project, people have invested time, effort, participants have participated, they have given their time, your research equipment has been utilized, and you do not publish, it is almost criminal. So I think everybody please note, if you have a project, you have to publish the results. It may be a technical report or a publication in any organ, a good peer-reviewed journal, even a technical report, but public should know what came out of your research. It is not always necessary that your research should have a very positive kind of a result. You may say that we did the effect of this on that and there was no effect found. Even a negative conclusion is extremely beneficial to the scientific community. So please be honest with your research output. Whether you get a good result or a bad result or equivocal stuff, publish and disseminate. The rest of them, if you do any research which is not essential, again you're wasting time and effort. Like Pratap had mentioned yesterday, in 1973 it was evident that streptokinase was helpful, but we were still giving lidocaine. So when the research is not essential, the research committee should, the ethics committee should also do a little, whenever you get your proposals. Supposing you are a member of the committee and there are 20 proposals to be discussed, normally they give it to you 3 weeks a priori. It is your duty to read those 20 proposals. And also do a literature search whether similar work has been done earlier by somebody else or not. So you can't be doing repetitive research. Now what you can do, if there is some research effect of A on B at such and such a time, which has been, say, in Tamil Nadu, the exact same research can be done in Assam or in Nagaland or in Jammu and Kashmir. Because now the ethnicity of the people is different. You can't do the same thing in the same place and waste time, effort and money. So similar things can be done where the population is different. So we talk of what works. It was mentioned yesterday that there are some things which may work in a Western population, which may not work here. So depending on the ethnicity of the people, there are research projects that you can undertake. It has to be absolutely voluntary. You can't force people. You cannot exploit them. I hope everybody understands inducement, especially in multi-centric trial. You have a trial in collaboration with somebody from Europe or US, and they give $500 a day to the participant to come to the hospital and spend an entire day. Now, way back in 2002, when I was in Toronto, $25 was the parking fee in the University of Toronto. So if you're going to spend almost $50 on parking, $50 to $100 for snacks and coffee, lunch, dinner, etc., $500 is equivalent to 500 rupees here. So your transport, you're spending the day there and going back, it is generally covered. If you give that same $500 to people here in that multicentric study, people who should not be participating in that research, for example, if it is not meant for diabetes, that the drug may cause very severe or maybe even death, people will hide their diabetic status and participate. That is inducement. Because $500 for some people may be monthly or even annual salary, depending on, which strata of the society they belong to. So that is inducement. You have to ensure that the data is kept confidential. The ethics committee may ask you, how are you going to maintain confidentiality? You just can't say we'll save it in a computer, because that computer may be accessed by hundreds of people. So you have to make arrangements to ensure the safety of the data, because loss of confidentiality can cause a lot of harm to the patients. Two very interesting examples. The BRCA gene, sometime way back in the 90s, Lancet published. Now the breast cancer gene, we are all aware of the BRCA gene that was. It said the high incidence of breast cancer in the Ashkenazi Jews in the US. Now the Ashkenazi Jews are a very close group of people. You can work on them because there is very little variation. For example, when you do animal studies, people work on a very specific breed of dog, the beagle, because they are very, very similar. There are not too many variations. Now, the minute this study got published, many employers threw out the Jewish women from their companies, that we cannot afford to pay their insurance premium, which has gone up because of their cancer affliction. Then they went to court. The court decided against it. Everybody was re-employed, and those kind of things happen. When HIV/AIDS came in, some patients' HIV status, when it became public, the entire society ostracized them. It happens for much lesser in our country. People with tuberculosis or cancer, families tend to shun them. So things are changing, but we need to realize that we have to keep the research data and the patients, whether you are in healthcare delivery, the reports very confidentially. One thing that happens very commonly in our hospital, you can walk into the ward and tell the sister that Mr. So-and-so is admitted, I need his reports. I'm his relative. And they give it in most hospitals. In the corporate culture, it is changing. Now, even in the government setup, it is changing. Access to report, lack of confidentiality. In your office, somebody has some issue, everybody knows by evening. So these are very confidential things. We should learn to keep them confidential. It affects people adversely. You should be professionally competent before you take up any research. That again will be decided by the committee. And that is when they ask you for past work done. Have you published in this area? Have you done work in, etc. Those kind of things. Even before, you won't even get funding if you don't have a track record. Then maximization of benefit. How do you maximize the benefit to the participant? To ensure that they stay in the hospital is comfortable. And then there are benefits which may accrue even post-trial. If they have participated in a trial which has a very specific drug for their condition, and the end of the trial has been successful, you can't withdraw the drug. This happened in sub-Saharan Africa when AZT, anti-HIV medication was tried. It was a very successful trial, but the drug company went back home after the trial, leaving everybody to suffer. A huge hue and cry, and then they provided post-trial benefit to that entire tribal population. Your institute should have arrangements, and the head of the institute has to ensure that you are given adequate facilities in whatever is available you can use for your research. It doesn't belong to me that I won't let you use it. I have to, in fact, even sign that, yes, I will permit such research to go on. Transparency and accountability. Your communication with the participants and everybody should be totally transparent, except the findings of your research and the data, etc., will be kept confidential. Even the police cannot come and access it. Regulatory authorities, members of the ethics committee and other regulatory authorities like SEDESCO and Drug Controller General of India, they can access it. And if it has to be accessed by somebody else, it has to be through a court order. So do not get intimidated by government authorities to disclose your research findings. Only through a court order or only regulatory agencies. Is there anything we've left out here? So environmental protection, your research should be carried out in a manner in which there is no damage to the environment, and this is the recent guidelines. Now, what are the general issues? Most committees struggle with benefit risk analysis. So risk is basically as we envisage, and yesterday we were discussing risk. I think Pratap Tarian mentioned risk. please make sure of two things. When we talk of minimal risk, which is the kind of discomfort you face during routine physical and mental examination, etc., etc. Everybody doesn't go through a routine physical or mental examination every day. There is a risk in crossing the road. So just to get into perspective how we accept risk. How many of you have traveled here in vehicles or used a four-wheeler or two-wheeler after getting here? Please raise your hand. How many of you have not? See, this is a thing which is very common. When there are 100 people sitting, 11 will raise their hands and the rest will not on both yes and no. How many of you are here on horseback? Funny, right? 4,000 fatalities, deaths, when I last Googled, now it must be a little more, every single day in our country from automobile accidents. There is not one single death on horseback riding. There have been 7 deaths from equestrian events, show jumping and recreational horseback riding. So we all know that animal-driven vehicles, horses or oxen, bullock carts are far more safer than vehicles, but we choose to drive because it is convenient, time-saving. The benefits are immense. But if you Google number of deaths every day, you will go see it's in thousands. So there are some risks that as a population we are willing to accept. We don't even think about it. We blindly get into a car and drive. We have no control over the other drivers. At times we don't have control on ourselves. So these are issues that the committee has to ensure that you just can't say, no, 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 this is very risky. It's not your subjective feeling. Back it up with either evidence, you do a Google search, or from past experience of other researchers, like that 20 mm death by lightning. Do we? Not at all. So please, there are a few things which have risk, but we do not think about it. So minimal risk. Always remember, risk has 2 characteristics. We were talking about odds ratio, etc. yesterday. Risk has a probability. This will happen to so many out of 100,000. Right? There is another perspective of risk. Risk as an uncertainty. Like the market is risky. We have no idea whether it will be good or bad. We have no idea about the harm that it will create. We cannot even predict it. So risk is not just the probability-based thing. When you do it in research, when you consider risk, it is also something based on uncertainty. So please never apply the word minimal to risk and harm without considering risk as a probability separately and risk as an uncertainty separately. Because the minimum risk definition is terrible. This I have already mentioned. Payment to participation and inducement I have mentioned. Now compensation for research-related harms. Most of you are working in government setup and doing your research. If there is any harm that befalls the patient, and especially if you are doing a clinical trial, how many of you know the definition of a clinical trial? Please raise your hand honestly. So, if you are trying any intervention, a drug or a device, which is already in the market, well established, it has been certified by CETESCO, Drug Controller of General of India, but now you want to try it in a slightly different dose. Everybody knows, okay, antibiotic 500 milligrams, 4 times used a different dose. So tweaking the dose makes it a clinical trial, new clinical trial. And if you are not aware, it can get you into trouble. Tweaking the dose, tweaking the formulation, tweaking the route of administration. If you change any of this or the indication, it becomes a new drug. If you do not have permission for it, then you are in trouble. Only ethics committees that are registered with CETESCO can review a clinical trial. We'll talk about ethics committees later, or maybe we won't even talk about them, guidelines are there. The other thing is, for the anthropologists and for the social scientists, If you are going to bring about a change in the behavior of a certain population, that becomes a clinical trial. Yes, ma'am.
Sir, in case during the study we find uh, we are observing something, some practices or which is part, and we are finding that the guideline doses or something is not followed and all. In some other things also, the way it should be, it is not happening. No, no, sir. As a researcher, during the study, if we are finding something like this, do we need to see that these things are rectified? It's not part of the protocol as per se, but we are finding that this should be done, but it is not followed. Some
Change the protocol then.
not per our protocol. We are supposed finding how the things are being done, health system analysis or something assessment, and we are finding something is not followed properly.
Can I just have two questions? That we is you yourself are doing the research?
I am doing the research, and suppose in the field we are finding in the some in PHC or somewhere some dose is given by some health worker which is not a correct dose, which should not be given, and it is being given.
Now, is it part of the research? Okay, wonderful. So there is a researcher who has a primary health center as one of the sites, and they have a protocol. A specific drug has to be given in a specific dose, and when you go there, you see that the sister in charge or the other people are not giving that dose. So one is it has to be pointed out, and if X number of participants have received that wrong dose, it has to be noted in your findings. It's not a clinical trial where they have decided to use a different dose, but by mistake the dose which was supposed to be studied is not being studied. So those results will not be valid. So get that subset aside, maybe rectify the mistake and you can study them later, and talk to the people and ensure that they are trained. That is why the entire research staff, especially in different sites, are brought together and trained so that they are on the same page, including informed consent. I am not going to touch on informed consent. It is probably the holy grail of modern day research. If the consent is not taken properly, things can go seriously south. Informed consent in the most developed of nations, to countries like Sierra Leone, where the development is next to nothing, is never complete. Nobody on the planet has taken informed consent like it has to be done. It's very difficult. But please try to do your best or it will get you into trouble. So when informed consent is being taken for multiple sites, the people involved are brought together to ensure that they give the same message to the persons. And it should be given in a language that they understand. That Google Translator does not work. For example, there was a Hindi translation of a novel technique. The Google gave Upanyas Tarika. Upanyas is a novel storybook. But research scholar who decided to use Google Translator was impatient, took the first one. Even worse was in one protocol I saw about seasonal affective disorders, SAD. People afflicted by SAD. The Google Translator said, Shiromani Akali Dal se Pidit. Now, Shiromani Akali Dal se Pidit afflicted. This could have created a huge furor. So please read the translated version. So back translation would be a good idea. You translate from one language to the local language, then give it to somebody else from local language back to English. If 80, that means you have the green signal to do the trial. But that means you have the green signal to do the trial. But how do you pay compensation if things go bad? You can have a group insurance, or the institution should have some arrangement. From every project, they can take a specific amount and keep it as a corpus for paying compensation. But the best thing would be to do a group insurance. Another word of caution for people who are working with multinationals. You have what is known as a clinical trial agreement, CTA. Please read the clinical trial agreement, read between the lines also, because the clinical trial agreement is loaded in the favor of the company. For anything that goes wrong, it is the responsibility of the principal investigator. And somewhere down in the fine print it says all the data that is collected, the ownership is with the company. So the presence of a lawyer in every ethics committee, legal expert, his or her job is to see the CTA and to ensure that it is not unfairly targeting the institution or the PI. The funding agency does not own the data. Ownership is with the participants, you are just the custodian. So there are issues with ownership. So they can't publish at will. So don't lucrative funding from a multinational organization and you sign, yes, I'm willing to do it, be careful, read the clinical trial agreement. Make sure that your lawyer vets it. Ancillary care should be available for patients who have any issue or participants who get into any kind of discomfort or other issues related to the clinical trial. This is something which I have already mentioned. It can happen even to postgraduate students in medical colleges or research institutions or PhD scholars, etc. So that compensation has to be created through means which may be group insurance ideally. Conflict of interest. What do you understand by conflict of interest? Anybody? Any member of the ethics committee here? Conflict of interest?
When you are actually indirectly, directly involved in that study, or you have some conflict. That is.
Okay, so conflict of interest, there are multiple connotations to conflict of interest and potential conflict of interest. If I am submitting a research proposal and I am an expert from the same institution in the ethics committee, clinician or a basic scientist, when my project is being deliberated upon or considered, I should step out. I should not participate in the decision making. In order to ensure that the committee knows, I have to tell them a priori, this is my project, it is funded by so and so, etc. That is one conflict of interest declared by a member of the committee. Now the other conflict of interest is PI may be somebody else. They may be trying some drug. They may own shares in the company that produces that drug. So that is the financial conflict of interest. That also has to be declared. Any drug or device. Now the first ever known and documented death from gene therapy was of a young 18-year-old boy called Jesse Gelsinger. Now he had a minor form of ornithine transcarbamylase deficiency, carbamylase. He could not metabolize protein till its end products. So he had a treat like half a burger a month. That was the maximum he can do when it came to proteins, but he was living a very guarded life. Now the gene therapy that was targeted to him, participate in this gene therapy experiment. I'm not sure, I think it was University of Pennsylvania or Philadelphia, not very clear. He said he will do it because he was told that if you participate, people with this disease, other youngsters will benefit. What they did not tell him was that there were some issues with the animal experiments. And he was not the right target for that therapy because he had a milder form. So he ideally would not even have been included. But since he was healthy, they chose him. He died of a very severe anaphylactic reaction after the gene therapy was delivered. And the allergy was to the probe that was used for delivering the targeted gene. And after that, the hangama occurred. But the biggest part was, apart from having withheld information from him, the dean of that institution had shares or was marketing that probe. So then it made a lot of noise eventually. So I think conflict of interest has to be declared. And then there is something like potential conflict of interest. Please raise your hand if you know what cricket is. Do you think Virat Kohli is an excellent batsman and an automatic choice in the Indian cricket team, even though he is retired? Yes, no? Yes. Now if you, it's a no-brainer, it will be one of the finest. Now if you are the father of Virat Kohli and now you are a selector, no matter how good or how bad Virat Kohli is, people will say Virat Kohli ke baap ne usko team mein dal diya. That is potential conflict. I mean, we know the talent. It is absolutely. So that is conflict of interest. So how it is visually perceived is worse than what it actually may be. So please be careful of conflict of interest and declare conflict. Conflict of interest doesn't mean the committee will say, no, you cannot do this research. If you have, you have to make sure that people know that there is a conflict of interest, and conflict of interest can be mitigated. Equitable distribution of resources, equitable when it comes to selection of patients or participants in research, etc. Community engagement. Community engagement, especially when you are doing work in the tribal belt or with vulnerable population, or even when you are sharing information with the community. And yesterday we were talking about implementation research. In implementation research, it becomes extremely important to engage the community. This I have already mentioned, extremely important. Now, post-research access to the benefit of the drug or device or the intervention, which may be a behavioral intervention, and how it is shared is the duty of the principal investigator and the funding agency. The ethics committee has nothing to do with post-trial benefit because it's an indirect benefit. But they can ask the PI to please give details of the post-trial benefit. You can't say we'll do research on some drug and when everything is fine, we will not give the drug to the person. So that's something, an arrangement can be worked out with the ethics committee. Now these are the various reasons why everybody should follow ethics. You may get into trouble with the law. People are very, very aware now of their rights. And because of the culture, you don't have to take photographs. We will share the presentation. So, corporate culture where you are not a participant, you have a right, especially when it comes to healthcare. You can get sued if you have not done due diligence and followed all the ethical procedures with the participants or with the patients. So please be very, very careful of what you do with your, I have a little time. These are the few tick boxes that you must do if your research is supposed to be called ethical. It should have value. The first job of the ethics committee is to ensure that scientific validity is there. If the research protocol does not follow a good methodology, don't waste money because those results will never be. So please make sure that your study is valid. And then of course, fair subject selection, risk-benefit ratio, independent review by a committee, not by members of our own who don't read and let him do it. It happens in a lot of institutions. Informed consent, please go back and read details about informed consent. Very, very important. It is only based on informed consent that the participant make an informed decision about participating in the research. Informed consent should be in a language that the participant understands, which technically should be class 5, class 6 level school, class 5, class 6 level education. It should be in a language that the patient understands or the participant understands. If you are talking about Hindi, it should not be Doordarshan Hindi, it should be Zee News Hindi. How many of you understand Doordarshan News totally? So those of you who, if at all, did you understand? We have one liar here. I'm just kidding you. So very, very, clished Hindi, you should talk colloquial, what the patient or participant will understand. Now, more importantly, there are issues with minors. At what age group does get known as a minor? Pardon me? Less than 18, that's right. Now, if you have consented somebody less than 18, it has to be consented through the parents. For high-risk trial, both parents. The day the person becomes 19 during the course of the trial, and you have to consent the person again as an adult. So that is one requirement you take their date of birth, you have to be careful. Now, more importantly, there is something known as assent. An 18-year-old understands. So 12-year-old upwards, for every minor, you will take parents' consent, written consent. But assent has to be taken from the minor. 12 to 18, they understand pain, they understand things. So when you talk to them, their written assent along with parental consent. So written assent along with parental consent. Between 7 and 12, it is verbal assent which is recorded on the parental consent. So it's not just signature of the, a lot of people don't know assent. So the committee will ask for assent. So I think in fact, informed consent is excruciatingly important. Each and every one of you must go and see how it is done, how the translations are done. And then of course, respect for subject which we have spoken about already. Now, the ethics committees, where composition, role, responsibility, procedures and accountability is all here in this presentation, I will appreciate it if you tell me when to stop. What is my timeline? Another couple of minutes? 4 minutes? We'll make it 5. The first and the most important duty of the ethics committee, all the members who are sitting here, please take note, and all the researchers, please take note. The first and foremost duty of the ethics committee is to ensure that every project they get has to be approved. Do you agree with me? Yes.
Especially for subjects like us, disciplines like us, all research may not require ethical clearance. Is it true?
I'm not talking about those. There are some which may not require ethical clearance. When you're doing any research on human subjects, even gathering information, question-based, you need consent. Be aware of that or you can get into trouble. Now the issue is, my question was, should the ethics committee approve every protocol they get? The answer is yes, but there is an asterisk on the approved. Supposing you have submitted a protocol which has got a lot of violations. So my duty in my endeavor to approve it would be to point out to you, okay, Dr. So-and-so, the following issues are raised, please correct them and resubmit and we'll approve. So what I mean here is, when you get a proposal that is flawed and has major or minor ethical issues, the principal investigator should be informed, they should be counseled, guided or even educated about those issues, then let them make corrections and resubmit because research is important. Meaning thereby also that you cannot refuse a proposal without giving a justifiable, valid reason. If it is absolutely trash, you can say, sorry, will not be done, rejected. But before rejection, if there are any issues, infringements, ask the PI to correct them and resubmit. So basically, you would be enabling research and correcting those unethical things at all. And you got to review, approve, and continue oversight. Your job is also to continue oversight while the trial is going on. It's not just that you are given approval and then you forget about it. All research activities involving human subjects, even questionnaires, Google Forms, they are all under the jurisdiction of the IEC. The other problem is, if you want to publish, every publisher will ask you, do you have the permission of your institutional review board or ethics committee for publishing this? So it is safer to take ethical clearance for everything you do with human subjects. You may get waiver of consent if it is minimal or less than minimal risk trial. But that is the job of the ethics committee. As a PI, you can in your application say that since the risk involved is less than minimal, and since the identity of the patient will not be revealed or they are anonymized, I am only collecting information, I may be granted waiver from informed consent. The committee will consider that. They may even expedite that review. So these are the responsibilities of the ethics committees. Their credibility or effectiveness in fulfilling their role has been challenged severely. So I'm going to just give you the challenges that happened. Now these are a few events. I'm not talking about Tuskegee, Willowbrook or the Aboriginal women, or there are a few other trials. What happened in India? In 2030, the parliamentary committee report says that more than 25,000 girls, this is that human papilloma virus vaccine, right? There was a huge upheaval in this country. In 2013, Supreme Court came down very heavily. All clinical trials in the country were halted. For almost a year, there were no clinical trials. All the funding was withdrawn. It was from researchers. In Andhra and Gujarat, there were almost 10,000 informed consent forms signed here and 6,200 here. There were some almost 2,000 thumbprints, almost 4,000 thumbprints, and almost 3,000 here, and the consent forms were signed by hostel wardens. Hostel wardens have no business to consent on behalf of students, nor do principals of colleges, except for parents, because these are minors. Except for parents, nobody can even permit medical health checkup in school, now home for permission. So these things were going on, but they were terribly wrong. So please note, and there were more. In 69% of the total form, there were no witness signatures. Informed consent has to be signed by a witness also. There were no signatures of witnesses. There were no dates on the informed consent forms when they were scrutinized. There were thousands and 25,000. One person had signed 7 forms. These are observations from that parliamentary committee report. Principal investigator had not signed at all. Parents' names and signs were not matching because some things were, when the committee had asked submit, then they did some hodgepodge and submitted. And date of vaccination was preceding the date of consent. They gave the vaccine, then they realized that a parliamentary committee is on, then they ran around getting the consent. So the conclusion was that institutional ethics committee are more concerned with protecting the institution and the researcher than the participants. So members of the IEC, your job is to protect the participants. Focus on just 2 things. Informed consent process, if that is perfect, things are good. Risk-benefit assessment. If that is fine, the trial is good to go. You can tell them to follow the procedures. Any change, supposing you have done blood sampling and you want to do 10 tests, committee has approved. After 2 months, you realize that 2 more tests can be done and should be done. This is known as variation in the protocol. If any variation or change in the protocol, you have to go back to the ethics committee. They will permit and go ahead. Without ethical committee or ethics committee permission, do not change the protocol. Yes.
Sir, a researcher, that researcher is a doctor and she has done, she's going to do a study in her private clinic with her patients. Now, she is applying to National Ethics Committee of another institute. So, what is the purview of that institute? Should they take it or not?
Excellent question. Now, here is a lady who is working in a private clinic, wants to do some research, but does not have an ethics committee. Because it is mandated that every institution should have an ethics committee if you want to do clinical trials, any research on human subjects, even bioavailability trials, etc. Now, in small institutions who are going to do only one trial, they don't need to form a committee. They can have an MOU with an institution. Similarly, that lady can have a memorandum of understanding with the institution in the locality, preferably a medical college, that my research will be overseen by that. So a memorandum of understanding is all that is needed, but you cannot do a trial without an ethics committee clearance.
Permission from AIMS New Delhi. It should be within. Please keep that in mind.
Sorry, beg to differ with that. The guidelines say very clearly the rule for 50 kilometer applies only to the independent ethics committees and not to the institutional ethics committees. And current independent ethics committees registration portal is not functional. It's going to take a little more time for ICMR to begin that portal.
I think you got that. One is, we are talking about independent ethics committees. You can reach an independent ethics committee. There are a few in Mumbai, very robust ones. They take a very stiff charge. They may charge you 50,000 plus to review your protocol. So you may approach an independent ethics committee, but ideally an MOU with a nearby institution would be the ideal thing to do. And when you talk of multi-centric, because he mentioned AIMS New Delhi, multi-centric trial. Supposing you get a trial which is being done in your institution and you submit it to your ethics committee along with the clearance given by AIMS New Delhi Ethics Committee to their PI for the same trial. Ladies and gentlemen and members of the ethics committee, just because AIMS New Delhi has given approval for a trial does not mean that your committee should also blindly give approval. You go through the merit of the trial in your area, the procedures here, and see if everything is comfortable in this place. Especially in tribal areas, especially in the northeast, there may be a few regulations which are different. So your protocol has to be tweaked to ensure that the cultural practices and the sensitivities and sensibilities of the local area are not offended.
In that sense, suppose I am in a private hospital, then I want to do something in research. If I include one of the investigator as my co-PI, then I can take the permission from the institutional ethics committee, then that study can be...
What she is suggesting is a jugaad. That the young lady there had asked the same question as a clinician in a private clinic. Have an MOU with an institution. This is a little smarter. Let me take an investigator as investigator from that institution so that the committee agrees. People do that also. But why do you want to include somebody if it is not absolutely necessary only for the convenience of the permission? Then you better sign an MOU. Then the entire thing is your responsibility. Otherwise, nobody will hijack your project. Tomorrow you have a co-investigator in a college and he publishes, then you are sitting in your private clinic and twiddling your thumbs and fingers, and there is very little you can do because you already handed over him as PI. So when we seen in 31 different publications, it was seen that these were the infringements in various approvals by different ethics committees. Conflict of interest, societal issues, faulty signatures, faulty functioning, training deficiency, and regulatory issues was this kind of a thing. So the issue is that the ethics committee, apart from giving permission, has to ensure that they monitor the trial. If there is a serious adverse event, it has to follow a protocol, proper timeline. If things go bad, you are also accountable. So please keep an eye on the trials that are going on. This was published in JAMA. Ethics committee, because they play the mission creep, they are known as the police. People are upset with them, including the PIs. Excessive paperwork, inflexible interpretation of regulatory. They'll say, no, the rule says this, I will not permit. That is not how they should function. Attention to inconsequential details. They'll pick up some arbitrary thing and start hassling the PI. And most of the PIs know when they have to answer multiple questions, things are not right. This seriously affects or stifles research productivity. So members of ethics committees, please be reasonable. Assess the total procedures, assess risk benefit, guide the PI. It has little to do with protect. All the things above does not protect the research participation. It is just creating hurdles. So ethics committee members should not consider themselves as policemen or regulatory authorities themselves. Remember, you are part of that same environment. You are also a researcher. That's why you are there. And that's why there is a member from the lay community who understands things from the non-medical or non-research perspective. They may point out something which is very trivial, but may be something good for the participants. So these are the key challenges to any ethics committee. Some of these are your purview, some of these are not. When scientific fraud, etc. occurs, it has to be reported and only then acted upon. Now, multi-centric ethics committees have been formed. This is a recent thing. We were streamlining that for a long time. Because one thing says that all multi-center research should have the same protocol. Changes might be required in different areas, so there are a few challenges there. These are the elements of an ethics committee. And this is very, very well laid out in the ICMR guidelines. Now, I will just give you a very brief view, maybe less than a second each, of the number of slides that are here, which are all part of the ICMR guidelines with a few comments. This is the rationale for setting up the committee. It should have an independent review, and informed consent is a very, very important component of the rights and welfare of human participants. You need to consider the following thing when you are doing a review of a protocol. There are two things missing here, which are extremely important, and most ethics committee members are careful and scared of. Please pay attention to that. These are your responsibilities. These are the key players. Watch out for the sponsor and CR when they make the clinical trial agreement. This is the composition of the ethics committee. This is given in the guidelines. As per the revised Schedule Y, you need a pharmacologist and a legal expert, especially. These 5 are important if you are doing a clinical trial. Clinical trial review has to be done by a registered committee. Make sure that your committee is registered, and the member secretary ensures that they know when the permission after 3 years is going to expire. For example, on 31st of May, AIMS New Delhi Ethics Committee tenure expired, the permission. So now they are applying again. It should have been applied on the 1st of May when there were 30 days to go. So please keep an eye on it. It happens very often. You don't renew and you are still clearing trials and you get into trouble. So you should know very clearly when your renewal date is near. These are the important elements of an ethics committee if it has to function properly. I think I'll stop with this, because everything given here is part of the guidelines. This is going to be circulated to you, and you can look into this and see what all is. These are the terms and references, terms of appointment, registration requirements. Registration requirement can be pretty painful. There's a lot of documentation. Start doing it beforehand. Registration is only for 3 years, so please know when you have to renew. Whenever there are changes in the ethics committee, you have to inform. So many members have left, so many new members have come. Some members may be kicked out for not attending. There are rules that are again laid down. How do you maintain records? Everything has to be maintained. How do you communicate with each other? How do you communicate with the principal investigator? How do you communicate with the regulatory authorities? Everything is very clearly laid down. How do you train? It is mandatory to train the members of the ethics committee. For example, this could be called as a GCP training session for the RMRCNE. Administration and management is very crucial. You should have a separate office for the ethics committee. And this is the functioning, the flow chart, what you should do from calling the proposal, assessing that they are scientifically sound, then the research benefit and the processes for informed consent and subject whether treated equitably. The first and foremost is scientific validity. PI may shout your business is only ethics, do not check the scientific validity, but any project which is not scientifically valid is unethical. So the first job of the ethics committee is to ensure that it is scientifically valid. This is what you do before the meeting, during the meeting, and after the meeting when the decision have to be made. This is the scheme. How do you follow from fixing the date to revised proposal to be reviewed? Reporting of serious adverse events and subsequent reporting in case of death, who does what and to whom and in what timeline. This is also clearly mentioned. Responsibilities of the chairperson, responsibilities of all the other members of the committee are laid out here. Division of duties, legalities, social issues, special concern and general issues apart from the science where you have the clinical or basic scientist. You have a scientific review and an ethical review. You may have categorization of exemption, expedited or a full review. Ideally people do full review. Expedited can be for less than minimal risk and exemption also. Expedited can also at times be in natural calamities and disaster where consent is not necessary. Minimal risk is exempted. These are a few examples of exempted ones. Expedited review, when there is a little more or no more than minimal risk. And this is when, in emergency situations, expedited review is done. For example, in a tsunami, earthquake, etc. Full review for other routine processes. Monitoring procedure, you should monitor basically the informed consent process and a few procedures if there are any intervention, and assess the risk. These are the categories: high, medium, and low risk. Monitoring, how it should be done and by whom. Periodic and continuing or interim. Protocol deviations and violations have to be mentioned to the committee, and the committee will take note of it and give remedial measures. Everything should be written and documented, from your membership, governance, review procedures, SOPs, various versions of the SOPs as and when they come. You have these checklists like the ICMR checklist. Follow the same checklist. You may have conditional decision making. How do you communicate to the PI? That is also given. Communication to the Drug Controller General of India, communication to the patient, and format for according approval. In the approval, you have to mention received your protocol and the following documents 1, 2, 3, 4, etc., including informed consent in various languages. The thing was considered by so many members and it was approved, or is conditional to changing your informed consent form or including this or whatever changes you have suggested. This is approval. Communication to participant. You may need to communicate to the participant. The participant has a right to approach the ethics committee if something goes wrong. For international collaboration, you need to have, and since we've had a discussion, if there are any more questions, I'll be happy to field them. Otherwise, thank you so much for your forbearance. I see one hand going up. Before I wrap up, I'll take this question. Yes, sir.
I just had a very small query. In case of any multi-centric research, where we actually incorporated a lot of hospitals into it, once there is a full committee report in the parent institution, do we have to have subsequent committee clearance at every hospital as well?
Every multi-centric trial, if it is done in 10 sites, all the 10 sites are trial. And the new ICMR guidelines, there is the lead multi-centric trial. For example, if the trial has been initiated by you and there are 11 other sites, so you have a central mechanism where you may have one ethics committee, still being a little fine-tuned, but yeah, we are working to that. But make sure every single site where clinical trial or research on humans is being carried out has to be cleared by the authority in that locality.
Just a suggestion that when the risks, basically I'm working on cancer patients. We collect the endoscopic resections and OT samples. Basically, when in our country, the late reporting of cancer cases are most frequent. So when the subject actually has risk patterns like different at different instances, so what will be the best practice to sort of, for the informed consent?
Late reporting, when you identified that the report is late already.
Yeah, most of the patients are late reporting.
Okay.
The risks are like, it has to be assessed by some physician or someone. So, sir, what should be the best practice to?
See, there are two ways of looking at it. As a clinician, the best practice would be to inform the patient about what exactly the risks are and how, they are going to be personally affected. But as a PI with a project that is approved by the ethics, it is not there in your protocol. So when you're dealing with a research participant, you are a doctor and treating a patient on one side. You are a researcher and the same person is your participant. Don't mix the two. So for late diagnosis, the counseling that you do to the patient is your job even as a healthcare giver. The protocol may not have that in your research. And there's very little you can do because there are going to be a wide variety of patients. I think I haven't probably understood your question very well. So we can discuss this one-on-one. I think I'll call it a day here. If there are no more questions, I'll be very happy. If there are questions, we'll take it up over tea. Thank you.
Thank you so much, sir. Thank you so much, sir, for your session, which is very interesting, informative, and engaging. A very detailed and elaborate presentation, and you have addressed practical issues and examples with light-hearted humor. Thank you so much, sir. And we would like to hand over a small memento as a token of our love and gratitude towards sir. So before handing over that, this particular memento, I have forgotten to explain yesterday. This has a very important historical significance in Assam. It is called Ranghar, which is a historic two-storage pavilion located in Sivsagar district of Assam, which is around 2.5 hours drive from here. And it is considered to be one of the oldest amphitheater in Asia. This was built more than 250 years ago during the Ahom era in Assam. This structure is unique. It is made of thin baked bricks. And for charging or for gluing the bricks, the motor used was a mixture of rice, pulses, fish, and eggs. So there was no cement or no lime or no other adhesive chemical things that is used. The only thing that is used is rice, pulses, fish, and eggs combination that is holding the bricks together. I wish we could have taken you all to this site, but next time maybe when we conduct this kind or another kind of program, we would try to plan such kind of outings. Thank you so much, sir. So once again, I would like to request Dr. Naren Mehta, sir, to kindly come up on stage. And I request Khan, sir, to kindly hand over, I request Director, sir, sorry. Director, sir, to kindly hand over the gift to sir. Thank you so much, sir. Now we would break for tea. And actually, we are running behind our schedule. So I request all the participants to kindly have a quick tea and come back by 11:15. 11:15, we will start the next session. That is on Introduction to Research Methodology. This session will be taken up by Dr. Prem K. Mani, sir. Thank you all. Yes, we would like to begin our next session. This session will be on Introduction to Research Methodology. And for this, we have with us our eminent resource person, Dr. Prem K. Mani, sir. He is the Vice Dean and Professor, Division of Epidemiology, Biostatistics and Population Health from St. John's Medical College and Research Institute, Bangalore. So I would like to invite Prem K. Mani, sir, to kindly take his time. So pointer is here. So if you want to
Good morning. I'll be covering what's called the Introduction to Research Methods. You must be wondering why it is titled as such. I'll try and cover the practical aspects because you've heard quite a few sessions yesterday and today. The traditional way of structuring the research methods course is what the schedule that is there given to you yesterday and today. But I have a feeling increasingly you not only need to learn how to conduct research, which means to add to the body of scientific knowledge that is already there, but increasingly we have to filter out too much information. That's the problem that is there because most of the research methods course schedules are actually what we had 10 years back on how to add to the existing body of knowledge. But as scientists, we also now have to figure out how to make sense of the information, which is a information overload actually. And the third one is also to be able to learn how to actually actively counter misinformation or disinformation, which is also there too much. And what are the key steps? Textbooks won't tell you this, but essentially we must have a little bit of curiosity about the world around us. And when I say that, it means we must be a little curious on some things which we want to identify as a problem, study that, and find a solution for that. So that's the first step for the research. You have to then figure out whether you want to be just observing or you want to actually intervene. The next one is I call it materials and methods, and then you should be able to get results and dissemination. Those are the usual standard definitions of research and medical research that you can look up. But what we are going to look at is the details of the scientific methods. And I like to introduce this cycle as a starting point for how to think of it. You should be able to do this PPDAC, which is identify a problem, make a plan, collect the data, analyze it, and then come to conclusions. And you will see that it's not linear. By the end of the research, we always finish saying, I have found these things, but there is scope for more research because everything doesn't come to an end by the end of one person's research study. Usually it's a cyclical process. We try and understand one part of the problem, figure out something which will be of use at a given point in time, but also there's more to build on that. That's the details of the different aspects of this cycle. So the first one is we should be able to define the problem. If you're figuring out that you have been treating tuberculosis for many decades, the thought that comes to our mind is if it's a disease that is known for the last several thousand years, why is the prevalence of sputum positive tuberculosis the same as it was 50 years back? If you are treating anemia, the question that will come to your mind is, why is the prevalence of anemia the same as it was among pregnant women and children under the age of 5 years as it was 50 years back? Okay, so some of these things are questions that should come in our mind. So that's the first part of identifying that there is a need to study something. There's a problem in my department or in my field which I can actually study a little bit in detail. And then I must make a plan, which is what we call the methodology section, how to be little rigorous, how to be detailed, and how to be very focused. So I must, if I want to go to Big Boy, I must make a plan and I must decide whether I will go by car or whether I will go by bus, what strategy will I undertake. That's the part which is the planning. Once the planning is done, I must collect the data. It can be quantitative data, it can be qualitative data, it can be a mix of the two called mixed methods. I must be able to figure out what will I do with all the data that I collect. So that's the analysis plan before as part of the plan, but also after the data is collected, I must clean all the data, analyze it, and be able to make sense of the data. You must have heard these terms earlier. There are different types of research, primary and secondary. Much of what we do are taught to do is what is called primary research, which means that I am collecting the data. As opposed to secondary research is where data is collected by somebody else. I have access to the data. I am only analyzing the data. The course contents for yesterday and today is largely for primary research, not the secondary research. Pratap was talking yesterday about doing a systematic review. So that is a secondary research where we take the research findings and try and make sense where somebody else has done the research. The other words that you will hear are quantitative research and qualitative research. Quantitative research is usually where by the different items in the center, we can try and differentiate quantitative research and qualitative research. Both are different approaches. The purpose is different. The data types are different, as you can see, where there is numbers on the left side. On the right side, you don't deal with that many numbers. The approach to analyzing that, interpreting that are all different. And also, as Dr. R.M. Pandey was saying, the bulk of the training for this research methods workshop is for the primary research and it's for quantitative research. The next thing to decide is, are you going to be an observer? That means you're going to admire a problem, or are you going to come up with a solution? So normally we call that observational study designs and experimental or interventional study designs. As you can imagine, the one on the left side is the simpler one to do, relatively speaking. And when you're in your early part of the career, we always start with doing a observational study design, and then we move on to the experimental or interventional study designs. So normally, when you're problematizing it, so that's the first part which I was talking about, when you're identifying a problem, in your actual practice, is that if you're walking in the corridor and somebody asks you, what is your area of study, what should you be able to say? You should be able to answer in a single phrase. So in the corridor, you don't want to explain too much in detail, so you'll say, I'm interested in studying adolescent anemia. Okay? But that's not enough when you are writing a protocol. In the protocol, the first part, what we normally call the introduction, I like to draw the diagram with an inverted triangle. That means in the introduction, I should be able to write about the topic in general. So as an example, if I'm talking about, if I'm interested in studying anemia in adolescent girls, because usually anemia is studied lots in pregnant women and children, but not so much in adolescent girls. So then I say, oh, maybe they are all becoming pregnant with already depleted iron stores, and they're already anemic, and therefore that is why the problem of anemia is too common during pregnancy. Maybe if I can start identifying before they become pregnant, during their adolescent period, maybe I will be able to sort out that problem. So then I start thinking differently, and then I say, I want to study adolescent anemia. So in the introduction, I should first be able to start saying, anemia is a major problem, is a large public health problem, and I'll give a citation from WHO. And then I say, anemia is common in India. It is already found from the National Family Health Service that anemia prevalence in different age groups is so common. And then I say, anemia is very common in Assam. And I get the statistics for Assam state from the different sources available. Anemia has been studied largely in rural areas, but not in urban adolescent girls. And that's towards the end of the lower part of my inverted triangle. I start with what is already known. I give the data to say that I have read up, I know what I'm talking about, and then I identify what is called the research gap. Towards the end of my introduction section, whoever is reading my protocol or my proposal should be able to see clearly through my eyes what is the problem that I have identified. So by the end of the introduction, towards the end, I should be able to say what is the need for this particular study. And that's the identification of the research gap, which I should then convert into a research question. Do any of you want to share any research questions from your proposals? Does anyone have a question that you've already written in a previous application for any grant proposal? Nobody has written a proposal? Grant application? Or for any funding or a thesis?
Sir, I have applied for the intramural grant from NIGRIMS, and the topic is a pilot study on the effectiveness of EMG guided versus blind method of Botox injection. Botox question will be more effective whether we inject blindly or EMG guided.
So did you get the idea that she has in her mind? And when you're starting to think what is different in the two different arms or different treatments, the way to write the research question is to remember that you remember what was told yesterday, that PICO, have you heard that abbreviation? So while writing the question, start thinking of ending with a question mark. So that's the first thing to remember. The question should read like a question mark to a person who is reading through the proposal. The second one is, roughly you want to make sure that you are mentioning which is the population or the patient group which you are interested in. And if it's a interventional study design or experimental study design, you will say what is one intervention, and you will talk about the usual or current intervention, which is called the control intervention. Control, and also what is the outcome. If it's an observational study, you don't have an intervention and a control. And therefore, as opposed to her study, how will I write my question for adolescent girls? I won't have a comparison arm and an intervention arm. I'm just trying to find out how common is the problem of anemia amongst adolescent girls in Bangalore urban slum. So mine is a question I'll put a question mark at the end, and I have got the P and the O in my question. I now have to go to the next step. After writing the question, I should write, those are just some finer details on what is it that I want to describe or find out as part of the question. So on the left example I've given for observational studies, and on the right side I've given for a experimental or a interventional study design. The other thing to remember is the next step after writing the research question is to write a study objective. And it's good to learn from the mistakes of others. On the red indication, I've tried to indicate the usual mistakes that are made while writing study objectives. And on the right side, I've tried to give some pointers to how to make the study objective sharper and better. The first point to remember is not to write too many study objectives. Okay? If you're starting off first project, preferably have only one study object. As you start getting little more senior, you can increase the number of objectives, but always be clear to have one primary object. You can have more than one, but then you should be able to identify if there are two objectives, you should be able to identify one of them as the primary object. The reason for that is the study design may or may not be the same if you have too many objectives. And for each objective, the sample size will keep differing. Okay? So let's take my example. I want to study how common is anemia among adolescent girls. I'm also interested in finding out why are they getting anemia. Because if I study amongst a group of adolescent girls, not all will be anemic, some will be anemic, some won't be anemic. So my second objective is, I also want to find out the factors associated with anemia. So in my mind, there are two objectives. How will I write my two objectives? The first one is to find out the prevalence of anemia, and then the second one I will write, I want to see what are the factors associated with anemia. I can write them as objective 1, objective 2, but even better will be I'll write the first one as primary objective and then write the word secondary objective and put the second objective as the secondary objective. The first objective, I will use a different formula to calculate how many people should I study to come to the correct conclusion about the prevalence of anemia. I can do an observational study design. To find out the factors associated with anemia, I can do a observational study. So fortunately, the study design is the same for both objectives in this example. But the factors associated with anemia, why are some girls anemic? What are the factors? Causes for anemia?
There can be hemoglobinopathy, malnutrition, food, deficiency in the food that I take, other infectious diseases.
There are multiple causes, and when I'm trying to study factors associated with anemia in my setting where I'm interested in, I have to do a review of literature to find out what are the factors that are already been identified as associated with anemia. I should be able to find out either the odds ratio or the relative risk from previous studies and calculate a sample size for the second object, which may or may not be the same as the calculated sample size for the primary object. If I have to satisfy both my objectives by doing the study, I should then choose the larger sample size. So if one sample size comes to 250 for one objective, And for the second objective, now it comes as 800. I'm now stuck. I have to do 800 students or adolescents I must study in order to be able to answer both objectives. So that's why we now say, choose which of them is what is of primary interest to you. If I now say prevalence is of primary importance, I call that as my primary objective, and the secondary objective will become the factors associated with anemia. Once I clearly demarcate like that, I can now go ahead and collect only 250. Primary objective must be satisfied by the end of the study. Secondary objective may or may not be satisfied by the end of the study. The first point to remember. The second point to remember is I must use what are called scientific measurable language words while drafting the objectives. I can't say to find out prevalence of anemia. I can't use those words. I'm not playing a hide and seek game like a 6-year-old child saying, I want to find my friend where he is hide. Okay? So for drafting objectives, there are some preferred action words that are recommended. I must write to estimate the prevalence of anemia amongst adolescent girls in Bangalore urban slums. That's a full objective. It's got a measurable word. What do I mean by a measurable word? By the end of the study, I will be able to give you a point estimate. That means I will tell you 28% of girls are anemic. I didn't know that before starting the study, but by the end of the study, I will give you a number in the form of a percentage. The words on the right side are not appropriate for quantitative studies. They are good for qualitative research. So usually for all our observational study designs and experimental study designs, I must use the words on the left side. Yesterday we also heard an example of one of the experts saying if somebody is doing a comparison study between two drugs to see whether drug A or drug B which one is better. Do both of them have benefit and harms, both the drugs? Yes. So I want to study how safe is one drug A, how efficacious is the drug A, and compare it with another newer drug. The wrong way to write the objectives is to first say to study the efficacy of drug A in the treatment of typhoid fever in children. Objective number 2, to study the efficacy of drug B for treatment of typhoid fever in children. Third objective, to compare drug A with drug B. That is not required. All I need to do is choose the appropriate recommended action verb from this side, on the left side, and state it as one single objective with PICO all included in the single objective. So I will write the objective as to compare the efficacy of drug A versus drug which is the control, and what's the outcome that I am interested in. All of that in one single objective, and I've got a measurable action verb. And by just using the word to compare, that word itself is now indicating to you, I have to study safety and efficacy of drug A, I have to study the safety and efficacy of drug B, and I have to make a comparison. If drug A is 80% effective and drug B is 72% effective, I can tell you and compare and tell you drug A is 8% better than drug B. So I'll give you a estimate by the end of my trial. The next thing to remember is, of course, you have to do a review of literature. Key things to learn while doing a review of literature, you have to, in parallel, figure out how to improve the scientific English for your own field. Okay? So I'm just giving you an extreme example to make it little funny. But if you are in the field of infectious diseases, you have to learn the words that are very specific for infectious diseases, whether it's infectious disease epidemiology or infection transmission probabilities or communicability or virulence of the organism. All the scientific words associated with your field, you have to read up so that you are mastering the English of your own field. The second one is to be able to do what is called a smart technique whilst doing the review of literature. Take out your phones, go to Google, type PubMed quickly. Practical exercise. Go to Google, type PubMed, open PubMed. And if you opened PubMed, type anemia, Assam, click, search. Because you're interested in studying anemia in Assam, you're saying, I want to find out what is studied till now about anemia in Assam. How many study results? 109, okay? Anemia, Assam, only 2 words you're writing and search, okay? Instead, now you do what is called Boolean operators, okay? This is a search. What do? You write anemia within double inverted commas. Write anemia within double inverted commas, space AND, and in capital letters, space Assam again within double inverted commas. What's the number? 89, okay? Number keeps dropping. You can do the same. I can now do it anemia and Assam and prevalence. Anemia within double inverted commas, space, and within double inverted commas, Assam, and then again A and D in capital letters, and then within double inverted commas, prevalence. How many? 35. Once you've got this, now you can remove all the double inverted commas and the ands and just write only anemia, Assam, prevalence. Just write the 3 words and search. $65. So can you see the difference in the numbers? If you use the traditional way of how we search on Google, PubMed doesn't understand what exactly we want. It just gives words wherever it sees anywhere in the title or abstract. If it sees prevalence, it's giving us that word. If it sees anemia, it's giving us that word, that article. If it sees Assam, it's giving us that article. It doesn't know that this person is specifically talking to me in one particular language. So Boolean operators, that is words like AND, or, not, I can to restrict the number of articles that I must go through. In addition, on the left side in PubMed, when you are searching, we can even restrict the type of articles. If I'm starting newly, I will just click review articles and then use Assam, anemia, and prevalence. Basically, what am I trying to do? I'm trying to be as lazy as possible so that I have a small number which I can manage, because I must go and do other things in the evening. Okay, I can't be reading through 140 articles or 1,400 articles. So I must be smart while searching, figure out what is very important, and learn how to talk to PubMed. There's also a third option called a mesh terms, which if you click on the top, it will specifically use only mesh terminology, okay, medical subject headings. While looking at the different types of study designs, ICMR now calls for proposals and grant applications under these headings. They want to ask you, please tell beforehand what type of study are you going to do. And they've classified it like this: descriptive research, discovery science or discovery research, development research, and delivery research. So we should click one of those buttons while applying for ICMR grants. So it's good to be clear in our minds, what is it that I am looking for, this is slightly different from traditionally whatever we are taught elsewhere. So it's good to map the different study designs that you know to what currently ICMR looks for. In the center, I have tried to show traditionally what is called the hierarchy of epidemiologic study designs. From the simplest at the bottom, the more complex, and it's also sometimes considered that you are closer to the by doing the study designs at the top. Okay, so that's why it's called the hierarchy of study designs. And you will see randomized control trial, non-randomized trials are all at the top. And in the green, in the center, the three study designs are what is according to ICMR when they call for proposals. That's the descriptive study that they want or they're looking for. So usually in descriptive research, as per ICMR, it's the cross-sectional study or descriptive study. Little more complicated, the case control study, and then the cohort studies. The ones in red in the center are not under the descriptive research that ICMR is asking for. The next one is discovery science, and that is mostly the basic science or the lab sciences. So that's a different bucket. So first you have to describe the problem. In parallel, there is the cellular level or molecular level understanding of the different diseases, which is usually from lab sciences. So that's the separate stream. Once these two are done, we have discovered something, we have described a problem, and then you want to develop a solution for that problem. So development research is usually all what we traditionally call green ones in the center, the interventional or experimental study designs. So you say, I have understood this problem, I want to make a new kit, or I want to compare two different drugs. There's a new drug in the market, I want to study the new drug and compare it to the old drug that I've already been using. I want to do it in the form of a trial. I want to see whether an educational package will improve care. I don't want to randomize. So I can then do a non-randomized trial of a new health education intervention. All of these are slowly developing the field so that we have a solution. All of these come under development research. I can even then propose a systematic review or a meta-analysis to take the field even further based on all the existing old studies. Once all of this is finished, how does science proceed? When there is enough evidence, after that it will get into what are called clinical practice guidelines or standard treatment protocols. Once all of this is done, it will get into if the disease is of national health importance, it will get into the national health program. The next part, which Vishwajit was talking about yesterday, is once it's part of a national program, how well is it delivered? There are gaps in implementation, and therefore somebody may be interested in saying, I'm not interested in an old epidemiological study design, I'm not interested in lab science, I'm not doing trial. I want to see why the program in my district is not functioning optimally. I then will do what is called implementation research or operational research. These two are the delivery research. That means there's a problem in delivering the proven cost-effective interventions at a large scale. Once I have written the introduction, written down the research question, converted the research question into a research objective, keeping in mind that I'll have one primary objective and shift the others to secondary objectives, I must identify my study design. When I use the word identify the study design, it means that there are different options available. I must pick the study design which will answer my study objective, most appropriate for answering my study objective. Read through the first one. What study design is that? First, tell me whether it's observational study design or interventional study design. Observational. Under observational, one answer case control, one answer cross-sectional. As the outcome and exposure, two things you have to look out for in different study designs. What's the outcome and what's the exposure? Which is the exposure? Circumcision is the exposure. Outcome is HIV status is the outcome. Okay. How do you identify exposure and outcome? Exposure should precede the outcome. For that patient, biologically speaking, exposure should be there first, and then you are trying to find out what happens after the exposure. Here we are trying to see whether circumcision is associated with subsequent development of the HIV positivity or not. Here there are two groups that I am comparing. Some are HIV positive and some are HIV negative. And I am looking into the status of circumcision for both of them. The only difference is I didn't tell you that I got so many positives and so many negatives. And therefore, this is not a case control study. This is a cross-sectional study. I can't fix the number of how many of them will be HIV positive when I go to a clinic. I have no control over how many of them are going to be positive, how many are going to be negative. In that STD clinic, maybe 5% are positive. So I have 5% of them positive, the remaining ones are negative. So I call all of the remaining ones as the group that I am comparing. Okay? Then read the fourth example. Observational or interventional? Observational or interventional? Observational. Now what study design is this? This is a case control study design. Okay. What's the exposure? What's the outcome? Sunscreen use is the exposure, and then I want to find out if it's related to cancer. That's the outcome. Exposure and outcome are not dependent on how I collect the information. It is dependent on the biology of the disease that I am studying. So for the biology of that disease, exposure is sunscreen use and then followed by waiting to see if there is cancer developing in that person. And here I am trying to tell you that I've got a group of patients who I call cases, and I've got a group of comparatively no disease persons. How many of them? Equal number of them. They're siblings, so I've got maybe 1:1 or at the most slightly more than 1:1 ratio if they have more than one sibling. I am now fixing the number of people who I am studying and fixing the number of people in the other group who I am also deciding how many I am going to get. This is a case control study, and I am looking to see what happened to them in the past. Read through the second one. Experimental or observational? Experimental. Why is it experimental? Because the person who is doing the study is going to decide they will fall into which group. The women will go into which group. The women are not opting. Here the person who is doing the study is deciding which group they will fall into. What's the outcome? Mortality from breast cancer is the outcome. Exposure is undergoing screening or not. Okay? So I now decide as a researcher which arm they are both going into. That is, the women will go into. Can the same study be done as a cohort study? With the same objective? With the same objective, I can do the same study without doing experimental study, but I can do a observational study design. If I am not intervening, but I am going to a hospital and taking the group of women who have self-selected to be in one of those two arms. I can still find out the ones who are going for mammography versus those who are not going for mammography, wait for 10 years, collect the outcomes, that means find out the mortality in the two arms. Only difference now is, as a researcher, I am observing the outcomes to see what's happening in the two groups. That's a cohort study design, where I don't tell you go to this group and you go to this group. I have the two groups available with me. I am observing to see what's the outcome. So you can see with the same objective, I can do the different studies on whether I want to intervene or whether I want to observe, and I'm not the person who is conducting the experiment. Read through the third one. Observational or interventional? Observational. Why observational? Because I am not telling them whether they should breastfeed or not breastfeed. Practice and obesity or practice and obesity or childhood nutritional status at the age of 5. That must have been the objective, and they have done a cohort study. Can I do a different study design for the same objective? Can I do a case control study design to understand and come to a conclusion about the same, with the same objective? I can. For this study, where will I go for recruiting for a cohort study? In my hospital, I'll go to the neonatology department. I'll go to the neonatology department and say, can we do a study together? We will recruit them when they are newborn babies. And then we will ask history to the mothers, what they are feeding in the first 6 months of life. And after that, who's my co-investigator? Other than the neonatologist, I will now talk to the pediatrician. Because after the first few months, neonatologist is not seeing this baby. They are coming back for immunization, illnesses, everything. They are going to pediatrics department. And I have to catch these children how old? When they are 5 years old. So you have to wait for 5 years to figure out whether they are of normal nutritional status or whether some of them have got overweight or obese. In a case control study, I am not going to talk to my neonatologist. I can fight with my neonatologist. What am I doing? I am going straight to my pediatrician in the hospital. I am saying, oh, you are seeing 5-year-old children. Can we do a study to find out childhood obesity at the age of 5? How is it connected to what they were taking when they were small babies in the first 6 months of life? I don't have to wait for 5 years. What do? I collect 5-year-old children, take their anthropometric measurements, classify them into obese and non-obese. I just have to calculate the sample size differently for a case control study. I now talk to 100 mothers of obese babies, 100 mothers of non-obese 5-year-old babies, and ask them historically, tell me what you were feeding in the first 6 months of life. Who are my cases? Babies who are obese, who are 5-year-old. Who are controls? What does the word control mean here? In epidemiologic study design, the word control does not mean under control, blood pressure control, sugar control, not like that. Control here means comparison group. Who are my controls? The non-obese 5-year-old children are the control group. I am trying to learn from these two groups what did they do differently in the past that they have now become obese. That's a case control study. Can I do a cross-sectional study with the same objective? I can do a cross-sectional study with the same objective. I now do it differently. I don't calculate the sample size for cases. I don't calculate sample size for controls. I go to the pediatrics OPD and calculate the sample size for how many children should I study to find out association between current nutritional status and past breast milk feeding or other feeding. So the sample size is different. I go to the OPD. I don't look for children with obesity and without obesity. If my sample size is 250 5-year-old children, I will collect 250 children. I now measure nutritional status or anthropometry in those 250 children and ask the mothers of the 250 children, what were you feeding these children earlier? The only difference now as compared to a case control study is, in the case control study, if my sample size was 125 cases and 125 controls, I have a large number of cases and controls. While doing a cross-sectional study, I will only know how many of them are obese after I start the study. Maybe 25 out of the 250 are obese. 10% of the children are obese. The remaining 225 are non-obese. So I don't have a strong enough comparison group between the ones who are obese and the non-obese. As you can see, I've got only 25 obese children as compared to my case control study where I had 125 obese children. So with the same objective, I can do different study designs. With the same objective, I can employ different study designs. Preferably, I must use one which is higher up the hierarchy of study designs wherever possible. Of course, if I have 5 years' time, I can do a cohort study. If it's not feasible, even though a cohort study is superior, I will then go one step lower down and say, let me do a case control study. But at least I can do a case control study instead of doing a cross-sectional study. that will be closer to the truth than a cross-sectional study. Okay? What do I mean by closer to the truth? Do you all watch Indian movies? Yes? There's a newly married couple. The husband and wife are in the first floor. Their parents are in the ground floor. In the night, the husband and wife who are living in the first floor, the ones who are living downstairs heard loud noises coming from the first floor. They were having some argument. And then after the loud noises, they heard some vessel's noise also from the first floor. And after that, everybody went off to sleep, switched off all the lights and went off to sleep. And in the night, one burglar got into their house. He went into the kitchen, and at that time in the night, the girl suddenly felt very thirsty. So from the first floor, she came down to the ground floor and switched on the light. Who did she see there? The burglar. The burglar got very scared. He didn't expect this. So to prevent her from screaming and making a noise in the house and waking up everybody, he took a knife which was there in the kitchen and stabbed her in the abdomen. Now because of the pain, she screamed. What happened? After the scream, who comes first? Husband, the farthest, but he came quickly down to the kitchen where the light was on, and he saw her stabbed. And after that, who came? The husband took the wife in his arms, and the knife was in her abdomen. Who came after that? The parents came, and what did they immediately think? They heard noises the previous night. Okay? And who came after this? The door opened. Tell me, you're watching Indian movies? The police came. The police came, looked at the scene of crime. What did they do? They arrested the husband and took him away. You all agree? Is he the culprit? The police did a cross-sectional study. You all did a cohort study. Cohort study is good for identifying causation. Cross-sectional studies are not good for causation, they're only good for association. So usually in the hierarchy of study designs, the ones lower down will give you some idea of association. But you can't be too confident that is the cause. To find causation and say this is the cause, I must do a cohort study, or even better, a trial. How do we know that? Do you know that tobacco is harmful to health? In the 1940s, they didn't know that. So as part of army rations during World War II, all the soldiers will get a package from the government, from the British government. The soldiers' uniform, helmet, guns, along with that some alcohol and packets of cigarettes for the soldiers. At the same time, in England, the doctors who were treating patients saw guys coming into the hospital with breathlessness, chest pain, cough, vomiting blood, or I should say coughing up blood, and they were increasingly many such male patients on whom the doctors diagnosed lung cancer in their hospitals. So they started speaking to those patients and asked them, what did you all do? They first went to their government and said, smoking is causing lung cancer. The government said, get lost. There's no connection between the two. They must have been doing something else. That's why they got lung cancer. So the British doctors then, in the late 1940s, got a whole number of patients with lung cancer and asked in the neighboring beds or in some other part of the hospital patients lung cancer and asked them detailed history to both the groups. What did they find? Several of the lung cancer patients, most of them had been smokers for a large number of years. The ones who they called controls, that means the patients with it, without lung cancer, but who were there for other treatments in the hospital, were they all smokers or non-smokers? Were they all non-smokers? No. A small number of them were smokers, majority were non-smokers. They again went back to the government and said, smoking is very bad, it's connected with lung cancer. What did the government tell them? No, we don't think it's the smoking. They must be living near some high tension wire which is causing some problems in the air, or they must be exposed to something else in the workplace from which they have inhaled that harmful thing. That must be causing the lung cancer. So they went back to the British Medical Association, of which they were a part of. They wrote to the association members and recruited more than 10,000 doctors who are members of the British Medical Association and said, please give us your history now. And they collected the history of whether they were smokers, non-smokers. If they were smokers, when did they start smoking, how many cigarettes per day, and they collected the information for more than 10-12 years. And at the end of that 10-12 year period, they could figure out how many of them during that period had got lung cancer. What were they now doing? What study design? They assembled a cohort of doctors, obtained history, and made sure they didn't have lung cancer in the beginning. So this is people without lung cancer, but with only the exposure. At the end of 12 years, they were able to clearly figure out who were all the smokers, and in that group, what numbers of them developed lung cancer, and in the non-smoking doctors, a small number of them developed lung cancer. And then they were able to convince the government. And that's when only from 1964, cigarette packets started having a message, cigarette smoking is injurious to health. Okay? So it takes years and ages to actually go through different study designs to prove things which are now known to us. So keep in mind, identify the problem, make a plan. The plan will include details about the methods. You have to write the study design, write something about the study setting, calculate the sample size. That means how many should I study to come to the correct conclusion. That's what it means, sample size. How will I go about maintaining high quality data collection? What's the study instrument that I must use? All those will be in the planning stage, which we call the methods section. Figure out how to collect the data. Are you going to collect continuous data or categorical data? Because it matters how we are going to analyze it, how we are going to present it. So it's good to identify the type of data, and then we make plans for analysis and discussion and conclusion. So as I told you, it's not too difficult. We need some curiosity. That's the starting point, which formally we write as introduction, research gap, research question, and the research objective. You identify the most appropriate study design, appropriate in terms of scientifically valid number as well as your own feasibility. What is possible within your work setting? Calculate how many should I study to come to a correct conclusion, which means that you will use a formula, not I will study 50 or I will study 100, not rounded off numbers like that. Use a formal sample size calculation In fact, sometimes even increase the sample size a little bit to account for those who don't come back, to account for loss, to follow up, to account for all of that, increase the sample size. And the sample size that is calculated, remember, is not how many I should start with. The meaning of the word sample size is, if after losing all of them, how many will I end up with? So that's the aim of calculating the sample size. So if it's sample size is 124, I shouldn't start with 124. I won't end up with 124. So I must say, how many should I start with so that by the study is over, I end up with 124 to make the correct conclusion. Write down all the materials and methods, and then the results and plan for dissemination. Any doubts?
So when the sample size for RCT is quite high, which is not, covered within the budget which we have been given, so we tend to go for a pilot study so that we can keep the number of participants in a small scale. So is that justified to do for all the cases, depending on the budget constraints?
Usually for sample size calculation, I say you must marry a biostatistician. Okay? Now, don't take me too seriously and cause trouble in your household. What do I mean by that is, I'll let Dr. Pandey speak after me. He is the king of biostatistics. But the way to approach sample size calculation is, when you're talking to the statistician, you do a review of literature. As Dr. Pandey was saying, if I want to study anemia, I have to calculate prevalence of anemia. If I go to a statistician, I want a sample size. The statistician, what do you think they'll ask me? First question, what do you think is the prevalence of anemia? So it doesn't make sense a little bit. When I was young, I thought, I've come for prevalence of anemia, and he's asking me, what do you think is the prevalence of anemia? Okay. So similarly, for a trial, if I'm going to study 2 drugs, I must do a review of literature to find out already what is the expected difference in the treatment effects of those 2 drugs. If I think one drug is 90%, the new drug, and the old drug is 80, I must do a review of literature first to find out that the old drug is already 80% effective. And why am I waiting for a new drug? I'm not happy with 80%. 20% are not getting better, so I'm hoping that this new drug will be better. I must get a expected difference in my mind, and I must then also do the second part, which is not just rely on one study. I must choose 2-3 different studies with different differences in effect sizes, so that I can go and talk to the biostatistician. In one study, the difference between the 2 drugs was 10%. In another, it is 8%. And in another, it is 20%. What's the advantage when I go to 3? With 3, of course, first is headache for me, because I have to do the review of literature. But when I talk to the biostatistician now, I am in a position to negotiate a little bit. With 10, you will get one sample size. If they put the value 80 and 90, if they put the difference of 8%, will the sample size increase or decrease? With a smaller difference, the sample size will increase. With the difference of 20%, the sample size will reduce a little bit. I now have 3 different sample sizes. If I really want to be very accurate and scientific, I can go back to those 3 different studies and find out in which of them the sample size was very big, because they all would have had their own different sample size. I can now give more weightage to the study which had larger sample size, and then tell the statistician, this study they studied lots, so maybe this is the actual expected difference. So I can get a range of sample sizes and then go ahead and say, if one, according to one formula, same formula, but three different earlier study designs. I get sample size of 112, 144 and 200. I can now say, based on feasibility, I will take 112. But I can't say I calculated the three different sample sizes. I will get only 50 patients. I will now call the study pilot study and go ahead and do a trial with only 50 patients. Can you see the difference? I can't arbitrarily fix the number based on what I have available with me and rechristen it and come up with a new word called pilot study. Can you see in my hierarchy there is no word called pilot study? I can't introduce a new word called a pilot study. That's what Dr. Pandey was saying yesterday. Pilot study is different. Pilot study is, if my sample size is 144, I now want to see whether the patient is understanding everything, my nurse is understanding everything, they know how to fill the form, they know how to enter the data. I can only do a feasibility study. I don't need a sample size for that. I will say, let me enter everything, collect the data on 5 patients. That is a pilot study or a feasibility study for me to test my own system, whether everybody in my team is understood everything, they are all working. That's a pilot study.
About this sample size, you're saying about the clinical trial. And yesterday also I said, sample size is like shopping, right? So you just cannot take one number and go and calculate, right? So it is like shopping. For example, if you're doing a clinical trial, you have 2 groups, A and B, and you have defined some outcome. Sample size would depend on several things, but most importantly, what is the outcome rate in the 2 arms and what is the difference that you're trying to detect. The smaller the difference, the rarer the outcome, the smaller the difference, it would lead to larger sample size. Suppose you have planned a trial and you have said, okay, this is my outcome, right? And you calculate your sample size. And number is so huge that is not feasible for you. Then you need to shop around. Why don't you change your outcome? Closer.
Would I use a cohort study or would I use a randomized control trial for rare disease? Because of the rarity of the case, sir.
In his question, he's already answered some of the things which I wanted to tell, which otherwise I would have asked all of you as a question. But you see, he already asked, can I do a cohort study or can I do a trial? So there are 2 treatments for this rare condition. And whenever we are talking of 2 treatments, we are hoping that one of them is the newer one and therefore it must be better or will it offer more help to the patients. One, of course, is said it's possible to do as a cohort study. So you calculate the sample size, but I cannot, as an investigator, decide who is going to get which of the. So you go to a place or a hospital where they are treating this rare disease in large numbers. So it's a regional hospital where patients are coming from all over, and there is, even though it's a rare condition, I have access to a large number of patients, and that's one condition. The second condition is due to some indication in the patient profile, the doctors in that hospital are already deciding. Some of them will get the old surgery and some are getting the new surgery. So one by one. So that is a cohort study where he is the investigator. He is not deciding which of the 2 patients, which of the 2 treatments or the surgeries the patients will get. As compared to the other way around is he is now saying, I have earlier read reports of study. In the cohort study, that means either in my hospital or in somewhere else, they are doing these 2 surgeries. From the cohort study, it looks like this is slightly better than this. I now actually want to randomize. That means I want to decide which surgery should the next patient who is coming into my study will get. And that's the reason, precisely the reason why you want to do a trial over and above the findings from a cohort study. In the cohort study, who do you think will get the two different surgeries? What are the reasons why they both get differences? Or who will get into one surgery arm and who will get into the other surgery arm? Any reasons? If it's a newer surgery, possibly the younger people in the department went and got trained in the newer surgical technique. And the older surgeons are doing the older surgery. So if I follow a cohort of patients, some of them getting old surgery and some are getting the new surgery, let's say the old surgery is better than the new surgery. Is the difference between the old and new because the old surgery is better, or are the ones who are doing the surgery much more experienced than the ones who are doing the newer surgery? The statistical analysis will come significant, but do you remember Dr. Pandey pointing this out yesterday? The statistical significance will come, but you can't figure out is all the patients undergoing the old surgery in the cohort study, is their outcome better because their surgeons were better, or is the surgery better? Which in a trial you can knock off that bias. You can knock off that bias by deciding patient number one, either they'll go into the new surgery arm or the old surgery arm. Two will again be decided. That is called randomization, where you are making all other factors, known and unknown factors, equal in both the sides. And any difference that you observe of the difference in surgery and not because of other factors like surgeon capability, experience of the surgeon, type of patient, our own inherent biases. I may suddenly in the hospital where they are working, where you did a cohort study, they may be having other factors. They'll say, oh, this fellow, okay, send him for the newer one. This one, just let's do off the old surgery. He's already very old. I don't think the prognosis is good. Why make them spend money? Are also playing a role in the observed difference between the two arms. There are a lot of biases. We use the word bias, whether it's a cohort study or a trial. The hospital is so good that they knew you were going to do a study and they filled all their medical records. So if you just take the patient's charts, you will get everything. If you work in such a hospital, then you can do a retrospective cohort study. Those are not good. But if the hospital is already filling all the forms, if I ask for all the case report forms, all the inpatient documentation, investigations, what happened to them when they came to the emergency, if everything is filled, pakka, 100%, then I can do a retrospective cohort study. I can do what is called. So I will suddenly see that halfway through, information is missing for several of my patients. Not because the medical records department or the doctors are not doing their job, but patients have opted to go to a nearby hospital. So I'll have a lot of missing information if I want to find out 28-day outcome, 3-month outcome, 6-month outcome. It's not there. They've not come to the hospital. So then I'll say, oh, it's easier just to quietly do a study into the future. I won't go to the medical records department and try to collect all this. I'll do it as a trial superior to a cohort study. So that's why I said wherever possible, for the same objective, if you can do a study design which is above the one below that, I would then do a trial. If you can't do a trial, you then at least do a cohort study. You can learn lots from a cohort study and then plan for a trial after that. There are also implications for the team which is doing the study. If I'm doing observational study, I can quickly get ethics approval and start off the observational study. For an interventional study design, my team and me should all be getting a GCP certification. That means somebody has to say, these fellows are going to do the study in an ethical manner.
Thank you so much, sir, for your elaborate and engaging session on the research design. Thank you so much. Now, we would like to felicitate sir with a small, small gift as a token of our love and gratitude. So I would request the organizing team to kindly arrange. And I would like to request our director, sir, to hand over the gift to Prem Moni, sir. Thank you so much, sir. Now we would go to the next session, which is on manuscript writing. For this session, we can't ask for a better resource person than Dr. Samran Panda, sir. Sir need no introduction to ICMR or have published in the IGMR. Sir is a very eminent person in the ICMR and outside. Currently, he is Dr. A.S.
We have a problem in our mind, a question that had been bothering us. So we wanted to do a research, and Dr. Subarna Roy yesterday talked about our desire to publish. Why do we want to publish? But publication happens after we write the manuscript, right? So that piece is what we are going to deal with. So once we know that, we can happily go for lunch because we have sent out our manuscript, which will be published. So first of all, I want to sort of know from you your experiences in terms of the struggles or the difficulties that you have faced, irrespective of you being able to publish, or maybe a few papers, 4 or 5 papers, or if you are a prolific publisher or writer who have sent out manuscripts and you have become successful. From that perspective, I would like to know, I'm writing the word struggle here. So what has been the struggle or in terms of writing a manuscript and getting it published? Anyone can, microphones are there in the audience. Anyone can just say, even if you have not published anything, you have tried, but something just didn't happen. Just raise your hand. It's not a question answer session. Yes, go ahead. Sorry? Finding? Oh, finding out the most appropriate journal. So you have everything sort of set up, done, but the journal is not there. Okay, the platform. So the kind of study that I have done or I want to publish, I want a right kind of platform or a journal. Okay, but I'm saying that actually is almost there. If there is a journal which I can find out, I can send it across. My question is, what is the struggle that I'm facing? Any one of you. I do it all the time. Every paper I try to write, again, the same feeling of the butterflies in my tummy and the difficulties. So I'm a bad example, okay? I have another disclosure. Somebody introduced me as editor-in-chief, and you thinking, here is the guy who is a nice one. He's going to tell us how to write a manuscript and publish. I'm actually not a nice person to know, because I'm messenger of the bad news. Indian Journal of Medical Research has 20% acceptance rate. So for every 100 people who receive a letter with my name, only 20 of them get to hear, okay, major revision, minor revision, or the rest, 80, say, sorry, we cannot publish. So I'm messenger of bad news. So Dr. Panda is very well known, but he did not complete. For what? He did not say. And of course, I mean, you don't say that kind of thing straight on the face. You discuss it over lunch or over evening coffee. All right. So we will try see if we can identify the struggles, because what Dr. Prem said, if you find the problem, then you can plan. Here I am talking about a different kind of problem. So what is the struggle or the obstacle?
Begin and what to begin.
Begin.
That is
Ah. Do I begin at the beginning, and where is that beginning? Okay, how do I begin? Excellent. Now we are really getting to the core of it. Go ahead, Suvarna, go ahead.
My problem is that where to end? What is the message that I am going to give?
Okay,
the one thing.
Great. Where to end? All right?
So
Any other?
while preparing the manuscript, like we have to fit in the number of words, and also they give like this many, you only have to have maximum 2 tables, 2 figures. So if I have a lot of data, how to put everything in those tables, so those things.
So
while
in other words, the journal editors, like people like me, who is of course not a good person, I myself am declaring and disclosing, they are not kind. I have done a very good study. I have written it up, which can only be written up in 7,588 words, with references and everything, and they are saying, well, with the references it can go up to 4,500, and the main manuscript cannot go beyond 2,900 to 3,000, something like that. So that's a struggle, okay? Yes,
sir.
Go ahead.
Sir, during our time, when we started our journey, we didn't understand what was actual protocol was. We didn't have this type of workshop or something
Research
like
methodology
that.
workshop?
Yes. Actually, we didn't know what is actual protocol was.
Okay.
After that, what will be the type of study? That was also not told.
So again, Dr. Prem did talk about the problem and the plan, and then you have the data collection, analyze, conclude, but the conclusion actually doesn't end because that might not be able to answer all the questions that you had in your mind, or maybe that raises further questions. Okay, excellent.
Sir.
Yes, go ahead, please, sir.
Yeah, in selection of the title of the manuscript and review of literature.
Title and review of literature.
And,
okay.
And I am the one, last month I listened the bad news from your journal.
Title. Okay. All right, naming the baby. The baby is born, but I cannot find a name. The whole team cannot find a name. We have given birth to a baby. Oh, well, that's a tough thing. Yes.
Sir, what happens sometimes we find a very beautiful like portion, like I want to refer something. Then what happens, sir, for that same topic or same point, I find so many references that which one to keep and which one to avoid. Is it the earlier one, the first one, according to the year, or according to the citation, or according, like, wo nahi samajh mein aata hai, sir.
So I'm in the mango orchard, there are so many mangoes, which one do I pick? You know, I want them all, but I cannot have them all, so which one to, you know, pick and test? Okay, so which one to use from the references? Excellent, which one to use? Particularly in terms of references, because all of them are nice and beautiful and excellent, so which one to use? Go ahead, please.
So, bibliography.
Okay. Okay, meaningful discussion. Okay, I want to discuss, and he said, how to write a meaningful discussion. Okay. Anything else?
So, writing the bibliography part and whether to include references or cross-references.
Okay. Bibliography, and that's related to, actually somebody talked about references. Okay. Go ahead.
The cost of publication charges.
Yeah, probably you are from which institute? Negrims. Dr. Nalin Mehta is gone, but I will pass on the message. Cost of publication. Who is going to give me the APC, they call it, article processing charge, or manuscript handling charge. That's the term generally used. Okay. Anything else? Yes, go ahead.
Avoiding the overlap in discussion section from the result section.
You see, I am gradually falling in more love with Dr. Prem, because you see, by taking this shortcut, I am getting half of my sessions done. Yes, she said that repeating the same thing in the discussion from the results. And let me tell you, on a daily basis, I'm not exaggerating. I was typing on the laptop something yesterday, and on a daily basis, let me tell you, 5 to 10% of the rejections about which I make a decision and convey is related exactly to this. Discussion is not, don't tell me again what you already told. I have read it. And you are again talking about the same percentage, and again the males are suffering more than women. I mean, okay, you said that in the results. So what do I do? So somebody actually said that, how to write the discussion. You are going deeper into the problem that, do I repeat it or what do I do in the discussion? I've said it all. Dr. R.M. Pandey said something, and that I will also refer to, that the difference between the manuscript and the proposal, research proposal, is that it is up to the results, because the results can only be done once you have done the research, but before that, the rest is the same. Is it so or not? We'll look into this. More or less it is true, but there are subtle differences. Any other? Yes, ma'am.
Sometimes it happens that the person that you are working with is such a perfectionist that you keep correcting without any end, and then you ultimately lose your interest, and then that just lands there without getting published.
Right. So there is a term called magnum opus, you know. So that perfectionist, or that person, the whoever your boss is or whoever you work with, he or she is so much obsessive about making everything perfect, the manuscript never comes to completion, like it never gets written. So naturally you do not send, and then it is kind of a project which is never ending. Okay, that's a very important point. Yes?
Sir, after we submit our manuscript, we get multiple revisions. After getting multiple revisions, we get a hope that it will get published. But after around a year time, we get the result as it won't be published. So that time, that is the problem, sir.
Yeah, so long time quoting, and then nothing is really ending up in the final culmination of marriage or founding a family. So revisions, so how do I do the revisions so that I do not get rejected after that long process of revisions? Go ahead. Then you.
How to avoid paraphrasing and plagiarism. How to avoid paraphrasing and plagiarism.
Okay. So paraphrasing and plagiarism, and how to not fall into the trap of it. Go ahead, Ipshita.
Sir, abstract, I find one of the most difficult one.
Okay, abstract.
With the word limit. Say.
Anything else? Yes, go ahead.
Sir, my struggle is before the publication. Sometimes I have confusion whether I should exhaust all my data to attempt a very good quality journal, or should I publish, 2 papers, from my available data.
Okay, so today, while Dr. Nalin Mahita was talking about, and again Dr. Suvarna Roy yesterday, they used a term, salami publication or slicing. So you have a data set as a part of one project written and grant received. Now you are in 2 minds. Do I make a slice of it and present as 2 papers, or
Yes.
I do it as one? Actually, that's unethical, because if you have a research question in place which required designing a project with stated objectives, which you have completed, so the answers, your data is providing you with the answers, right, to the question that you had. So, and all those answers are directed towards answering that question. So if you take away part of the answers, you are not answering the question. So that's called slicing or salami publication, which is unethical, because you are given the money and you involved human participants to find that answers. So not taking it to the logical completion, it will be an unethical practice. So better to avoid, but should we do that or should we not? We should not. Okay. Anything else?
So I had a question.
Okay.
So how to stick to the references that actually stick to the topic itself, the topic that we are presenting, or we are for publication we are giving. Most of the references that we find is out of the context. So how to sticking to the context is the main.
Okay, so it is again about references. Which one do I use, and do I talk about the far places? So now, this kind of feedback that I took, is it a quantitative research that I did? If you do not mind me calling this a research, because I was trying to find out something, right? So over the last 5 minutes or 10 minutes, I was getting inputs. You want something? Authorship.
Multiple people, like, yeah.
Authorship is a different criteria, although my focus is about manuscript, but that's a very important question that you have raised. So how to decide upon the authorship while we are writing a manuscript, and there are multiple authors, and how do they get credits, etc. All right, thank you. So I was trying to get the inputs. If it is a research, if you don't mind me calling this research, was it a quantitative research or a qualitative research? Can you be a little louder, please? Huh? So, mixed method. Both quantitative and qualitative. You are saying qualitative. 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 So, okay, majority of the people think it's qualitative, and in the qualitative research you see now from any other, you are actually repeating. If you are writing on a qualitative research that you have completed, you need to indicate, although there was a mention yesterday about the sample size, but you need to be able to reach out to sufficient number which gave you a saturation point, or which helped you to reach a saturation point. So that's very, very important. So one important component of writing a qualitative study and trying to publish it. All right. So we will go through these are the struggles or the difficulties. Can I have my presentation, please? And we'll try see whether we have been able to address some or most of the issues that you have raised. In terms of authorship criteria, as you asked, there's a clear-cut description in 2 places, and there are books also. I can share that with you. Excellent. How do I change it? This one? Okay. Okay. Where does it start? If you listen to Dr. Prem's presentation, there is a lady. Can you see a woman there? A beautiful woman from Assam? Can you see? Yes. But do you see any English letter there also? C. Why it is C? Because that lady is curious. She lives in. And that's what Dr. Prem said, that if you really want to be a good researcher, he did not use it as good, but he said research with all its seriousness, then you need to have curiosity about the world you are in, right? When you are curious, you don't maintain one position. You lean a little farther forward with a magnifying glass. You want to understand. And understanding is the term. Again, Dr. Prem said that might not be amenable to measurement, quantitative terms. So here we are clearly talking about qualitative research, where you can try understand an issue or a phenomena, and then quantitative research, where you try quantify and try understand the relationship between the two, association or be it effect of an intervention. And when you go closer further, because you are curious about the world and the phenomena around you, you get to see this. In the Northeast, you wanted to do research, you are curious about things, And you got to know about HIV having a very rapid and explosive presence because somebody did surveillance, and you come across these kind of objects which are there in the community. What are you looking at actually? Any idea? Sorry? What is that? Go ahead. Whatever is coming to your mind. This is not a question answer session.
Sir, is it Morse code? Morse code sending machine?
No. Have you seen this kind of? No. Magnifying glass is there in the hand of that lady who is leaning forward because she is curious about the world, and she got to know about this HIV prevalence detected, in the Northeast, and in one of the northeastern states. Let's assume that it's Mizoram or it's Nagaland. In the community, these kind of things, or Manipur even, these kind of things are, young people are carrying them. Drug. What is the drug part in it? I can see a little cap, and I see another, container, which can be probably capped with the help of the cap, and then I am seeing something like a needle, but the needle is not fitted with a syringe as we understand, but in our childhood days, I saw this kind of thing like ink dropper. So an ink dropper fitted with a needle, and now I need to understand it much more, because I am familiar with hypodermic syringe and needle, but it doesn't look like that. But why young people who are in the drug use are carrying this ink dropper fitted with needle, and will it pose greater risk compared to the hypodermic syringe and needle if those are used for drug injecting? And what is this Vicks? If you are familiar with the term Vicks, this is Vicks bomb, the container. What do they do with this? In order to understand all of this, what kind of study do you need to do, quantitative or qualitative? What kind of study do you need to do, quantitative or qualitative? Clearly qualitative, without understanding. So there is a need. So don't fix your mind first. So all the research is not hypothesis testing. Hypothesis generating studies, and qualitative studies are great in that. Like the question that I'm asking, will they have greater chance of a bit of a residual? I push it in the vein, I then squeeze it, and then the moment I squeeze the drug in and I release it, a little bit of blood will come back too, and I will pass it on to my friend. If Dr. Prem doesn't mind, I will pass it on to Dr. Prem, and then he takes it because he considers me as kith and kin. So he injects, but he is injecting again after taking that drug which is dissolved in the container, the Vicks container, but that is getting mixed with a little bit leftover blood which is there in the ink dropper, and he loves me, he likes me, he trusts me to the extreme. So he doesn't bother, and neither my face can tell whether I'm having HIV or hepatitis B or hepatitis C. So will that have an increased chance? Because we are not, decontaminating our syringe and needle at all. Will it have a greater chance of spread of virus, for that matter, compared to the usage of syringe and needle? If you have curiosity, then you want to explore further, either through quantitative research or qualitative research, and there has to be observation. You could do different ways. You have to think. You need to have knowledge, because yesterday I was actually noting down when you said why all of you are here in this particular research workshop, research methodology workshop. You said many things, but then curiosity is something which I did not hear any issue. So first thing, probably because why I'm talking about this in the manuscript, we'll see that. If I am not curious, if I have been thrusted upon, sometimes the manuscript writing also happens like I'm a senior person, I really want to help my junior, and I say, look, here's the pile of data, please analyze it and write a manuscript. What is this? There are some data which can be analyzed and then the manuscript can be written. I do not know in which context that data was collected, what was the method employed for collecting that data, what was the ultimate objective, how was the data collected, how did the measurements happen. So never ever get into this trap of so-called good the help. And people sometimes think, oh, I need to just write a manuscript. Sorry. It's a continuum. That's what I'm trying to highlight. So happen like de novo. Then formulating a question ought to be done. So that's the point I wanted to have. So yesterday you had a session which you could not have, unfortunately, due to the proposal writing. So are they different or are they the same? Of course, R.M. Pandey said that proposal stops before results because results have not been generated. But do you see any other difference between a proposal? Because there has to be a rationale, there has to be a research question, there has to be an introduction also in your proposal, which you have in your manuscript as well. Which is future tense? Proposal is future tense. Proposal cannot be a future tense, then you will never get the money. You have to live in the present. And while you are discussing, we are discussing with manuscript, manuscript is future. You do not know what would be the result and you will up. So proposal, of course, is in the past in terms of thinking that you submit something to ICMR for getting the fund, but then you have to have the proposal in place and then submit it. But why ICMR should give you money? Why ICMR should give you money? What should be there in your proposal that ICMR says, aha, let's give Ipshita, right? Okay, let's give Ipshita some money, maybe 2 crores. Because the first day I was listening to somebody saying 2 crores, 3 crores, I always get scared, I have never seen. If you have such a persuasive argument that they see it would serve purposes in terms of all the 4 sectors, whatever that 4. Dr. Prem talked about 4 Ds, right? Any one of those which ICMR might perceive as important, they will. So that argument. But you need to have that argument again repeated in your manuscript. That's one. And in the manuscript, you have to have basically a story told. So if you are, so that is the basic difference. In the proposal, when you are writing it and applying for the grant, you do not have the story built as yet. So these are the 2 basic differences, okay? Now, what is the shift in role? When you are writing the proposal and when you are writing the manuscript, you are the same Suvarna Roy, Samiran Panda, Prem Moni, but then why am I saying that there has to be a shift in role understanding or conceptually, you need to appreciate that there is a shift in role. What is that shift? At the time of proposal writing, what do you do? When you are talking about a problem or a gap or something that has already happened, but yet you have a question that is bothering you or you want to do a study, so what do you do? How do you develop your question? Just by observing? Gaps in what? Yes, you are right. Gap. Missing. Missing in what others have already done. Right? So, you will be reading others' work. So, you are the consumer. Some people have done some research. They have also published them. You have been reading them and you realize that something is still missing, but you can challenge them or you can expand it further or you can even try refute it because you are observing certain things. So basically you are reading somebody else's research papers. So you are the consumer of what is already available, but while writing a manuscript, you are the producer. So your role is shifting from being consumer to the producer. So if you are writing and publishing something, you want others to read it, so that requires certain considerations. So reading somebody else's paper, making up your research proposal is fine, but then when your research is done and you really want to sort of collate that information which you have obtained, analyze them, be it a qualitative study or be it a quantitative study, you need to be able to tell your story in such a manner that others read it. So it requires some of the things that you have already highlighted. How do I begin? How do I go with the title? How do I go with the introduction or discussion, etc.? So that's why understanding, so this is the craftsmanship. It does not happen automatically. So you have written up a research proposal, and then you submitted it to ICMR. ICMR gave you the money, you have the results, and then you are thinking, oh, manuscript writing is cakewalk. No. It requires a craftsmanship. And that's why you raised all those questions. That's why I was so very grateful to you that you really addressed the struggles or the difficulties in writing a manuscript. So why write? While you are writing the proposal, you see, while you are reading somebody else's work, you want to remember that the conversation between him and the other researcher, and then you are writing to understand that your own understanding is okay and your line of thinking is okay. But while you are writing the manuscript, you are thinking about the story or the journey, like when you identified the problem and then you found out some inkling about the answers, and now you are trying to capture it in the form of a story. That's very important. That's the difference between the proposal and the manuscript writing. Okay. So you might write, you might publish, but people might not read. This holds true for both proposal and the manuscript. So you have to earn a reading. Because if your language, of course that was again highlighted by Dr. Prem, and then the structure of it, but what more? What more do you need? If you have your language okay and the structure okay, because you have talked about the structure, be it proposal or be it manuscript, if you need to present someone with a garland, you need to have a string run through. So it has to be a linked journey or argument chain which helps people to understand the story that you are trying to tell them. So that thread is very important. So it has to have a logic. In science writing, please remember, I have seen that I had to, because ICMR wanted me to examine many policy briefs or brochures, say they do 6 pages of policy brief. I said, forget it. No policymaker will read 6 pages because manuscript writing, it is science writing, yes, policy brief or a brochure, but that is very different from manuscript writing. So never ever apply the skill that you will learn and successfully, and you have already, some of you have already applied the skill in manuscript writing that does not get translated or cannot be taken in toto to policy brief writing or scientific brochures. Okay, are you familiar with the term IMRAD? Yes, so I need not to elaborate upon that, but you have certain questions about the sections. Now, I have deliberately put title and abstract as a different section and others as a different section. Which one should be dealt first with? Sorry? Results. So one need to wishes to dive straight to the results. Methods followed by result, okay. What should be dealt with first? Introduction. Okay. But nobody is saying I'm happy. Someone wrote a love letter to his girlfriend stating, I would have written a shorter letter if I would have had greater time in hand. If I would have had greater time in hand, I would have written a shorter letter. So it is easier to write longish things when you do not have much time. So abstract writing requires huge amount of time. So it takes huge amount of time. It is written at the end of everything, but it is put at the top of everything, you see. So never ever, and at some point in time there was no abstract. Do you have any clue that at some point any branch of science would not have any abstract? Did you know about this? No? So this is given to you, any research paper, 1830 to 1870. If you see the scientific papers, even before that, of course, before 1830 also, there won't be any abstract. So the concept of abstract came much later, and then we will discuss why so. Okay, so you are familiar with this, we need not to. Now, what is the purpose of the introduction? Somebody said. Now, I have put up a picture there. I won't go into great details, but why is that picture there? What is it doing there? We are discussing about introduction. I don't know. I'm seeing a picture. You have to tell me what is happening there. Where are you? Can you see yourself there in that picture? Huh? Ab chota, wo chota banda hai. He is right. Give him a big hand. He said, I am that small guy there, on the shoulder. On the shoulder of whom? Excellent. Give him a big hand. He is saying that that big guy is the big all literature, all the researchers. And taking help of that, I want to see a little further, a little more. So there is a term, I am not going to get into that, that's called Ubuntu. That's a group in Africa. But introduction is about where do I begin. Keeping the readers in mind, you have to read or write the introduction, and keeping the reviewers in mind, because we are living in a world where nobody has any time. Same thing happens with the editors, particularly bad ones like me, and the reviewers. So how do you get hold of them? So if you write a good title and the good abstract, probably you are getting him or her hooked. He will read introduction. If he becomes very dissatisfied with the introduction, he or she won't move further. So that's why there are two purposes. First of all, keeping the readers in mind and keeping the reviewers in mind. So introduction, in the introduction I need to know which disease, if it is about a disease, are you talking about a disease or a risk factor, or is it about a therapy, is it about a technique, or a group of patients, or it's a phenomenon observed in the community. And this will dictate immediately that, okay, then that should be the title. Depending on, is it an observational study which could answer whatever is being told in the introduction, or is it going to be an interventional study? But somebody said that, how do I begin? So start with a few words on the overall topic, but don't wander too far, okay? So don't say HIV. Don't say that HIV was first identified in 1981 amongst a group of healthy homosexuals when Michael Gottlieb in Los Angeles. Please. Please. You said, no, Dr. Panda said not to wander too far. So you go. No, I have to revise myself. So in 1986, a group of sex workers in Chennai, while they were tested in the Stanley Medical College, I said, is it about Stanley Medical College? It is about, I don't know. But after reading the first paragraph, I have no clue. Then I said, okay. Very. So try understand that introduction does 4 things. Just get it by your heart. Number 1, what is being, okay? Don't wander too far. And what did others do? Just 4 things. If you remember, done. What did others do? Previous research, the bada banda, the big guy. And then you have identified some problems, so the gaps in what has already been done, and then what have you done to fix those problems. Just 4 things. And don't go beyond at all. Now you know where to begin. So somebody had a question. Where to begin? That's how to begin, and that too on specific lines. So one area here we can tick. So somebody asked a question, and that's done. If that's about the introduction, we go to, is there some loose connectivity somewhere? So what is the first one in introduction? The topic. The topic, it could be about the patient, about a risk factor, about a therapy, about a group, or a phenomena observed. What could be the phenomena observed in the community? That Vicks container, empty ones, and the ink dropper fitted with a needle. That could be a phenomena. Okay, now method or methodology. So try to learn what to use. There is a reason behind using methodology. But ethics committee clearance you have to have, and then people or participants, who are you dealing with, the time and the place where you have done the work, tools and the tests, how did you measure, what did you want to measure, so that people know exactly which path did you traverse, and the analysis. So what are the, how many components in methods? Let's start with ethics, okay? Time and place, tools used for measuring whatever you wanted to measure, and analysis you have to have, right? And participants, how did you select and how did you have an access? In qualitative research, you will realize that we did talk about, let's say, injection drug users. It's not possible for you to get the access all the time. So you might need a gatekeeper, that's called gatekeeper, okay? So having or befriending a gatekeeper and then having an access, that should be the part of the discussion in the qualitative research paper if you are one. Okay. Now, salient points that you need to cover is, but the most important is how did you adjust for the confounding if you are doing a quantitative study. So, Dr. Pratap Tharian, he did show that there were some guidelines. And for example, observational studies, Dr. Prem has talked about it, Dr. Ranampand has talked about it. So if you are reporting or writing a manuscript about observational studies, there is a guideline which is called Strobe guidelines. And Strobe guidelines is a one-pager checklist. Just use that checklist in order to address everything. Then your chance of getting accepted for your publication will go up. Now, this is about the results. So you have data. There is a difference between data and information. Try understand that. And as you write, sometimes, particularly in basic science research, it even comes from the journal office that you do some more experiments before you, because whatever you are claiming, you cannot really, establish that with the help of the data that you have generated. So you need to undertake some more experiments. And then you need to take care of the issues as the last bullet point. So we have talked about 4 sections. Now you go in the discussion. There are certain or absolute no-nos. Don't do. If you do it, IGMR is not going to take it. And forget about IGMR, no journal will take it. So in the discussion, as you said, that don't start repeating the results again. So you really need to know what did the study show. So what are the highlights? What might that mean? I mean, whatever you are saying, what does it really mean? How else could the results be interpreted? Could there be alternative interpretations? Have other studies had similar results or they did not? They might have similar results. They might have a little different results on certain counts. And is there disagreement in the field? Because if you are working in the field of, let's say, HIV among injection drug users, and people say Northeast India, sorry, Northeast India is not a mashed potato. Manipur is different from Mizoram, Mizoram is different from Nagaland, Nagaland is different from Tripura. So there could be, difference, and difference due to several reasons. And then what are the scientific and the clinical implications of the findings that your research has come up with? That's what you need to write in the discussion. So somebody asked a question, what do I write in the discussion? These are the exactly, 8 points that you have to remember. And then what exactly should happen next in the field of research that you end with. Somebody asked a question, how do I end? Because you cannot say, that's it, that's the final word in this field. No. So there might be necessity of other, research in the same field that you were successful in getting the grant with after writing the proposal, and now you are writing the manuscript. So we have also tried to answer, how do we end our manuscript? Okay, now title and reviewers. This is, yeah, go ahead, please.
Discussion should also show limitations of the study and how you will address those limitations.
Of course you should.
Yeah, that should
clearly.
also.
Give him a big hand, which shows that he is listening. And he is also putting, points in which are important. Right, so strengths and weaknesses are there. You are using the term limitations, that's fine. But we should all remember that no study can be absolutely flawless. You need to know that there are. Now this is something interesting. Acha, if after reading your title, the reviewer, I mean the journal, in-house reviewer says, sound interesting, I should read this, has your chance gone up or gone down? If somebody gets hooked and shows interest in reading your article, your chance of actually getting read is going up. But then there could be other reactions. Oh, not again. HIV, AIDS is not caused by HIV. You know that, right? There is a group in the world, there is a group who believes that AIDS is not caused by the virus HIV. Okay? Or can't be, like you are claiming. So always avoid the claim of primacy or supremacy. And it can't be that kind of feeling, or how dull it is, like the title, or even the abstract. So that's why I say title and abstracts. Or if he says, what is this? You know? One pill a day and once, you are done with HIV. Excellent. And the reviewer would say, one pill a day and once. So don't claim that kind of thing. So now we will go through quick exercise, because it's a skill building. You had your questions, you got some answers to your questions. Now, if you cannot think of a title, you start practicing this. To say this kind of statement, my study shows that. Dr. Prem, I think in his life has spent much of his time in the corridors, so he gave corridor example. I love elevators. So some people say elevator speech. Your director say, what are you doing? I don't see you these days in the institution. No, sir, I am working, sir. What are you working on? But the elevator is already reaching the director's floor. Huh? He will go out. So you have to finish within 5 seconds or 10 seconds. So be it elevator or be it corridor, you have to be able to say in a sentence, what did your study show? Because if your director is angry, you are not going to get the intramural fund. Right? Okay. So if you say that my study showed that women were more likely than men to discuss diet and exercise with doctors, you can just turn it into title. Patient-physician communication about lifestyle. So diet and exercise is all about lifestyle. We are talking about one gender having more communication around a topic, and communication with whom? So it is the patient-physician communication. Is this clear? So should we go for the next? All right. So let's try this. My study, your director asked you, and you told the director, my study, sir, showed that after adjusting for age, women were not more likely than men to discuss diet and exercise with their doctors. We are getting into slowly the analysis part, but I'm not getting into that. I'm just now trying to have a skill building in how to frame a title. Would you give it a try? What can be the title? When you are writing this manuscript, you have already told this to the director in the elevator or in the corridor. You want help? Yeah? Okay. You see, effects of participant gender on participant-physician communication, on lifestyle, you can add on lifestyle, are confounded by age. That's exactly what it is. So you can bring it up in the title. So it is a very good practice to say it in simple terms. My study showed, and then you can turn it or flip it with the technical terms to form a title. You want to try the next one? Okay, there's a willingness. My study showed that treatment with mouthwash reduced the incidence of thrush. This, if you have understood research methodology or research study design, you should be able to get the right kind of words like that. In order to do this kind of study, what kind of study should you do? If you want to show that use of mouthwash reduces the incidence, all of you know, I need not to elaborate, of oral thrush. I'm talking about oral candidiasis. Do you want to think of a title? Give it a try. You want to? Go ahead. Yes, at least there is, willingness. Give it to him.
Sir, effect of mouthwash in reading incidence of thrush.
Okay, very good. How can we see? So if it is mouthwash is being compared against something else and the incidence is coming down, okay, fine. I'm taking your, or do we want to refine it? Study design, what kind of study design would be required? Okay, so somebody is saying that study design, if that can be indicated in the title, that would be better. Does anyone want to refine it further? So you see, if we try, it's possible, it's doable. Now look at this. Randomized blinded trial of mouthwash in the... So you have the design indicated. You can do all, like what did you do? So these are the kind of interesting exercise. But what happens if you do a study where it's not very original? You don't naturally say yet another study of association between hypertension and stroke. Neither do you say measurement of arterial blood pressure. So if you yourself are convinced that it's nothing great or novel, try find out if there is anything novel in it. Like, is it the first time in the recovering alcoholics? Or is there anything rigorous in your study design? Like somebody said mixed method. If there is a mixed method study and you see that nobody has tried this ever, and you see that mixed method is very important to address this particular issue, then you can highlight that in the title. Or is it the first study where you are using an ambulatory blood pressure monitoring device? Or anything surprising about your results? Or maybe you found that hypertension was not associated with stroke in people who are below 50. Now abstract. So when do you write the abstract? In one of the workshops, when I asked, when do you write the abstract, what was the response, you know? Sir, in the morning. Okay, you may even write an abstract, okay? So here the question is, when do you write it, abstract? At the end. You have fairly good idea about introduction, you have fine-tuned it, the methodology you have fine-tuned, results, discussions. Abstract needs to capture it all, and yet it has, somebody said, word count, word limit. Abstract will be 250 words or 300 words. If you are lucky, some of the journals would allow you 400 words. Okay? So that's why abstract in the era of information explosion is going to be very critical for your success in getting your manuscript not only written, but even getting accepted or getting rejected. So abstract is the first foot forward. When you are in love with somebody, right? So what do you do? Now I see a whole lot of excitement. Okay, what do you do when you are in love with somebody? Yes, sir. Everybody is smiling, but nobody is willing to speak up. What do you do? Exactly. You want to, do you see? So we do not want to show our real self. Real self is in the introduction, then in the methodology, then in the results. But abstract, at least I want to impress that person. And I put my best foot forward. Okay? So abstract is, that's why very, very important, and it is very important. It serves very important manuscript getting accepted. It could be structured or unstructured. So you have to have the background, the research questions, design, everything there, but include and what is being indicated. So you see, in the discussion, the last how to end was the future direction. So everything is there in the abstract, but don't take it casually. If your abstract is not right, you are not going to go right. Acha, is there any term called, now I am talking about all the narratives, textual things. I have come to the end part of my presentation. Just a few interesting photographs I will show. But before I show the photographs, is there any term called honest graph and dishonest graph? Because in your manuscript, it won't be only text. You would have sometimes, graphics. Graphics has a different meaning than exactly what you understand from line graph. But is there a term called honest graph and dishonest graph? No? How many of you think that there is no such term called dishonest graph? Show your hands, please. There must be there, okay. Look at this. The same thing, the same thing is being shown, but the impressions that is being conveyed is very different. Okay? And that has happened because the y-axis scale has been changed. So that's dishonesty. If you want to convey an impression, and in order to convey that impression, you are changing the scale as it suits your hidden agenda or intention. You shouldn't do that, you know. But graph can also help you not only show the data, but also insert other kind of information. I will talk about this HIV self-test in Mizoram. So I know that people are here from Mizoram. And every time people think, we can come and help Northeast. Sorry, Northeast can also help you. There are great examples of great studies done in Manipur and in Mizoram, Nagaland and Tripura, which the rest of India can actually learn from. So you see, there could be these kind of graphs. You have graphical lines. This is HIV self-test uptake going up, and then HIV detection going up, and people are getting connected to the antiretroviral therapy. But these blue lines are indicating sustained efforts. Sustained are indicating sustained efforts. Sustained efforts in terms of community voices. So community voices is completely qualitative study. And then you have a rapid review for response generation. That is synthesis, rapid synthesis. And then you have the present study, which is a mixed method study, as Bishwajit was talking about. So present study is a mixed method study, and all of these together within a period of 2 to 3 years, it produced such a momentum in Mizoram that HIV self-test uptake went up, and amongst youth, within 6 months' time, more than 2,100, if my memory serves me right, 2,100 HIV self-test, of which some people turned out to be positive, 100 HIV self-test, of which some people turned out to be positive. They got connected to the confirmatory, and then they got connected to the antiretroviral therapy. You see? So if you do studies, so your question should guide what should be the study design. It's not, I love case control study. Okay, you love. But the question that you are asking might not be amenable to that design. That's my point. So there is no love with any design. So your question is the king. Nobody else. Neither your comfort with any particular thing. Okay, so in the backstage, now you have written the manuscript, you have sent it to the journal. That's the journal backstage, okay? You do not know what is happening behind. Somebody peeps out and says, hey, rejected. Or somebody says, hey, accepted. But is he doing at his whims or fancies? No. In your study execution, could be fatal flaws with the design. Your design might not be suitable, because Dr. Prem actually helped you out in terms of seeing or being able to see that a particular question can be addressed by different designs. But what I'm adding up here is that not can be addressed by any kind of design. So question should determine, there could be multiple options depending on the time you have, depending on the resource you have, but not that any kind of design can be just undertaken to address a particular question. There could be no merit. And in the IGMR, I can tell you, if there is no merit for external review, external peer review, you get a letter within 7 to 10 days. So it is a very quick turnaround time. Within 7 to 10 days, max 15 days, that no, we cannot take it to the external review. And identifying the potential reviewers, that's a big problem for some of the articles. And then some of the authors become really unhappy with the reviewer's feedback. Somebody told that the reviewer did not understand. So I could read your mind and have that inserted in the slide from beforehand. Good. And then you have addressing the potential reviewers are also human beings. They have their biases. So they have biases against race, they have biases against gender, sexuality, and other aspects. Now, rejection, Subarna Roy is the best person. If you are rejected in terms of love or marriage, just, consult him. I'm no good there because nobody has chosen me ever in life. So I'm not the person. I can tell you how to prevent rejection in terms of articles from the journals. So if you are that is freely downloadable, okay? The ethics guideline of ICMR involving human participants. There is another guideline, this is research integrity unit. Every institute you should have. So that is also freely downloadable, so read them. And then there are guidelines for observational studies, clinical trials, diagnostic accuracy studies, etc. So if you follow them, then your rejection, the chance of rejection will be much less. And when you are coming out of your comfort zone, then your chance of rejection also goes down. You know, because you need to go out in the community if the need be, if there is a phenomena that you want to study, and if you wish to study that, come out of your comfort zone, it will be easier. And this book, okay, I should have a disclosure. I do not get any percentage from the proceedings of sales of this book, but this is a book which is about ethical practice. And actually unethical practices that happened with HeLa. There's HeLa cell line and there's Henrietta Lack. And then as an author, you know, you should not have any of these practices which are unethical practices. And if you do that, and the previous slides, as I indicated, then your chance of getting rejected will be less and less. Because don't think that manuscript writing something there, you can just say ethical clearance was obtained. Because Dr. R.M. Pandey very clearly said something yesterday. Do remember that he said, okay, if I read this kind of statement in the analysis, I immediately know there is something, you know, gadbad. He used the term gadbad. There is something gadbad. So that is the way we quickly get to the heart of a thing. Now, I'll just show quickly some of the photos. This is about Manipur. You see, can you diagnose this? Herpes zoster. So in the Oxford textbook of medicine, it was written that if you hit some part of the body, for the rugby players or for anybody, then the reactivation of the herpes zoster virus happens on that part of the body, right? So in Manipur, we were starting the injecting drug users, and where do they inject? Mostly on the extremities, on the hands or on the legs, because the veins are prominent there. Reactivation, because many of them were HIV positive, so there was immunosuppression. How many of you had chickenpox in your, when you were a child? I had. You can ask your mom, then the number, but huge number of people. So the virus is hidden here at the posterior nerve root ganglia, okay? So if you go through immune suppression, then it can come up in the form of herpes zoster, okay? So this is herpes zoster. And Oxford textbook of medicine said that when you have a physical trauma repeatedly, but so then I would expect the herpes zoster coming up at the distribution of the dermatomes on the extremities, upper extremity or the lower extremity. But that's what we did not find. Majority of the herpes zoster happened on the truncal distribution in the body. So that's why we discounted. So we observed certain things and we challenged the wisdom, so-called wisdom, that it happens, the reactivation of the herpes zoster happens in the areas of repeated trauma. Because repeated trauma, yeah, microphone, okay. You do not want to miss one word of me, even one word of me, that's okay. So, all right, so that was Manipur, how observation. This is from Nagaland, so remained home. And by talking with women and the boys there, we could figure out the drug-sex interface. And it was way back in 1992. And so you see, here you don't need a quantitative study, because quantitative study already revealed HIV epidemic, but the transmission, further transmission possibility and diffusion of HIV in other population groups, that got unraveled through this study, qualitative study on drug-sex interface. Now, this is Unnao. Dr. R.M. Pandey said something. He said that before quantitative study, you talk with the people, and this is Unnao. Unnao is a district in UP, and there suddenly HIV outbreak was unknown, known. So it came to the surface. And then we talked with the people and realized these members are working, sort of working with the panchayat members. And then you see, we set up even a laboratory in the field, the interviews, and we realized that there will be one guy with a cocktail of medicines, including steroid, and if Prem has 20 rupees, he gets 1 milliliter of shot from my syringe, but then Suvarna Roy, being a director of an institute, has more money, you know, so he gives me 30 rupees. So for his illness, from the same syringe, I give you 2 milliliter or 2.5 milliliter. That's the way it is going. So in Unnao, we could not figure out any injection drug use related issue, any commercial sex work related issue, but HIV outbreak happened, and that was happening in a therapeutic setting. So this was a qualitative study got published, and then in Manipur, we saw the herpes zoster epidemic happened following HIV epidemic. We called it satellite epidemic. But during CAM, This is COVID-associated mucormycosis. You see, actually, I do not know why I was in both the places. In Manipur in the early 90s, early 90s, 1990 and 1992, I was there for 4 years. That's why Manipuri khara khange, Manipuri khangdre, khara khange. Little bit I know, but their herpes zoster happened in the form of an epidemic, following HIV epidemic. That was a satellite epidemic. And here you see the long bluish, the bars. That's like the COVID happening, the bar, but the red line actually followed COVID happening because of the associated treatment with steroids and other pathophysiological factors due to COVID, which led to mucormycosis. So the mucormycosis epidemic, as recorded, but the mucormycosis epidemic happened after COVID epidemic happened, and there was a clearly gap of 1 month, 1.5 months, as you can see. So this is very, who can identify the festival there? Winter Scenery Fest. Where does it happen? Mizoram. And this is from the Press Information Bureau. They're talking about from stigma to solution. And the stigma to solution is about HIV self-test in Mizoram. I showed you that graph where I talked about the mixed method study. So that's exactly what happens. If you can do an implementation research of that kind, where you had a huge, number of people working together, then there is a recognition that yes, HIV self-test can be a game changer, and it was actually accepted by the youths in Mizoram, and that's a great example of how do you choose your research design. And if you write your manuscript in such a manner where all these sort of come up, you succeed in terms of getting your study published. So when I became the editor-in-chief, I asked everybody, I do not know Dr. Prem, but from my medical curriculum, we always used to believe the seniors, exam is near, please tell me which book to read. Chota, small book. I do not want to read the thick books and all of these. So the same phenomena, the attributes I had when I became the editor, I asked gurus, what attribute should I have to become a director? He said, get your skin thick. And so I made my skin, I did something so that my skin becomes thicker, and then I, these days, look mostly like this when I look at the mirror. Because I not only convey the bad message, when I go to the workshops, some people drag me to the side and say, sir, that paper which I sent you, sir, you did not publish that, sir. I said, yeah, that's true. And it so happened, I remembered this particular woman, I'm not going to tell you the name, she is from one of the ICMR institutes and in the ICMR, but she was very nice in Gorakhpur. They sent a study which is about a tribal group. The ethical committee clearance said it's a community-based study, and all the participants were from jail, from prison. I said, wait. So that means this whole tribe is in jail because your ethics committee clearance is about the community-based study. So you see, we take ethics so lightly. And then she goes, yeah, sir, that was wrong. I said, that's why we could not take it, you understand? So basically, don't worry about me. There are occupational hazards everywhere. So the journal editors also have occupational hazards if you like it. But that's okay. You now know, I guess, some of the issues that you raised, how to deal with them. Community-based observations and studies as well. You can collaborate, can do with other states, but you also have issues that can be addressed, be it qualitative or be it quantitative, but each requires its own considerations, and all the best. Thank you. What is the time now? This is 2. So I think that we should break for lunch, but before that, if you have any burning question, please feel free to ask. Otherwise, I'm available over lunch. If you have anything burning now, just raise your hand and ask me. Otherwise, we will break for lunch, I guess. Is that right? Okay.
Thank you so much, sir, for addressing all our publication struggles. and for giving us a little peek into the backstage as well. Thank you so much. I request the organizing team for the felicitation, and I would like to request Pandasa to come forward. And I would like to request our director, sir, again to kindly hand over the memento to Pandasa. I know it is well past our lunch time, and I hope many of you are hungry. But what my economic professor told me in college was that the hungry are not angry, so I hope none of you are angry. But he went on to say that the moment the hungry become angry, there is revolution or change. So even if we get angry because we are hungry, there's a way of change. So bear with me. So just if there's any quick 1-2 question regarding this session, sir will be happy to address. Just a quick 1-2 question, or sir will also be able to address after lunch.
So I had a question. So while giving a title to our so while giving a title, what would you suggest? Would it be a very conclusive title, or would it like stir up some sort of curiosity in the minds of the readers? What would you as a editor would suggest?
Do you want your story to be read by the reviewers and the readers when it gets published? You want that, right? Any one of us would be liking that. So don't give all the suspense out, and never be declarative. Like I have seen papers, particularly from basic science, it talks about the declarative statement in terms of a particular pathway, the way it works. But if you have a discovery like structure of DNA, I can tell you that maybe, but no, declarative titles are not to be, but these guidelines, if you are having an observational study, STROBE guideline says that you have to mention about the study design and then the study that you have done. So do it in such a manner that it gets the hook, the right kind of hook, but you do not want to say all that, and then it becomes a very unmanageable long title which nobody would read, and maybe that tiredness, that sense of tiredness would creep in. So you have to be very very moderate in terms of how much to say, but you say enough so that people feel interested in reading it further. Because from title they would go mostly to abstract. If people have gone to your introduction, you are very lucky. Then probably it will flow well. So that flow is very important. Okay. Yes, please.
About the audience of the paper, so it would be like very specific to some discipline we are targeting, or that one or slightly broader one.
Okay, so audience of a paper. So naturally, actually, the paper gets into a journal, right? So the journal has a fixed audience, that's for sure. If you categorize the journals, so if you choose your journal, it will go to the intended audience. So that audience, even knowing that this journal particularly produces or publishes articles of their interest, the criteria remain the same. Whether they would read even your article after getting published is dependent on the way the title flows to the abstract and the abstract flows to the introduction. And then I'm bad on many counts, so a bad reader like me goes straight to the discussion, okay? And then if the discussion is, then I come back to the results, and then I go even to the references. But the references I read the last. But results I don't read even. Abstract, if it is interesting, I immediately go to the introduction and then to the discussion. That's what I do. I'm not saying it's a good practice, but I'm saying that there are readers like that. Any other?
Sir, I had two questions. First is that traditionally we used to write papers in third person. Nowadays many people do write, we have done this, our results show. So what is the editor's take on that in present scenario, what do editors like? And second is about the title again. Some people do put a question mark also on the title. So what is your
good.
thoughts?
Both the questions are good. So one is that you will see more of first person relating to the study implementation and the results in the qualitative research. Even in the quantitative research, these days we see that. So long you are not, or it doesn't sound beating your drum, own drum, you are okay. But you can say the methodology has been described us by earlier, and then you give the reference. So there you are using the term us or we, that's okay. In the discussion also us or we, but in the introduction generally it is avoided. It said that this study or this investigation actually in this context or against this background, this study, the present study intended to. So that's one. And the second one was about question mark. Question marks, generally they go okay in terms of the short research communication, particularly the letter to the editor, where you are raising an issue but not necessarily it's an original research. So in the original research, generally the question mark is not given because you have your standard guidelines, but question marks are okay so long you want to attract the attention of the readers on a particular issue, which are generally shared in the form of letter to the editor or brief research communication, etc. Any other? Okay, I don't see any hand. Do you want to? No. Okay. All right. Thank you. Thanks.
Thank you, sir. This session on ICMR publication ethics and guidelines, this session we will postpone it post-lunch. So we will break for lunch now. Although lunch is given 1 hour, so possibly we can finish within half an hour because we have so many sessions lined up for the post-lunch session. And since this is the last day, we would want to finish all the sessions. So if we can come back by around 2:45, let's make it a collective effort so that we can start early and finish on time. Thank you so much. This session will be continuation of the sessions we had in the first half. This session will be taken up by Dr. Saurabh Jyoti Padgiri, Scientist E of ICMR, RMRC, Northeast Dibrugarh. So I would like to request Padgiri sir to kindly take your time.
This is not working.
ইয়াৰ পৰা লগাই দিয়া ইয়াৰ পৰা কৰি দিয়া এইটো ভিডিঅ'টো নহয় এইটো বেলেগ হয়
So welcome back and a very good afternoon to all the dignitaries, experts, participants, my senior colleagues and friends. So to begin with, I think I have to thank all the eminent speakers who have spoken before me, because all of them have made my job a lot easier, and it will be very easy for me to explain the publication ethics, because all the major work has already been done. So the title of my presentation is ICMR Publication Ethics and Guidelines. So I shall be speaking primarily about the guidelines on publication ethics that are published by ICMR, and I shall also be touching upon a few other recommendations from other reputed organizations such as ICMJE, COPE, and WAME to supplement the ICMR guidelines. So let's start with the presentation. So if we look at the research workflow and progression, we shall see that as DG sir said yesterday, everything starts with an idea. Once we get an idea, we actually have to confirm whether anyone else had this idea before. And for that, we have to do a detailed literature review. After doing a literature review, we will be able to develop research questions and identify research gaps. All these have been dealt with previously. After we do this, we will select our research design and develop a detailed protocol or a methodology for our study. After that, we have to take the necessary approvals, for example, the Institutional Ethics Committee, IRB, and all other, in case of animal studies, Institutional Ethics Committee, animal. After all these approvals, we'll actually start our study, and we'll start collection of data. We'll analyze the data, interpret the results, and draw conclusions. And at the end of this very journey, the time will come for us to communicate our findings. And these findings will be communicated through publications. So how to write or frame a manuscript, all these details have been discussed. So what we'll be basically discussing in this session are the ethical guidelines that we need to keep in mind while submitting an article for publication in any journal. So in today's environment of cutthroat competition, it is a common saying that we have to publish or perish. But to that, I would like to add that if we do not follow the proper ethical guidelines for publication, then also we will perish, and this may in fact be quicker. So the basic objective of this slide is to just emphasize that research integrity and publication ethics go hand in hand. Without a good research protocol, without good research integrity, we cannot have an ethical publication, we cannot have a good publication. In fact, the ICMR policy on research integrity and publication ethics, also known as ICMR RIPE, states that integrity is the cornerstone of credible biomedical research. So integrity is at the very top of this pyramid. And ICMR's policy aims to uphold the highest ethical and professional standards. And the third cornerstone of this policy is to focus on responsible conduct of research, which is also known as RCR in short, and ethical publication. So the basic objectives of this presentation will be to understand in short the basics of publication ethics. So here we'll be discussing about authorship, who can be an author, what does not merit authorship, all these things will be discussed. Then we'll look into a very important aspect, which is plagiarism. We'll look at what is plagiarism, what are different types of plagiarism, what are the tools that are currently available for detection of plagiarism, and what are some of the strategies which can be used to avoid plagiarism. Then we'll look at data presentation ethics. We'll try to understand what is fabrication, what is falsification, and we'll also try to discuss a few points which can help us to select good journals for publication. And recently there is a lot of use of artificial intelligence in publication. So we'll also try to discuss what are the ethics that or ethical principles that we need to keep in mind while using AI in publications. So the basic points will be discussed in view of the ICMR guidelines, and we'll also try to discuss a bit on how these guidelines can be implemented at an institutional level, because for ICMR and its institutes we already have this guideline, but for other institutes, for medical colleges, we can kind of modify these guidelines a bit and make a workflow which can be implemented at the institutional level. So along with this, we'll also supplement the guidelines which are available from reputed bodies internationally, such as COPE, ICMJE, WAME, and we'll try to supplement the ICMR guidelines with these recommendations. So let's start with authorship. So there was a question in the previous session regarding authorship. So ICMJE, or the International Committee for Medical Journal Editors, has clearly given 4 specific criteria for authorship. The first is substantial contribution. So anyone who is an author or who wishes to be an author of a publication must make substantial contribution to the conception or design of the work, or the acquisition, analysis, or interpretation of data for the work. So substantial contribution is a must. Second criteria is drafting and reviewing the actual manuscript. So many authors who are to be given actual authorship, they should be involved in the drafting and reviewing phase of the manuscript. And the third criteria is final approval. So all the authors should be in a position to finally approve the manuscript before submission. And the last criteria is accountability. So all the authors should be accountable, they should be able to take responsibility for the publication. If you look at all these 4 criteria, we shall find that every criteria ends with the word and, A-N-D, which primarily means that to be an author, all these 4 criteria should be met. If only 2 or 3 criteria are met, then this does not qualify for authorship. All the 4 criteria must be met by each and every author of a manuscript. So what are different types of unethical authorships that we commonly see in research settings? The first and the most common one is gift or honorary authorships. Many a times we may name our heads of departments, directors, or any other person, any other influential person with the hope that our publication will get kind of better acceptability. So this kind of gift or honorary authorship is actually against the ethical guidelines of publication, and this should be avoided. Then we have ghost authors. This is actually the result of paper mills who who recruit authors or writers against a particular fee. So these authors prepared a manuscript with our data and they give it to us against a particular fee, and in that manuscript the names of the actual authors are not mentioned and they are even not mentioned in the acknowledgement. So that is an example of ghost authorship which should also be avoided. So as we have 4 criteria for qualification as an author, we also have 4 criteria for situations which does not qualify for authorship. The first is any person who is involved in fund acquisition. That person actually cannot be named as an author if he does not qualify for the other essential 4 criteria. Supervision of work, minor laboratory support and administrative support also does not qualify for authorship. Few additional key concepts that we need to keep in mind in this authorship are as follows. First is acknowledgement. So people who do not meet the 4 criteria, they may be meeting 2 or 3 criteria, need not be included as authors. They can be mentioned in the acknowledgement, and this in fact is a part of every manuscript that every big publishing house now demands. Contribution. Individual author contributions are also mandatory, almost mandatory in major journals nowadays, and this should be mentioned. The person who has written the manuscript, the person who has actually done the work, data analysis, everything should be mentioned against each particular author. Then we have group authorships. Group authorships are also ethical, but this group should have a particular name, and this naming is actually the responsibility of the corresponding author. And just as a group, in order to qualify as an author in the group, each individual author in the group should again meet the 4 eligibility criteria. Coming next to guarantorship, this is actually the responsibility of the corresponding author, and right from preparation of the manuscript, submission to final publication, the entire responsibility lies with the corresponding author. Last key concept to be kept in mind is the use of artificial intelligence in manuscripts. So nowadays a number of text generating chatbots are used in publication, and it is not unethical to use artificial intelligence in publication, but whenever such technologies are used in publication, it is the responsibility of the corresponding author to actually mention it and describe it both in the covering letter as well as in the acknowledgement part that AI has been used for this and this purpose. And chatbots cannot be listed as authors. So coming next to image manipulation, yesterday Subarna Roy sir has given a number of examples of image manipulation, so I will not give many examples here. So what we need to understand is that there is a very thin line between what is acceptable and what is not acceptable in case of image manipulation. Sometimes we may make some minor adjustments to the brightness, contrast of an image. All these kinds of modifications are actually acceptable. On the other hand, if we do these modifications to such a degree that certain vital elements are hidden, then it qualifies as unethical in case of publications. For example, if you look at the image on the left, there are 4 images. The images on the left hand side are actually the original images, and the manipulated images are on the right hand side. In the first set of images, we shall see what we can see is that the band in lane 3 is completely removed. So this actually is unethical and should not be done. In the next image also, we will see similar removal or manipulation of the particular bands, and these things should not be done. So the Committee for Publication Ethics actually has a very good webinar on image manipulation, how it can be detected, what are the tools that are used for detection of image manipulation. So that is available as a webinar. I've shared the link to that webinar, and if anyone is interested, you can go through that in detail. The next thing that we need to keep in mind is the protection of research participants. Any kind of identifying information, whether it is the name of the participant or anything which may reveal the identity of the participant, should not ideally be published without the written informed consent of the participants. So this we usually do, we take the consent of the participant in order to publish any identifying information, but what the guidelines say is that those patients whose data will be published or data will be shown in a manuscript, they should be shown the final version of the manuscript before submission. So that part is usually skipped, and that should not be done. Informed consent should also be obtained if there is any doubt that anonymity cannot be maintained. So in the left, if we see an image, this is a common kind of image that we see in, especially in case reports. We show the photograph of the study subject, and we mask only the eyes. So this kind of masking cannot ensure that the patient will not be recognized. So it may still be possible to recognize the patient, and so the anonymity cannot be maintained. So in these cases, we have to take a separate informed consent from the study subject. So the next thing which is very important for us is plagiarism. Actually, there are different definitions of plagiarism. I am highlighting two definitions here. One is from ICMR, from the ICMR RIPE policy, and this is actually a modification of the ICMJE recommendation. According to this guideline, the wrongful appropriation and stealing and publication of another paper or another author's language, thoughts, ideas, or expressions, and the representation of them as one's own original work or duplicating one's own publication. So this is the definition of plagiarism. If you look carefully, there are 4 components here. It's not just words. Even thoughts or ideas, if it is taken from someone else's publication, that also qualify as plagiarism. And the third and fourth components are, it may be someone else's work, or if we lift components from our own publications, own previous publications, then that also qualifies as plagiarism. The World Association of Medical Editors actually defines plagiarism in a different manner. It states that a condition where 6 consecutive words are copied or 7 to 11 words are overlapping in a span of 30 words. So these numbers are important for us: 6 consecutive words and 7 to 11 overlapping words within a span of 30 words. So both these definitions are used for defining plagiarism. So there are different types of plagiarism. The first type is direct plagiarism. Direct plagiarism, as the name specifies, it is the complete or partial direct copying or word by word copying of someone else's work without acknowledging the original author. So this is direct plagiarism. Self-plagiarism is the same, only here the author will refer or take up material from his own work which has been published earlier. So there is also a kind of plagiarism known as mosaic plagiarism. Here, as the ICMR definition says, it's not just the words, but even copying ideas and the general structure or concept from someone else's publication, that qualifies as a type of mosaic plagiarism. Accidental plagiarism, where the author may not be aware that he has actually plagiarized, or he may have forgotten to acknowledge the actual source. So that is kind of accidental plagiarism. Redundant or salami publication, as Sir has already said in his presentation, when we try to divide our work or project and make a number of publications from a single work, then that qualifies as salami publication. So how to check plagiarism. This is very important for us, and a number of tools are actually available for detecting plagiarism. Many of the tools are free, but most of the good ones are paid and they are expensive. So I have experience working with Plagiarism Checker X previously. That is comparatively cheaper. I think the license fee is around 3,000 rupees and it is perpetual. But the one that we currently use at ICMR and which is recommended is Authenticate. And this is one of the best plagiarism tools available currently. But the problem is this is a bit expensive and the license fee is around 6 lakhs per year. But the good thing is that this can be used by a large number of participants. The basic license package that we have can be configured with around 100 users. So if a university purchases one license, then that single license can be used by a number of institutes under the particular university. So I think that can be used by all the researchers. So I'm just showing the dashboard of the Authenticate software. It's a very simple dashboard, and we have document upload or text copy facility. We just have to upload the document, and once we start the program, it will give us a report in this manner. So what we have to actually keep in mind is the number at the extreme left of the report, which is the similarity score. Here it's showing 5%. So as per the ICMR guidelines, the target should be less than 10%. And this is less than 10%, not even equal to 10%. So it is basically equal to or less than 9%. The next important consideration in publication ethics is the use of artificial intelligence. So ICMR actually has published ethical guidelines for use of artificial intelligence in research, but there is no mention of the use of AI in publication as such in this particular policy document. So we have to move to other organizations such as COPE and WAME, which have actually given a few recommendations for the use of AI in publications. So regarding authorship, the guidelines are clear. AI tools cannot be listed as an author of a paper. We can use AI tools for drafting a manuscript ethically, but these chatbots or AI tools cannot be listed as an author. Transparency is the second requirement. If AI tools have been used in drafting a manuscript, then this should be declared by the corresponding author in the acknowledgement part and also in the cover letter submitted at the time of publication. And the responsibility cannot be shared with the artificial intelligence tool. It is the sole responsibility of the corresponding author. And along with the responsibility, accountability of the use of AI also lies with the authors. So there is an interesting interview by a journalist called Todd Carpenter. He had an interview with ChatGPT, and the question he asked was that, is it ethical for an author to use AI in developing a scholarly article? So the response that ChatGPT gave was that there is no inherent ethical issue of using AI in research or writing as long as the AI is used appropriately and ethically. This is actually published in a COPE editorial in 2023. So even AI is recommending us to use AI ethically, and this should be kept in mind. So there are a few guidelines for avoiding plagiarism that are specified in the ICMR RIPE policy. The first thing is acknowledgement. Whenever we use material from some other publication, we should give proper acknowledgement to that original source. The second is reframing. So whenever we want to use a set of words or set of data from a particular publication, we should try to reframe it so that it is not exactly the same as that of the original publication. So reframing is important, and reframing also helps us to reduce the plagiarism similarity score in the different tools that we may be using. And the third is preventing manipulation of references. So we should only cite those references which are relevant to our work. We should not try to manipulate and include irrelevant references in our work, and this is also highlighted in the ethical policy. So in data presentation, there are two concepts that are to be kept in mind. The first is fabrication and the second is falsification. So fabrication actually is a kind of manufacturing data. We have not done any particular experiment, we have not done any work, we are just manufacturing data and we are trying to publish it in a particular journal. So that is kind of data fabrication. On the other hand, falsification is a kind of manipulation. So we have actually done some amount of work. We have the data with us and this data is original, but after data analysis, we may find that the data is coming out to be non-significant. So in order to make the data significant, we may try to manipulate our data. For example, we may remove few outliers from our database so that the ultimate results are significant. So that is actually falsification, and both fabrication and falsification should be avoided. So image and digital manipulation, I have already spoken before. This should be avoided at all cost. So once our manuscript is ready, following all the ethical guidelines, our next job is to actually select a good journal where we can publish our manuscript. Here we have to keep in mind the predatory journals, and there is a big menace these days, and it is really difficult to actually identify which journal is predatory and which journal is not predatory. So there are few characteristics which are shared by almost all predatory journals, and most of these journals are usually online. They have immediate or overnight publication. There is usually no peer review process, and they are either free or have very low article processing charges. If we find any journal meeting these criteria, then we have to be careful because this may be a predatory journal. In fact, UGC has issued a public notice in 2019 where it has said that any publication in any predatory journal should not be considered for academic credit or selection, confirmation, promotion, performance, appraisal, award of scholarship or academic degrees. So a list of good journals is available from UGC in the UGC Care website. I've just provided the URL for this website. So if you face any difficulty in selecting a journal, good journal, then we can go through this list, and depending upon the subject of relevance, we can select a particular journal. In addition, there are few other tools from big publishing houses which may help us to select a good journal. For example, Elsevier, Taylor and Francis, Springer and Wiley. These publishing houses actually have a journal finder tool wherein we need to copy and paste our title as well as our abstract. So based on the title and the abstract, the tool will give us a list of good journals which are relevant to our topic mentioned in the manuscript. So these tools can also be used to find a good journal. Now coming to the ICMR Research Integrity and Publication Ethics Policy, this guideline was actually published in the year 2019 under the leadership of our previous Director General, Dr. Balram Bhargava. And the ICMR Bioethics Unit, headed by Dr. Roli Mathur, actually had a key role in publishing this particular policy document. And the purpose of this policy is actually to ensure ethical conduct of research, to promote accountability, transparency, and responsible authorship, and to address and manage conflict of interest and misconduct fairly. So this policy actually applies to all ICMR institutes, all ICMR staff, even contractual staff, and it covers research at headquarters and all its institutes, even field units, and encompasses all stages of biomedical and health research. So according to the ICMR RIPE guidelines, the acceptable level of plagiarism is less than 10%, and plagiarism check is mandatory before submission of any article to any reputed journal. And the final draft before publication has to be submitted with a research integrity undertaking, which is also present in the policy document itself. This can be downloaded free of cost. Along with this research integrity document, the entire manuscript has to be submitted to the institutional framework. For us, it is either the director or a research integrity unit headed by the research integrity officer. For other institutions, you may have a similar framework in place. So this has to be submitted prior to submission in the journal. So next we'll come to a few components which can be actually included at an institutional level. Not necessarily an ICMR institute, but other institutes can also have this framework in place for practicing good research integrity and publication ethics. The first is a unit or institutional framework, which may be either the RIU or research integrity unit, the bioethics unit, and a research integrity officer. Ideally, what the guideline says that we should have 2 research integrity officers in a particular institute, because the research integrity officer may also have his own article or manuscript. So if that is the case, then the second research integrity officer will take over and assess the particular manuscript. Training and sensitization, which is done by the research integrity unit, is the second component, and this needs to be in place in every institute, and this can be done through workshops, journal clubs, or guest lectures. And finally, there should be a framework for misconduct reporting and investigation. So this is usually done by a committee appointed by the head of the institution or the director, and it comprises of 2 to 3 members, and the penalties are based on the degree of research misconduct. So this is my penultimate slide, and here I would like to show a flow chart which is existent for ICMR institutes. This can be modified for different institutes. I'm sorry for the small print. At the very top of this flow chart, we have the bioethics unit. This unit actually frames the different guidelines and policies for research integrity and publication ethics. And this is in turn implemented by the research integrity unit. So after we have the guidelines in place and the research integrity unit has implemented the policy, we can undertake the research activities after following all the necessary ethical considerations. And once our manuscript is ready at the end of our study, we have to do a mandatory plagiarism check. So it may so happen that we have a plagiarism detection tool, and some institutes may not have a plagiarism detection tool. So in cases where a plagiarism detection tool is already available, the author himself can perform the plagiarism check, and if it is less than 10%, it needs to be submitted to the director or the head of the institution directly for approval. In case the plagiarism checker tool is not available, then without checking the plagiarism, it can be submitted to the director, and the director will then forward it to the research integrity officer, who will perform the plagiarism check. So if everything is okay, if the plagiarism score is less than 10%, then the article can finally be submitted for publication. If there is any misconduct, then depending on the type of the misconduct, we can either send the manuscript back to the author in case of minor misconduct, or if the misconduct is at a severe degree, then we can constitute a 2 to 3 member committee which will look into the research misconduct, rectify the issue, and do a recheck of do a recheck for plagiarism, and then finally submit the manuscript for publication. So basically the take-home points from this presentation are as follows. Number 1 comes integrity, and the ICMR policy emphasizes integrity at every step of research. and all stakeholders must work together to maintain research credibility. Integrity and credibility are very important. The training and sensitization program is also very essential, and it should be an ongoing process. The number to remember from this presentation is less than 10, which should be our target for the plagiarism score. And the rest of the things are that need to be kept in mind are the authorship criteria, who can be an author and what does not qualify for authorship. We should prevent or avoid predatory journals, and whenever we are using artificial intelligence, it should be done ethically. And finally, the institutional framework should be in place to deal with research misconduct. So that's all from my side. I leave you with this quote, which is self-explanatory. Thank you. If there are any queries, then I can take it now, or yeah.
Hello, sir. In your presentation, you said about UGC's academic progress and usage of publications there. So, is there any difference that someone is first author, someone is second author, or third author? So, the significance or the contribution of that author are serialized in that order. There is some kind of order like that? The first author is the most significant, second author is not that so, third order is not at all, this type of thing. Because in UGC academic progress, their form or their regulations, first author gets the most number, second author or third author not so. So can you please explain on that?
Yeah, actually there is the order for authorship, and there is no written guideline as to who can become what, means what is the sequence of authorship. That actually needs to be defined at the beginning of the study, because usually when we look at maybe 5 years before now, the first authors and second authors used to get the highest credit. But in our network, the first author and the corresponding author get the same amount of credit, and that needs to be decided by the entire group of authors as to who is to be the first author, who is to be the second author, and simultaneously who is to be the corresponding author. Usually the corresponding author takes the full responsibility, and usually he is the person who is actually submitting the manuscript to the journal, and he is the point of contact between the journal and the entire research group. When there are revisions and rejections, he will be the person who will be communicated.
So another question is that you have shown us some original figure and some manipulated figures.
Yes.
So when a author, if someone does that,
yeah,
so that person is submitting that paper with all those say manipulated figures.
Yes.
So how can the journal identify that this is a problem or there is some manipulation is there? Yeah. And another issue is in case of the authors, if some author feels that I should have been the first author or I should have not been this type of this. So is there any forum to raise any complaint?
Yeah, yeah. So coming to the first query regarding image manipulation, nowadays the big publishing houses have very sophisticated software for detection of AI induced image manipulations. So those tools are being used regularly, and the funny part is that they are not using it now, they are also using it retrospectively on papers published 10 years before. So many papers which have been published in 1990s or 2000s, even those are being retracted now because of the detection of AI induced image manipulation. So that is the first thing. And second is regarding the order of authorship. So when we are submitting a particular journal, we have to, the corresponding author actually has to fill up the names of the authors in any particular order, and after submission, the journal usually gives a mail or notification to each author, because every author's email ID will be there, and it will ask the authors to confirm their authorship. So if someone wants, he can also reject his authorship, or if someone is not happy with the order of the authors, then he can raise a query at that point. But it's usually not a very good thing to do, because that will reduce the credibility of the manuscript, and it is the best thing to do is to decide the order of the authors before submission. In fact, even at the time of initiation of the research, this should be decided.
So you showed an image wherein you showed that the subject, we have to not disclose the identity, we have to blacken the, normally we show that.
Yes.
And we are taking the written informed consent also, that we will not disclose the subject identity when we are taking the patient. Then what else is required? You were saying something on that.
Yeah, actually, for publishing the photograph with masked eyes, we will need the consent, written informed consent from the study participant. Along with that, we will actually have to show him or her the manuscript or the final approved version of the manuscript before submission, that this is the manuscript, this is your picture, this is actually what will appear in the journal. Apart from that, if we just mask the eyes, then we cannot ensure that anonymity will be maintained, because someone may be able to recognize that person even when the eyes are masked. So we have to take a declaration to that effect, that we are publishing your image with your eyes masked, but even then you may be recognized by someone who is known to you. So we'll need a declaration from you that this is acceptable to you. So 3 things are required.
But sir, that already we are taking in the return informed consent, that we will be using the results of this study for publication purposes, and already we are informing the subject about it before we are taking his consent.
That is
and then what else we can do, because we are masking the eyes? Is there any other way you can make it anonymous? What we should do?
This is usually done in case of case reports where there are some substantial findings in the face or any part of the body. So
that we are doing with the eyes.
So if we are masking any other part of the face, then our study findings will also be masked. So then it is basically useless to have that photograph in that particular
and in
case
the
report.
Practical scenario, if suppose you have 30 or 100 or 200 participants, then sometimes the participants follow-up is not there.
Yes.
So it's difficult to get the same participant back, and then because manuscript is prepared maybe a year down the line.
Yes.
Results are more than 1.5 years back. So getting the same person back, maybe contact is not possible. Then how you are going to ensure that? What you are saying that we have to bring in and show him.
Yeah.
So that is a, I think, practical difficulty also.
Yeah, ethically or theoretically speaking, if that consent cannot be obtained, then it is better not to publish that particular image. That is the guideline. If we somehow manage to get the consent at a later time of manuscript submission, then it is okay. But if it cannot be taken, then it is better not to show that image in the publication.
Yeah, I just wanted to add for your this thing that we can have co-authors, co-first authors, co-corresponding authors. So in that way also some small things can be solved. Like we said, no, that we want to. And I want to ask one question.
Yeah.
That is like, if suppose there are students who are coming up with data where the figures are manipulated and all, how do we analyze?
So that is a situation that has actually happened with a lot of principal investigators and project investigators, HODs. So at that time we did not have the tools, but now tools are available, and I think we should be using those tools. In fact, the newer versions of Authenticate, there is a proposal to include AI detection tools in these softwares. So if those packages are available to us, then I think we should be using those, because anyways the journal or the publishing house will be using those tools, and even if we do not detect the manipulated images at our level, it will finally be detected at the journal level. Maybe not now, but maybe few years from now. So we will be in trouble.
Sir, sometimes when we study, so what we find is that from different journals, we see that there is one article. So in that article, it will be written, it is part of this project and supplementary publication. So we will be getting the link for the different publication.
Yes.
So isn't that a slicing of publication? Because in the previous session, we were discussing we should not we should not publish partial result. So that is one part. And secondly, sir, sometimes what we do is that research ka methodology is being published, or the protocol development process is getting published, or interim result of some big project is being published. So are these also part of that salami publication, or these are ethical?
So actually there is a very thin borderline of between what qualifies as a salami publication. So there are many multi-centric task force projects where it is not possible to include all the results in a single manuscript, because these may be long-term prospective studies spanning 5 years, 10 years. So in these cases, it is completely acceptable to publish interim reports and even parts of the study as an independent publication. But there are some certain studies where maybe the duration is 2 years, 3 years, and the same kind of work is being done, and that is being divided and shown in different publications. So that is not acceptable. But in case of large multicentric studies, very big studies, it is acceptable. It is not unethical.
Sir?
Yeah.
Sir, as we know, UGC has discontinued UGC Care from last year. So is it, till now, can we publish which are the listed journal, or whether these have some meaning, extra meaning in terms of UGC?
Yeah, actually UGC Care has not discontinued that list. The last update is 2024 update. So the final update is last year's update. So that list still stands. So we can select journals from there, and if required, we can use the other journal finder tools which are available from big publishing houses.
But it was insignificant, I think, sir, because they have given a public notice like that. They are not going to update more journals.
Yes. So we will not have new journals in that list, but the previously existing journals are completely fine. We can select from that list. Okay, thank you.
Thank you so much, sir, for your elaborate presentation on ethics in publications, and for answering and addressing the doubts that participants have raised. We would like to felicitate Padgri sir as well. So I request Padgri sir to kindly come forward, and I request our organizing team to kindly bring the memento, and I request Director sir to kindly hand over the memento to the resource person. Thank you, sir. I would like to request Director sir to kindly remain, as we want to felicitate Dr. R. Laxminarayan sir also with a gift. Sir, kindly come forward, sir, please. Sir has been instrumental in getting this workshop approval process, and as well as for getting funding, arranging and organizing, and even being conducted here. So he has come and being with us physically since the beginning of yesterday, and he will be with us till the end of the session. Thank you so much, sir, for randomized clinical trial. This session will be taken up by Dr. Suman Kanungo, Director of ICMR RMRC and Dibrugarh. Sir, please take your time.
Where is that?
Okay, good afternoon. What I was planning to talk, some of the things have already been discussed. Like you have an extensive session on study design, where we talked about experimental as well as observational study designs. So, and Dr. Premani has talked about 4Ds, the descriptive, mostly the discovery and development are basically, development, keep it aside, discovery and descriptive are basically kind of description which comes from your descriptive epidemiology. I mean, kind of addressing a problem, finding out the disease burden, case series, case reports, these are. So where we do not, where the investigator do not provide any kind of say intervention. But that was he talked about. From one part he talked about, he touched upon, and Dr. Pandey also touched upon, is a cohort study. Cohort study, if we consider the time span, we look forward from one. Temporality is a major issue, a major point in discussion of a cohort study compared to the, when he was discussing the different type of study designs. You must have noticed that the temporality is a major issue in deciphering the cohort and case control study. So experimental randomized control trial is a modified, is a cohort study with intervention. He talked about PICO, I'll talk about PICO as well as FINER. So let's see, some of these things are common. So I was not going into that. So this is the study designs that he talked about. Under the descriptive is the case report, case series, cross-sectional. He showed beautifully, provided some examples. And case control studies and cohort studies. Sometimes case control studies look like cohort study in rare disease assumption, when that is a different issue. That's a deeper epidemiology, where when we talk about, say, cumulative incidence divided by, when we say risk ratio, it is basically cumulative incidence divided by 1 minus 1 is the full probability minus the cumulative incidence. If the cumulative incidence is very low, then your RR becomes CI. When your OR becomes CI, becomes RR. That is in under rare disease assumption. Sometimes through odds ratio we can find out the incidence rate ratio. That is different. So what we see the distribution, we find out the determinants, and we find out, try to have its application. In experimental study, the experimental study is basically the best one is the randomized control trial, and he talked about non-randomized control trial. So I will not go into the non-randomized control trial. He gave beautiful examples. I'll talk about RCTs only. And considering the evidence generation, it is of the highest. Still, it is not the highest, because one randomized control trial will give you a very robust kind of evidence, but it will not provide the final one until and unless we do a systematic review and meta-analysis. And again, that systematic review and meta-analysis can be graded also. Whether the review articles that has been taken, how many, what is the sample size, what kind of intervention, accordingly we can grade that. We call grade analysis. So that is what. If you look at why we do it, why we do studies, why we do research, basically to have a hypothesis generation, either from case report, case series, descriptive. These are basically hypothesis generation, which I talked about the last slide. That leads to your analytic. That hypothesis is being tested. Cohort, case control, cross-sectional, and most analytical epidemiology is basically clinical trials. So this is he talked about. I'm not going into that. Like, okay. So he's not going there. Whether we have assigned the intervention or not. If the investigator, if the researchers provide the intervention, if he or she determines how the intervention will be assigned, in that case it becomes experimental intervention studies. If it is not, as in cohort study, if the intervention or the effect occurs due to the natural cause or the choice of the volunteers, in that case it does not become an experimental study. So whether few things are important, whether it should be randomly allocated or not. If no, it is non-randomized. If yes, it becomes randomized control trial. If there is a comparator group, it becomes analytical. Is direction of exposure. Cohort study, as I said, it is a forward study. Exposure to outcome. Case control study, outcome to exposure. His number four study design was a perfect example, which you missed all. And cross-sectional study is a snapshot study. And it can be again a longitudinal thing. I'm not going into that. So this one I'm not in that. So why we do that? Mostly to identify the causal relationship between exposure and outcome. That is very important. We can do it by cohort study, by case control study. We want to have a reason why it happened and what was the causal association. I'm not going to the causal pie, but it has a lot of things need to fit in to have the causal. So one of the thing is to find out the causal relationship, to evaluate the effectiveness of preventive, therapeutic or public health interventions, to provide scientific evidence, to replicate findings under experimental settings, to strengthen causal inference in disease prevention and control. Last line is very important, to replicate replication. So robustness will lead to the your causal interference and replication. So only randomized control trial of sufficient size can adequately control for known and unknown confound variables to minimize bias. So why I put this line, you, right sir, you talked about bias, right? So this randomization, you all know that it is known or unknown confounders can be tackled, but still not 100% it is tackled. So that's why all the results have alpha. You know about alpha and beta. So my result is as extreme as this. Lower the alpha, less the alpha, the result become more and more stringent and more precise. And that is called internal precision, CI. That is the concept of confidence interval. So the narrower the confidence interval, the precision of the study design increases. So, but still the data reliability increases. But that's why, though we randomize, we try to control all the known and unknown confounders, but still there are chances we open a backdoor pathway or directed acyclic graph. So there we can, there sometimes it happens. Secondly, begins with a valid question, clinically relevant question, greatest impact if limited information or high variability in care outcomes, can be answered by properly designed RCT. All these conditions can be answered by the RCT. Let's see how it is done. So, I'm not talking about non-randomized trial. I'm talking about the randomized trial Why we favor randomization? Ensuring unpredictable assignment. This is more the unpredictability, more is the less chance of bias creeps in. So that can be taken, that can be dealt with how we assign the allocation. How random is your randomness? Sometimes we allocate, say, 1:1, 2:1. I mean, allocation with a permutated block randomization. There are different kinds of block randomization. We do it. So it can be like we randomize, one is the agent, another becomes the control. So now an idea which is finer. Let's picot it. You learned about picot. P is the population, say from where your study population comes in. That is the population. Say this is a typical one of our studies we have done earlier. Safety and immunogenicity of Rotavac and Rotacil vaccine administered in an interchangeable fashion among healthy Indian infants. So here P is the population, who are the population is the P. P is healthy Indian infants. Intervention, the two vaccines, rotavirus vaccines, Rotacil and Rotavac. What is the control? The conventional arm is the control and the intervention arms are the interchangeable. That is the control. What is the outcome? Immune response. There is another thing is T, timeline, at what point of time, how long we are following them up. That is called picot. Now comes the finer. What is F is feasible, whether it's feasible or not, and I is interesting. That is the prominent issue without a solution. It should be novel, it should be ethical, it should be relevant. So these are the finer point come. So designing an RCT, randomization, allocation and concealment and stratification. So these are the few things. I'm not going into details. I'll talk about the phases. I'll talk about the types. So allocation concealment is one thing you have heard, that when we allocate, it should be, if we blind it, you have heard the term blinding. It could be single blinded, it could be double blinded, it could be triple blinded. You all know that. So allocation concealment is basically a type of blinding where when we allocate this is a process that prevents any trial participant or investigator from knowing in advance the treatment which subjects will be assigned. Why I am telling this is a little bit historical, because now all the randomization are done through softwares. We call so softwares had an inherent property of concealment of allocation. So blinding seeks to prevent performance and ascertainment bias, different response to treatment or to measure effect or treatment due to knowledge of which treatment was received. Have you heard of health? This is in relation to, it is called investigator bias. So how we do it? This assignment should be random. One goes to the experiment group, another goes to the control group. So we have an outcome, we have a baseline data on pre-test, in the control group with the pre-test, then experimental treatment, post-test and the post-test. Subject designed randomly on experimental of control group, experimental treatment to those in the experimental group, pre-test and post-test are those measurement on dependent variables that are made before or after experimental treatment is performed. You know dependent and independent variable, right? I'm not going to that. All true experiment designs have subject randomly assigned groups. This randomness is very important. How random is your randomness is. So now, why RCT, why I was telling the RCT is important. If you look at the pyramid of hierarchy of evidence, RCT goes to the very, very high level, and above there is systematic review and meta-analysis, which has got a well support. Structured comparative control experiments in which 2 or more interventions are studies across groups. So this is supported and promising. Expert opinion, background information, these are basically this evidence pyramid. These are at the lowest rung. Systematic review is at the highest level. So this one I talked about. So how we do it? Baseline, from baseline exposure to the final outcome, one major nuance is of an RCT is that the exposure level should be equal among the intervention group and the control group. If you choose somebody who are not balanced, if it it could be cluster randomized, it could be individual randomized. If the differentiation in the clusters are different, we have to take the cluster effect or the design effect, and we have to find out intercluster correlation for that. That is reflected in our sample size calculation also. So there are 6 steps which you know, that is selection of protocol that has been discussed. I just removed all those slides. Selection, reference and experimental population, that is very important. Reference population means your control from where your experimental population comes. Then randomization, intervention, follow-up, analysis and interpretation. So this one is, again, this is very important, that your protocol will define the reference population from your study population comes in. This is very important. If you want to do RCT among hypertensive patients in adults, you cannot, you have to choose impact of, say, felodipine or say recent cilindripine compared to what is existing one. You have to choose patients who are having hypertension, who are 18 years, I mean, if you say adult, 18, so you have to define your age group, which comes from your inclusion criteria to make it more robust. So, and you measure the outcome among the control and among the experimental. So, the protocol says the objective samples all are pre-hoc. Please mind it, you cannot have a post-hoc analysis plan or post-hoc randomization plan. It has to be all are pre-hoc. That means you have to determine it earlier, that objective, sample size, selection criteria, allocation of study group and standard working procedure. If you remember yesterday when DG sir was talking, he mentioned one thing, just one line he mentioned, which is very important. I mean, attending a workshop for 2 days will not make you big researchers. We are all fantastic researchers, but until and unless you do and implement it, most important is you have to have a very strong implementation plan. We call it SOP or site implementation plan, adhering to the protocol. Until and unless we do that, an RCT cannot be performed. So this standard of working procedures, SOP, is very important. Protocol aims at preventing bias, because this protocol and reducing source of error. Pilot studies to pilot, we discussed that the feasibility study to assess efficacy, acceptability or feasibility. So drawing up of protocol has contents in the material method, have study design, study settings, study duration, everything all the speakers have touched upon. So these are the part of a basic protocol. Then the study participants, inclusion and exclusion criteria, number of groups, sampling, size, method, procedure, parameter studies. Parameter studies means your endpoints. These are, you have to clearly define the endpoints which you want to study it. And data collection method, confounding factors, statistical analysis, timeline and budget. So selection of study population, why do it? To make necessary inclusion and exclusion criteria. Those are not eligible, those who do not give consent, those basically are you're excluded. Then randomize experimental groups, control groups, manipulation and follow-up. So selection of study population is simply the reference population. Say I want to study typhoid vaccine. Some actually, I forgot. I think Dr. Pratap Tarian talked about typhoid vaccine. So if you want to study efficacy of typhoid vaccine, I'll choose a population. If his endpoint is my typhoid, I will choose a population where population is vulnerable for typhoid. Say maybe urban slum, maybe in the where the population, where the water supply sanitation is jeopardized. There. So if you want to study a challenge study or something, that is different. The CHIP model is now coming up in India right now. But efficacy phase 2, phase 3 study are done somewhere where mostly the disease is rampant there. So now, and One thing is now talked less, that is generalizability. If I do a study in a vulnerable population in Malawi, it should be same as in Bangladesh, or same in India. But that is a little bit weak association. Now it's an external validation. We mainly concerned about, researchers, we should consider our study as internal validation. Internal validation comes from your precision and your CI. Experimental, from that comes your experimental population, from which your study population. So randomization, it is sequence generation, allocation, concealment, and implementation. So it has got different nuances. Randomness means governed by chance. This is very important. So to avoid selection bias, it should be random. How random? Now our life has become very easy. Previously, in even early 2000, when we used to do it, we used to use random tables for our small studies. But now software has come in, so all these things have gone. So one thing is now, when in RCT, blinding is a very important procedure. What is blinding? It refers to whether patients, clinicians, providing an intervention, people assessing outcome, or data analysts were aware or unaware of the group to which patients were assigned. The goal of blinding is to eliminate or at least minimize remaining potential biases. Randomization takes care of most of the biases, blinding takes care apart from that. So three people can be blinded. One is the participation, single blinded. One is the researcher, single blinded. Then the participant, double blinded, and the analyst, triple blinded. So these are the blinding that has come in. So another method, allocation, concealment procedure. These are the, like I talked, Dr. Premani and others have talked about PICOT. I talked about finer. There is another term called SNOWS. Sequentially, it should be sequentially numbered, opaque, sealed envelopes. This was prehistoric, it's a little bit earlier, so we used to use the term SNOWS. So allocation, concealment, ways to, this is why I'm harping on this one. This blinding randomization is the key to what we do. Basically, that will give your, whole basis of the experimental design. So this is how we used to do that. That is, this is drag A, which number, that is concealed. Even if you put out the light against the light, you cannot see that this one is the, this is how it is done. So random group assignment, I talked about simple randomization, say 1, 0, 1, 2, 1, 0, like that. Block randomization, block of say 2, 4, 6, 8, actually it's divisible. Stratified randomization according to age, according to sex, and this covariate adaptive randomization is there. Another thing I did not mention here is that permutated block. The block size may be permuted. It's not a fixed block size. So that chance of predictability gets reduced. So, previously it was toss of coin, shuffling deck of cards, throwing a die. These are all kind of creating a chance. So when we toss a coin, it is 50-50. If it is probability is 1, we describe probability in terms of 1, then chance of getting the treatment becomes 0.5, that is 50%. So in case of deck of cards, it is 1:52 because the cards are 52 in number. Throwing a dice, 1:6. The 3 control, 3 treatment is something like that. So this is how it is done basically. Say these are control among those. This is. Can you tell me what is the? I've shown it. What is the block of? This is block of. This is a block of 5 basically. If you say the every fifth, but this is 5th, 7th, 9th. 10 patient. There are 10 participants. These 5, 7, 9 are given. So this is random allocation is given. So equal sample size, block should be multiple of the number of groups. If you have 2 treatment groups, 2, 4, 6, 8, something like that. 3, 5, 7 is difficult, but it can be done too. But so limitation is that Again, risk of predictability comes in. That's why nowadays we do permuted block randomization. Imbalance with unequal dropout can happen. Complexity in implementation, sometimes it is there, but it has to be it has taken care of. May not control all confounders. While it ensures balance in group, it does not automatically balance other covariates unless stratified. Because if you say something which is more common in the younger age group or the older age group, then the age stratification is required sometimes. Stratification, I'm not going to that. Limitation of stratified randomization, again, it becomes complicated to implement if many covariates need to be controlled. If there are, say, covariates means which are dependent variable, like along with age there is height. You have to, whether this intervention increases your height or something. So those needs to be controlled for stratification. But number becomes smaller in each sample, in each cell. Baseline characteristics of all participants is available, has to be available for it. In that case, it works better. So this is another one, covariate adaptive randomization. A new participant is sequentially assigned to a particular treatment group by taking into account the specific covariates. Method, Tabe's covariate adaptive randomization, Pocock and Simon method. These are the two methods we usually, because these are bigger one, I'm just telling the names. So, now in experimental group, placebo, you all do it. Placebo is becoming day by day difficult because your ethics said that you have to provide at least standard of care which acts in the control arm as the placebo. But if there is, now we are doing a dengue vaccine trial where there is no standard care of dengue, and at the same time there is no vaccine available. In that case, where there is no definitive standard of care, in that case you can use use placebo. But nowadays in the if you consider the PICO, in PICO most of the time, I mean for control arm, you have to have something which you can compare with the with the intervention one. So it should be inert medication or procedure, no effect, intended to give patients or perception they are receiving treatment. So that is the placebo. Follow-up and maintenance is another thing that we need to, very important once you do the intervention, because as I said that the RCT is basically a specialized cohort study. So as Dr. Mani has said, that cohort study is basically a forward study. There is a temporal, you're exposed, then your effect comes. So your exposure is your intervention. Follow that, you have the forward study. That during that time forward, if you have loss to follow-up, which can be differential, which can be undifferentiated, you will have your effect size undermined, or can go towards the null or away from the null. When we say towards the null, that means if there is an effect, you fail to understand it. That is your type 2. But if there is no effect, but you say there is an effect, that can also happen. It is not effective. The vaccine, the drug is efficacious, but you say, no, my result said it is not effective. That is because there may be loss to follow-up. And the loss to follow-up is, those who are in the control arm is more loss to follow-up. In that case, all these things happen. It can go towards the null. Null means zero effect, or away from the null, more effect. Where there is no effect, you said there is an effect. So when we discuss about say errors are best explained by judge. I mean in the court of law. What Indian Penal Code now, Bharatiya Nyaya Samhita, what it says? No guilty should not get punished. A guilty person can be said, you can go. By mistake, you can say that a guilty person non-guilty, but not a single non-guilty person should be said guilty. So that is also a type 1 error and type 2 error. Alpha and beta also represented by that. Am I clear? Because if you say somebody which is not effective, but you said effective, that is your type 1. But you fail to show its efficacy, that is type 2. You can do that. But if you say that if he is not guilty, but if you say guilty, that is your type 1 error. That is alpha. So all these things come in, basically. Assessment of outcome, again, very important. You need to define your outcome so well that there is no misclassification. If it shows important, but if you fail to detect it, then your whole study becomes less important. Another thing that I would like to show, what is these things come in. 3 things I will talk about: superiority trial, equivalence, as well as non-inferior. Non-inferiority is a subset of superiority trial. When we do superiority trial, my intervention, my baby, A is, I know it is superior. If you look at this one, 0 is no effect. My result will, if you look at on the right hand side, my result will fall in the right hand side. I know. So this is, my design will be such that it is on the right hand side. If you look at this one, if it crosses the blue, the line of 0, then it is superiority not shown. So if it is near to the superiority margin, superiority just shown. If it is on the other side, it is away from the null, superiority shown strongly. This is your superiority design. Sometimes I prefer doing superiority trial because of background information. But nowadays, most of the researchers do non-inferiority. What is that? I am coming to that. This is your equivalence trial. What is that? If you look at the 0, equivalence is the equivalence margin. This is the equivalence margin. This is your null effect, and our result should say treatment A and treatment B are equivalent. It is not worse. Treatment A is not worse than treatment B, so and they are almost equivalent. Within say equivalence margin, we usually say 10%. Conventionally, we say it is 10%. Now this is another thing, very interesting thing. This is a non-inferiority. Non-inferiority. We take a non-inferior margin, that my treatment is not inferior by say 10%, by 5%, by 15%, by 20% to the existing one. Say cilindripine is not inferior to amlodipine by say 5%. If it has efficacy of 60%, cilindripine will have an effect of 55% at least. If it is there, then my study is successful. So this is here it is your minus delta. This is your non-inferiority margin. All my results should fall within this region. So did you understand? Is it clear to you? Did you understand that this is the difference between the superior bioequivalence and bioequivalence? Mostly we do in case of kits or diagnostics. Superiority is drugs and non-inferiority is now most of the people are doing non-inferiority because compared to the standard of care. Standard of care is available. So now another thing I would like to talk about, that registration of trials. In India, all the trials are to be, if you want to publish it, it has to be prospectively registered in CTRI, Clinical Trial Registration of India. Previously, when CTRI was not there, we used to register in clinicaltrial.gov, but now CTRI is there, it is mandatory. You have to register under CTRI. And another one is how when we write the manuscript, there is a typical format of presenting your data, initial data, that is called consolidated statement of, this is called consort. Yeah, I don't know whether you can see it or not. See this is a typical consort that we usually follow. This is 2025. For observational study, Dr. Panda and Dr. Manish talked about stroke. This is the consort for the RCT. This is uniform. This we have to mention everywhere. So this is the consort checklist. It has from identification as a randomized trial to type B. This is a huge checklist it is. And this is the example. So I'm just showing that how it was done. And these are the phases that you all know. I'm not going into that because you are all very good researchers. Phase 1, phase 2, phase 3, phase 4. So in phase 1 in country there are 4 sites. ICMR is established. There are 4 intent network. There are 4 trial sites that they are doing phase 1. Preclinical in outside the country, chimp model is there. I mean, controlled human volunteers are there. But in India, chimp model is not yet started yet. And there are preclinical. I'm not going to the clinical studies. Phase 1, phase 2, phase 3. Phase 1, there are 4 sites. And there are other phase 2 and phase 3 networks are there. And you are part of, you are all doing phase 4 trials up to. Now this one, this is my favorite slide. I used to talk about it. What I was talking about. The most important thing is that from here, all the researchers as well as all the statisticians can say, I mean, most dreaded is type 1 error. If we should not say somebody guilty if he is not guilty. That is innocent, but we said he is guilty. Then that is called type 1 error. If he is a guilty, but we say innocent, that is your type 2 error. This is major thing. And if you look at your area of probability, that normal distribution curve, this alpha at 0.005 means your 95% confidence interval or alpha, and if it is one-sided. If it is two-sided, then it is 0.025 on both the sides, is as extreme as that. But if it is one-sided, 0.05 is the convention. Yesterday, first lecture was on that. So, and non-clinical studies are that in vivo, laboratory animals, pharmacokinetics, toxicology study, efficacy of the intervention. So with this, I end, and I'm not going to and there are giving an example, cluster randomized trial, how we do it. So it was to assess the protective impact of VI polysaccharide vaccine when administered under realistic public health condition in a cluster randomized effectiveness trial. This one we did in 2004. So how we did it? The whole area was divided into 80 clusters. 40 clusters received the intervention, 40 clusters received the control arm. So vaccines were coded at that time. So this was the analytical strategy. How I want to show you the indirect is a herd protection or indirect protection, how we do it in cluster randomized trial. So if you look at the vaccine trial, total vaccine protection is the recipient in the vaccine cluster. There may be some that out of, say if it is 500 people, if this is a cluster, out of that, say maybe 200 people are there, 150 people have received the intervention, 50 refused. It may so happen. Here also 50 refused, rest received the control vaccine. It means these are the 4 possibilities. A, B, here C, D, this population. Now look at that. Total vaccine, total protection is comparison between A and C. Indirect is B and D. Overall protection is A plus B versus C plus D. That is how simple. Actually, it was a little bit complicated. I made it simple. So, so this one, there are two things. Those who are randomized, but not vaccinated, those were or not drugged, no intervention received, those were left out. That is called per protocol. But if we take them as if they were randomized and took the intervention, we call it intention to treat. So that was the intention to treat versus per protocol analytical approach. Intention to treat, per protocol, and as treated. We prefer intention to treat because that is the public health setting. That is how we do it in public health setting, that some people will be in a big trial, some people will be receiving, some will refuse, some will not come, some will lost to follow up. So all this pragmatic approach, we use per protocol analysis. So this one is important, that is indirect protection. You can see this is a new thing that I talked about. And complete follow-up of patients is very important, and recruitment and retention policies should be there, and regular follow-up is absolutely mandatory. So what with that I'm ending. One thing that is for an RCT, two things are required: the intervention, control and outcome well delineated, and the implementation, so that there is much less follow-up, loss to follow-up, and if there is even the loss to follow-up, it is non-differential. If it is differential, then your whole result becomes desired. I mean, sometimes it will go towards the null, sometimes it will go away from the null. So very important. When yesterday Dr. Bahal talked about, he talked about the implementation. We researchers sometimes forget about how to implement it, how to adhere to the protocol, how to have not too lost to follow up. But we are more concerned about publication, we are more concerned about study design, we are more concerned about sample size estimation, blinding and all that. But follow up and catching the endpoints is also very important. Thank you very much. Is there any question? No.
Is there any questions from the participants? Yes.
Sir, you are talking about the RCT generally applying into the medical perspective. So my question is like, can we apply RCT models in beyond the clinical perspective, like in the controlling, understanding the behavioral or the cultural determinants of health? So if we can apply such model in such way, so is there any way or is there any precaution that we have to take off? So this is.
See, when you can randomize, RCT is the, I showed you the hierarchical pyramid. It is the most highest level of evidence generation, but sometimes randomization is difficult and unethical. Let me give you an example. You know that cigarette smoking can cause COPD or lung cancer. Can you randomize? You'll ask somebody to smoke and somebody not to, and you will see, follow them up for 10 years. You'll give cigarette to him, and you cannot. Say, you know, parachute saves life, right? So you will ask 5 pilots to wear parachutes and jump, and 5 pilots not to wear parachute and jump. You cannot do that, right? Sometimes in some societal context, RCT cannot be done. But in most of the where it is feasible, ethical, it can be done, doable. So these are a typical examples: smoking, parachute jumping. But I mean, there are some areas where RCT is not required basically.
your question, right? That was your question.
Thank you so much, sir, for your presentation on the nuances of randomized clinical trials. Based on your vast experience of clinical trials research, we would like to hand over a small token of a gift to our director, sir. To do the honor, I would like to request the organizing secretary, Dr. P.K. Bora, sir, to kindly hand over the memento to our director, sir. I request the director, sir, to kindly come forward. Thank you so much, sir. As we all know, we are running behind our schedule. That's why we will not have a separate tea break. I know it is taxing and it is tiresome. For almost 2 whole days we are sitting here, session after sessions. Just bear with us. We have a couple of sessions and then we will end the workshop. So to lighten our mood, I'd like to just throw a couple of science humor. So I just want to ask one question. Why the physics and biology break up? Anyone knows why physics and biology break up? Because there is no chemistry. Okay. So I know we have some microbiologists among us. Microbiologists, can you please raise your hands? Okay. We have some good microbiologists. You know, microbiologists love little things. Why? You know the reason? Anybody can tell the reason? They love little things. The reason is they are microbiologists. Thank you. So, yeah. So the next resource person whom I will call is not a microbiologist. In fact, he is an entomologist. So he may not love little things, but he may love insects. So to deliver a lecture on challenges in biomedical research in remote field settings like Northeast India, I would request Dr. S.A. Khan, sir, of Scientist G of ICMR RMRC, Dibrugarh, to kindly take the stage.
Thank you, How. Respected Director, sir, respected Subarna Roy, sir, respected Lakshmi Narayan ji, and speakers in various sessions since yesterday, my dear friends and colleagues. So now, with the cup of tea that you are enjoying, let us enjoy some of our experiences. So I would just be speaking about the uniqueness of Northeast and how things are different from so-called mainland India to Northeast India. We differentiate ourselves as mainland India and Northeast India. Mainland India has 25 plus states. We have 8 states. And peculiarity is that in the mainland India, if we just go with the language, there may be 13 to 15 languages the whole of the country. Whereas we have all the 8 states, 8 unique languages and sub-languages within those which run to around 300 dialects. That's the beauty of our country. Next start. Okay. So Northeast India falls under the Eastern Himalayan biodiversity. And we are the richest in biodiversity also, because 7% of the global species and high zoonotic diseases risk, including vector diseases, vector-borne diseases, are in Northeast Indian states. Apart from its rich flora and fauna, wealth of potential medicinal plants provide immense opportunities for drug discovery and ethnobotanical research, which is of special interest to our Subarna Roy sir. The region's relative isolation has preserved unique genetic traits and traditional health practices, making it ideal for epidemiological studies. It is home to over 200 ethnic groups with distinct genetic backgrounds, and therefore, Lakshmi Narayan ji will be knowing, any tribal health research has to include a Northeast component for population genetics and disease susceptibility studies. So these are some of the pictures which we shot. And of my 36 years of research career, most of the time, my time was spent in the field. Nowadays, most of us, and it is a trend rather, we do molecular biology, bioinformatics, biotechnology, but we were all field studies, okay? And from the field only, being an entomologist, we used to do whole night studies, whole night collecting mosquitoes. And as How was telling about small creatures, my darling mosquitoes are also very small creatures. And even smaller than that, what we are studying now, rickettsial diseases, ticks, mites, fleas, which are even smaller than mosquitoes. So each state boasts distinct traditions, from Assam's Bihu festival, which we are all familiar about, celebrating agricultural cycles, to Nagaland's Hornbill festival, showing distinct Naga tribal dances. The vibrant cultures influence community participation in research, requiring culturally tailored engagement strategies to build trust and ensure ethical collaboration. Northeast India hosts unique ecosystems, such as the one-horned rhinoceros, famous in Kaziranga and Manas Reserve Forest of Assam, to Arunachal's cloud forest, home to rare red pandas, unique orchids, and Tripura's bamboo groves supporting 19 bamboo species, and even the flute of the great flute player Hariprasad Chaurasia, the bamboo was used to go from Tripura. The region's history, from Manipur's ancient kingdom, 33 Sarkaira, Assam's Ahom kingdom, to Sikkim's Buddhist heritage under the Chogyal dynasty, fosters strong community identities. And what we have this Ranghar, as How was telling, 250 years old. This was by the Ahom kingdom. Historical narratives influence attitudes towards external research, requiring researchers to navigate local pride and sensitivities to foster cooperation. The food habits of the 8 northeastern states of India are very diverse, so peculiar. In Nagaland, you will have, in the same road, if you travel from Dimapur to Kohima, you will find 8 restaurants, all serving specific delicacies of 8 different tribes of Nagaland. So much is the diversity. The food habits are very diverse and different, mainly steamed, simple food, without the addition of spices. But in recent years, the spices have made their way in the diet of local people. Once we were in Manipur for quite a long time, and they use a lot of chili. So we asked something with boiled type. So they said in 5 minutes we will prepare it. And we had 3 big cabbages boiled and cut into slices. So that is boiled means pure boiled. So these are some unique features state-wise. Arunachal Pradesh, 600 plus orchid species, and we have the only orchid research center in Arunachal Pradesh. That is at an altitude of about, I think, 8,000 feet above mean sea level. Then Assam, one-horned rhinoceros, Manipur, the Manipuri dance, the Loktak lake, there is a floating lake, many of us might have seen, and Meghalaya, living root bridges, Mizoram, 300 plus medicinal plants, Nagaland Hornbill Festival, Sikkim, cordyceps plants, 100% organic harvest. That is the only state with 100% of organic products. Tripura, Manikya dynasty, as the famous singers from so if we look for the health profile, Northeast India faces a high burden of infectious diseases because being in the equatorial zone, we experience very highly humid and warm climate that what we call supports lots of microbes as well as insects. So Japanese encephalitis is prevalent with more than 500 cases annually in Assam. So Assam being a very small state, it burdens 30% of the country's JE alone, the state of Assam, having a population of 1% or 1.2%. Tuberculosis rates are elevated. For example, 200 per 100,000 in Nagaland versus 150 nationally. Emerging non-communicable diseases such as diabetes with a 5% annual increase and hypertension and complexity for health resources. So these were this non-communicable disease including hypertension and diabetes was not very common here. But this I think is the gift of mobile phones and social media because people's life have become very sedentary. Otherwise, this in villages of Assam, hypertension and diabetes was not heard about. And now mostly people have turned to tea plantations, small tea growers. So most of the land you will find fallow. Nobody is doing paddy cultivation because they get free rice. And so they have all gone to commercial plantations and life has become very sedentary. So this, there is an increase in limited infrastructure hampers research. Only 30% of rural health centers have reliable electricity. This is a very big problem. Poor road network, especially in Arunachal Pradesh, Nagaland, and almost parts in almost all the 8 states, restrict access to remote areas, delaying sample transport and field work. Internet penetration is very low. Only 45%, more than 45% lack access, hindering digital data collection. These barriers significantly impact the feasibility of biomedical research in this region. Biomedical research in remote areas face unique logistic, infrastructural, and cultural barriers. The 8 northern states present distinct challenges due to the geographic limited resources and diverse populations. Each of the 8 northeastern states of India is home to its own unique language, along with a remarkable number of dialects and indigenous tongues, reflecting deep cultural and ethnic diversity. Arunachal Pradesh, as I already told, alone has around 50 distinct dialects. And interestingly, these dialects are not very common. Even a person speaking a dialect would not be knowing the neighboring person's dialect. So peculiar, that means they have not emerged from each other, but they have individual sources of emergence. With specific tribal, complicating further the problems of researchers in communication and community engagement. Suppose we go to field, we hire somebody as a interlocutor. So that person won't be able to help me for the other tribe. And therefore, when we go to field, and so many research problems also come up. Like we have to pay something in the form of honorarium to that person. But we are asked to give some GPay kar do usko. We have to have some proof. But that person does not know what is GPay. Most of them don't even have Aadhaar. So what to do? So somehow we have to convince our account section or the administrative people that see we went to such area where we had to work for 4 hours, work for 3 hours. So we cannot generalize that we have the same bills or money receipts or e-payment. So these are some of the major issues which we face as researchers in the field. This linguistic diversity, while a source of cultural richness, poses significant challenges for biomedical research, from participant recruitment to data collection, dissemination of findings. Overcoming these barriers is essential for advancing healthcare, ensuring equitable research participation, and improving outcomes in underserved areas. So these again are some of the, see how sparsely the population is. So suppose you have to survey 100 houses. Just imagine How much walking that would be? That would be a 10 days walking exercise for you in a single day. So we can see only maybe 20 houses in this stretch. So when we are doing, as sir was doing, Chris method of survey, so you just imagine how you survey this 100 houses or 50 houses. So these are very common landslides, caving in of the land, because the hills here are soft soil. Very rarely you will find stones. Therefore, they cave in. And these are very, northeast is very much earthquake prone also. Overcoming these barriers is essential for advancing healthcare, ensuring equitable research participation. Okay. Now, scope. Biomedical research in remote settings involves community-based surveillance to monitor disease trends, interventional studies to evaluate health programs, environmental sampling to assess pathogen reservoirs, and bioinformatics analysis to model disease dynamics. Now, talking about the challenges. Training shortages limit skilled personnel for advanced research techniques. Cultural sensitivities require tailored engagement to respect tribal customs. Suppose getting access to a village in Nagaland might be very much different than getting access to a village in Mizoram. So tribal heads are there. So formalities, the hierarchies we have to take care of. Otherwise, you are in great trouble. Therefore, experience matters, how to meet the people, whom to meet first. Otherwise, you are gone. Therefore, and when we are moving, suppose you are moving to Nagaland, suppose you are moving to Manipur, going through Nagaland, you have to be very careful whether there is a bandh in Nagaland. Otherwise, in many occasions, the local group will come, why are you moving? We say, no, we are going to Manipur. But didn't you see the morning newspaper? We have called a bandh. No, sir, we don't know your language. Still, you should have seen that. But then somehow you have to pursue, sir, we are going for your people only. We are in malaria research, or we are going to this, that. But then the same persons who are so arrogant will become so helpful that they will say, okay, next 10 checkposts we have telephoned. We have given them their vehicle number. You will face no problem. Just keep going. So these are the peculiarities of Northeast, where they get convinced easily, but at the very first sight, you will really feel scared. Research empowers communities by involving locals in health solutions, fostering trust. Trust is very important in any research. So inaccessible areas, many, in 7 of our friends were deputed in 7 states. And Arunachal Pradesh, Papumpare was the district. So I was given the responsibility of Papumpare. We went with our vehicle. So the joint director said that, what vehicle have you bought? We said we have brought a old Mahindra. This modern vehicles were not there. So he said, you want to go there with a Mahindra? Leave it here. And he gave his own gypsy. Only this will go. And then he gave us chura, no? Flat rice and chana. He said, you take this because you will not get any food there. And I am giving you driver and 5 jerrycans of petrol. So this is how they help you, when they know that your intentions are clear and your purpose is fair. Okay? So that is how we conducted the vaccine survey. And otherwise, they said, 8 din paidal jabi, 5 din paidal jabi, means that distance is 5 days of walking. And there will be porters for you who will carry your luggage and you will be walking with them. Just imagine where you will sleep, what will happen. So, but now, these are things of the past. Roads have developed, most of the areas, but still population in areas is very sparse. There are some areas where polling stations are set for 1 voter in Arunachal Pradesh. So peculiarity of this Northeast, I would say that you go tour the whole world, if you have not seen Northeast, you have not seen many things. So maintaining cold chains is very difficult here. Blood, tissue, where to keep? Because we don't have dry ice. Only one is, plant in Guwahati that produces dry ice, and that twice a week. Tuesday and Thursday. So you have to stand in queue, give them indent 1 week early that please give me 5 kg of dry ice. But the dry ice, once you get to Dibrugarh, it is half. Again, if you send from here to Chennai or Kolkata, it is gone. Therefore, you have to procure, I think, 5 times or 10 times of the dry ice that you need. So these are some serious bottlenecks. And Northeast, very humid climate, risking sample degradation. Lack of prior reservation, as I have already told. So these are very common. Flood season, there will be temporary nala created, temporary water, so you have to use the local boats to cross. This is a wooden bridge. Obviously, your vehicle won't go, so you have to leave the vehicle on the other side and walk the rest of the portion from where you are destined to go. So this is mitti. So no vehicle will drive here. So recently we conducted one leptospira study in Karbi Anglong, sorry, Dima Hasao. So the railway station was there. That was a newly built railway station. We did the study, came back, and on the third day we came to know that the railway station has been washed away by flood water. No railway station. So these are the vagaries of nature in various parts of this region. Lack of advanced diagnostic tools such as PCR machines, trained personnel, limits on-site analysis, thus relying on distant urban labs. Insurgency in some areas threatens researcher safety, requiring armed escorts. We used to go with armed escorts in Tripura. Now these are things of the past. Now the situation has improved in many places, but very interesting. And that also armed escort would not go every day. Suppose we reach there on Monday and we come to know that the armed escort will go on Wednesday. So you be there waiting 2 days, and then the armed escort would go with a big convoy. All the vehicles which have accumulated, they will line up in front of the armed envoy, and you will go in a caravan. And so these are very interesting experiences. Access to next-gen sequencing, very rare. We have only access for 2%, whereas the national average is 15% people have access to next-gen sequencing. Bioinformatics tools are underutilized due to hardware limitations, hindering genomic and proteomic studies, critical for personalized medicine. Linguistic diversity, I have already spoken about. Now, what are the strategies to address these challenges? Portable diagnostic kits like CRISPR-based kits enable these far-flung northeastern states. Drone-based sample transport have been shown to reduce delivery time by 70%. And ICMR successfully conducted this COVID testing in areas of Manipur. And again, there are projects where ICMR is planning to deliver drone-based what we call anti-venom or similarly vaccines which need to be administered urgently, or medicines which need to be delivered urgently. So although most of the areas are now accessible, but intermittently due to landslides or something, the roads when they are blocked for 10 days, 15 days, 20 days, so drones will be of help during those times of emergency. AI-driven analysis and telemedicine platforms enhance data processing and remote consultations despite connectivity gaps. All-weather roads, as I have already told, improve access to 60% of rural sites, facilitating equipment transport. Solar-powered health centers. For example, introduction of solar-powered boats in Assam. This is a solar-powered boat in Assam. Ensure stable power for sample storage. Local lab upgrades and modular sequencing training programs boost regional research capacity. And I think in the whole of Northeast, power condition also have improved a lot after the commissioning of many of the hydroelectric power projects of Northeast. So increased ICMR, DBT, and other funding agencies, funding supports regional specific research. Public-private partnerships with startups deliver low-cost diagnostics, and NGOs are very they are playing a very good role, like the churches in Mizoram, in Nagaland, and various other organizations in different states of Arunachal, in different areas of Arunachal Pradesh. They are doing wonderful job, even NGOs. Extend research reach to remote areas, enhancing data collection and community health outcomes. Partnering with local leaders, community health workers, trained in data collection, culturally tailored materials in language like Bodo. So I will come back to this, some of the, oh, it's playing, okay. So these are some of the pictures. So we were on a mosquito collection drive, and suddenly there was a storm. We stayed back. This same road we went, now we are returning. So just see, all trees fallen, and our young guys, so they will clear the road, and then we are on the road again. So this is a bridge, the ancient Howrah Bridge. Huh? So these are our boys going for mosquito collection. So this is a half-built bridge, but no access on both the sides. So just imagine how we are accessing that. Bridge is ready, but connection on both the sides is not there. So these are quite common because contractors do half of the work and then they elope, then there will be another contract to finish the side roads. Very risky situations, but at the same time, we enjoy doing it because we don't have options. No, so we have to go to the other side. We are thinking how to cross it because the current is very swift. So one of the persons takes the challenge and he goes, we know the depth of the water also, so others will follow. Now what are the future directions? Develop eco-epidemiological models tailored to Northeast unique biodiversity and tribal health profiles, integrating environmental and genetic data to address diseases. So everything in Northeast has to be unique. Like as in our ICMR campus, we have some buildings which were made on Delhi model. All the buildings are flopped now. We don't have ventilators, which is a must here. We have roofs leaking, as our boss sitting here faces all the headache. Now what to do? Put another layer of what you call concrete, still leaking. Now what to do? Put some CI sheet. So experimentation goes on. That's why even the Britishers had Assam type houses. They were intelligent enough, smart enough. They said, no, all India things will not work here. So still people using Assam type houses here. And although it was Assam type houses, but they built in whole of Northeast because that nothing happens to it in earthquake, in high rainfall, even in high wind. They are adapted to all these vagaries of nature in Northeast. Participatory frameworks with local stakeholders ensure research aligns with regional needs. Train local researchers and aim to create mentorship networks by building sustainable research capacity. Leverage machine learning for predictive epidemiology, analyzing disease outbreaks in real time. So to conclude, Northeast India's unique challenges require innovative and tailor-made situations. Tailor-made not for the Northeast, but for individual states. The region's rugged terrain, ethnic diversity, and history of insurgency present complex obstacles that standard development models cannot address effectively. Poor infrastructure, frequent natural disasters, limited connectivity, and deep-rooted social economic dynamics hinder healthcare delivery research and economic integration. Addressing geographical isolation and logistic gaps with portable tech and socio-cultural complexities through inclusive engagement is essential for effective research delivery and community trust. Increase investment, foster collaborations, and prioritize ethical research. Sustainable progress demands greater investment in infrastructure and human capital, partnership between local communities, researchers, and policymakers, and a strong focus on ethical community-driven research practices. A region-specific inclusive approach backed by robust investment and collaboration will unlock Northeast India's full potential in biomedical research and public health while respecting its unique identity and addressing its present challenges. Thank you for your patient hearing.
Any quick question or any comments from the participants?
So if there is no question, I would like to have one question, sir. So firstly, it's an honor, sir, to ask my teacher the question. So being a preliminary researcher and now, and you being the having 36 years of experience in conducting research in Northeast, how do you see the future where we have this AI-driven advancements and we also have drone-driven to address this communication gaps and all? How do you see that pursuing a research in Northeast for budding researchers like us would have a promising future ahead?
Well, Pratik, see, these are some of the exceptions. Not that everywhere drone will go. So there is no alternate to building good roads. We cannot say that, okay, we will be delivering by drone, we will not invest in roads. So these are some of the exceptions which we have to take care. And artificial intelligence, what you said, this is universal. So like Northeast is developing, and we have seen it develop in the last 30 years. Okay? And I hope that it comes at par with the mainland India. Because now even earlier people from Northeast also would not go out. Now you will find one person going out for education or working, 1 in 2 families at least. So things are changing, and now people are going and having communication, and outside people also are coming. Earlier, like people from Northeast, they would not recognize. They would say, oh, ye China se hai. Because our Northeast people's expressions are like that, because we belong to the Mongoloid trade. Therefore, there are lots of unrest even in Delhi or other areas from Northeast, going and not getting that due respect. But things are changing now. Many of our children are there in different southern states, western states, or, and even from other places. You will find here in this group many of non-Northeast people engaged and working in Northeast. So that is the change. Perception is changing about Northeast. And I am a very positive believer that things will change very fast in the next decade. Thank you. Thank you.
Stay back. Thank you so much, sir, for your presentation and highlighting the real issues and field challenges in Northeast India. So we would like to give a small gift as a token of our love and gratitude to Khan, sir. So to do the honor, as director has passed on the baton to Dr. R. Laxminarayan, sir. So I would like to request sir to kindly hand over the small token of gift to Siraj, sir.
I am happy to do this because we work together in several fields, including Maharashtra and Northeast.
We have surveyed caves.
Because you may think otherwise what an administrator will do in the field. My duty is an advance party. I go as an advance party, I talk to the collector, I talk to the district forest officers, I talk to the police SPs, and then make all arrangements, and then you come. Then you will not believe, there will be about 25-30 people from Pune. I was in National Institute of Virology before coming to headquarters. So 20-25 people will be there. 2 vehicles used to come from Pune. For all this 20-20, I used to cook. I used to cook for them because मैं बोलता हूँ भाई मेरे को तो फील्ड में यही आता है.
And sleep in tents at night.
At night, and we slept in, and bat collection was the real eye opener for us. Bat collection studies in Northeast, and what you said, drones. ICMR has first trial tested this drone in Northeast only, and we have done several sorties in Manipur, Mizoram and Nagaland. Still it is going on, and for Northeast now I think he has been nominated as nodal officer for drone.
Thank you so much, sir. Now we move on to the next session. Next session will be taken up by Dr. Biswajit Borkakati, sir. And the one session from yesterday, which Sangamitra ma'am was supposed to take on writing a good proposal, he will also partly covered in this session. So writing good proposal as well as report writing will be covered by Dr. Biswajit Borkakati, sir. Sir, please take your time.
Thank you, Hal. So I'm the last batsman, and we had stalwarts of like Sunil Gavaskar, Sachin Tendulkar, and Virat Kohli's batted. So this is a slog over. So maybe I am the Satan Sharma of the 80s. Thank you, sir. So before going into this topic, so I'd like to say that ICMR has invested a lot in Northeast India. They wanted a lot of projects from this area, from all the 8 ANI states. So that is why we have called researchers from every institute, from not every state, actually from every state we have called people so that we have some good research coming up. So one of the things that we need is a good proposal. So good proposal will come. What are the things? Actually, we have a lot of funding for Northeast India, and these salient points I'll take as a reviewer of ICMR for last couple of years, reviewer of projects. I'll tell you some points, specific points for research project writing. And before that, I'd like to come to a part where we have discussed 2 important things. One is the study design. What I have observed is that in the review, the proposals that we get is sample size. Every researchers, if they are very experienced also, they lack, or they will be putting the sample size not properly. And another thing is that they will not properly write about the study design. So these 2 mistakes, at least 50 to 60% of the researchers do. Their idea may be good, they have a very good rationale, but the methodology may be inappropriate. So before that, I'd like to sum up. I have around 11 questions regarding study design. So it is a quiz. I'll take 10 minutes and you can answer. These are questions and you will be the fastest one to answer will get one small gift from my side. So can we begin? Everyone ready? So first question, it's an MCQ question. A researcher collects data on HPV infection status among 3,000 women in 8 northeastern states in a single survey. What is the study type? Is it cross-sectional study, case control study, cohort study, experimental study? Anyone? Is it? Huh? Cross-sectional. Please give. Thank you. It's from Assam. Okay, Assam team, Assam Medical College. Second question, comparing HLA class 2 polymorphism between ZAE encephalitis patients and asymptomatic ZAE IgM positive individual. Is it a cohort study, cross-sectional study, case control study, randomized control trial? Yes, he's right. Please give the gift to the front. So you are from?
Manipur University.
Manipur University. Thank you. So this is investigating whether a new herbal drug inhibits growth of MCF7 breast cancer cells compared to untreated cells in vitro. Is it a case control study, cross-sectional, experimental, or cohort?
Experimental.
Yes, from Arunachal Dreams. She was first. Please give her the gift. Next question, surveying parents in Mizoram about vaccine hesitancy and correlating with actual immunization records. So what is it? Case control, experimental, mixed methods, cross-sectional? Yes, this is a mixed method. Who said? From Nagrims, please. Great. So following a group of tribal children in Ani, India for 3 years to observe incidents of malaria. So is it a prospective? Yes, Madam from Guwahati University. Madam from Guwahati University in the front, yes. So a hospital reviews the medical records of 500 leptospirosis patients to identify factors associated with mortality. What type?
Retrospective.
Yes, who said? Who was? Okay, great. Sikkim, Manipal. Testing a new anti-malarial drug versus chloroquine in patients randomly assigned to each group. What type? Yes, everyone said, but yes, but I think so. They also said from which? Nehu. Okay, thank you. Great. Evaluating the effect of an IEC campaign on hand washing before and after its implementation in school. Quasi-experimental, case control, cross-sectional, or RCT? Yes, from the Nagrin teams, they got it.
Quasi-experimental.
They were first, the Nagrin team. Surshanpur. Okay. Surshanpur from Manipur. Assessing the association between vitamin D receptor, gene polymorphism, and tuberculosis by comparing TB patients and healthy controls. What type of study?
Case control.
Yes, case control. So that was an easy one. Please, Parismita in the front. From where? Okay, they got it. Again from Sikkim. Conducting gene expression profiling on CSF samples from AES patients to discover new biomarkers. Discovery study. Again, Assam Medical College. Great. This is the last question. A study on iron folic acid supplementation in adolescent girls in Meghalaya, weekly IFA tablets distribution in schools, how many schools constantly deliver tablets? Are girls actually consuming them? What type of study?
Implementation.
Yes, that's great implementation study. So we have come to the end of the quiz round, and I think the 2 days methodology, research methodology is a success, because these are the confusing things, which type of study we have to, and you have been able to answer all those within 10 seconds correctly. Thank you all. So, coming to one of the important part, I wanted to stress ICMR projects, actually, how we have to write. There are some marks associated. Which are the marks we will get? Because out of 100, there are some marks at every point. What is the rationale? What is the problem statement? What is the novelty? What is the methods? How is your implementation strategy? Every part has got some marks, and you have to concentrate where you are putting your effort. And before doing any study, we have to know what is the problem. Is it a national problem in Northeast India, or is it specific to any specific state? So when we get an advertisement of any that project proposals are being accepted by ICMR, DSR, or DBT, or DST, there are some important parts in it that we need from cancer, we need for cardiovascular disease, we need for tuberculosis, we need for malaria. Then your study design has to be accordingly, and you have to find the gaps. First, before developing, everyone, actually for last 2 days, this part has been discussed many times. So, what is not known? What is not known is very important. First of all, are we going to reduce the mortality of a disease, or are we going to reduce or anything, some public health problem, or we are going to benefit the society? As a health researcher, we are working for the community. We want to improve the health. And these questions are very pertinent. One thing is that our small research, will it benefit the whole society, the whole country, the whole world? Is it novel, or there is some gaps? So these questions has to come in mind before we develop any project. So what is the problem in Northeast India? We have to know. So generating the hypothesis or the gap in knowledge or the research question is the first thing you have to have in mind. So let's go back forward. Is it doable? Is it feasibility? You have a lab that doesn't have, suppose you wanted to do a genomic study, and you do not have transcriptomics facility, you do not have NGS facility, then you have to select which are the labs that have that facility, the bioinformatic data, then you collaborate with those institute. So you have to see the feasibility and the doability. So again, I'm just giving you an example, just because examples are the better way to define. We have a question out here. ZAIS, we know prior knowledge is that ZAIS is endemic in Assam and neighboring states. Most studies focus on clinical outcomes and vector control, but some individuals remain asymptomatic. It's not some. Actually, in ZAI encephalitis, 1 in 300 has encephalitis, rest 299 does not have encephalitis. Majority of the cases in ZAI are asymptomatic. So why out of 1 in 300 develops the disease? Is it host related? Your question may be, is it host related? Is it host related maybe? Is it HLA dependent? We know HLA type differs from person to person. So is it host related, or your hypothesis then will be certain HLA class 2 polymorphism are associated with asymptomatic ZA infection among individuals in endemic regions. That will be your null hypothesis. And you have to disprove this. That means if you disprove this hypothesis, then your alternate hypothesis will be that certain HLA types polymorphism will cause susceptible to encephalitis. So that is your alternate hypothesis. This you may design, suppose the class 2, and you have to see what will be the benefit. If I know the HLA class, that suppose HLA-B something is associated with ZA encephalitis, will it benefit the patient? Will it benefit the society? Or it will be just like a data? But we have to know in future we will have individual treatment or probability that this person is, then we can target those person for vaccination, this type of. But there are a lot of things in HLA. Suppose I'm telling you about, there are a lot of diseases that are associated with HLA. Maybe one example I can give, carbamazepine sensitivity. Some person like HLA B15 or 02, they may develop Stevens-Johnson syndrome. So you know from the medical personnel, they will know what is Stevens-Johnson syndrome. It's like a skin drug reaction, like TAN, toxic epidermal necrolysis that we have from Staphylococcus aureus. So there will be that skin manifestation everywhere, and that can be sometimes fatal also, Stevens-Johnson syndrome. But once, before giving carbamazepine, carbamazepine is an anti-epileptic drug that is given, and before giving carbamazepine, you do a HLA typing. And if it is HLA-B15-02, you don't give carbamazepine, you give another drug. So this type of thing, that if ZAE is associated with some HLA type, that person has to be vaccinated or something like that. And you have to see, because all vaccine will not work. All vaccine will not work. So you have to sense if some HLA types, because what are HLA? HLA molecule will capture some your immunoepitopes, and they will present it for neutralizing development of your cells. So the vaccine has to be modified. So ultimately your research will be beneficial to those that are developing vaccine, better vaccine for the population. So that is one research. I'm just giving you an example how we can think, because sometimes you will think, yeah, this is not important, this will not benefit the patient. But you have to see a broader picture. As I've told you, what is the importance of a study? Is it national, is it international, is it regional? Sometimes some diseases are regional and local disease. Suppose in Dibrugarh district we are getting a lot of cirrhosis cases. And these are non-alcoholic cirrhosis cases, okay? They don't take alcohol. We are thinking this is NFLD due to diet, but it may not be. There are a lot of periportal fibrosis in those cases. Then we take some antibodies and we see schistosomiasis antibodies is high, and we can link it. Maybe locally schistosomiasis prevalence is high out here, and so people are getting cirrhosis of the liver. So this will be a local problem. That like that paragnomiasis. Here we have hemoptysis, and it will mimic tuberculosis. So if we don't rule out, and you are giving anti-tubercular drugs that have got very high adverse reaction, 10 to 15 or 20% of adverse reaction, liver toxicity, and that patient was giving, taking anti-tubercular drugs for months and years, and he's not getting symptoms, hemoptysis still occurring, but he was suffering from paragnomiasis. Based on only the diagnosis of hemoptysis in the sputum, and we did not screen for paragnomiasis, maybe it is not justified. So these are some local problems. So we have to see the problem where it is, the local problem, regional problem, and national problem. Okay, this is the important part that I wanted to discuss about the research projects. Because for ICMR, there is a format, and for DSR also, how you will fill up. Title of the project, and they will give you 25 words. Within 25 words, you have to describe the title. And that title should be very specific, concise, and simple. It should not be filled up with jargons. And it should reflect your objective, what you are going to do. So once you develop your idea, once you develop your project objectives, then you can think about the title. First, you don't think about the title first. You think about the hypothesis, your problem, your aim. Then you think, because title is one of the hardest part. A good title is one that will attract, but it should reflect what you are trying to do in your project. So the title is one of the most important part where the reviewer will first see what he is trying to do, or what he or she is trying to do, and it should reflect properly. Another thing is that summary. Summary is one important thing that gives the reviewer an idea, because sometimes I got 40 projects to review within 5 days. I'll just see how the summary is written. Each and every step, it may not be difficult, because if the summary is not written properly, then we will look at the methods. If those two are not proper, then you will get a low mark. You will get a low mark. So sometimes reviewers don't see the whole project. They will see the summary, they will go to the methods. And if your methods and your summary are not good, you may score a low mark. You may write rationally, your hypothesis may be good, your problem statement may be good, your outcome may be good, but your methods and your summary part is not good, you may not qualify. And because why I'm telling you is that there is a huge competition. This year, ICMR got more than 6,000 projects for small grant. Small grant is up to 2 crores. Okay? So from this, hardly less than 5% are selected. Less than 5% out of 6,000. So you have to compete among all the person from all over India. And nowadays what happens? Lot of private companies, they will compete with you. So writing a project is one of the most important thing, how you can get the project. So these are available. As earlier I said, our previous presenters, they have already discussed descriptive, discovery, development, and delivery. These 4 Ds are very important. What project? Is it descriptive? ICMR generally does not propagate descriptive, because for last 50 years, we have been doing descriptive studies only. But it's not that ICMR will not accept descriptive studies. It has to have some significance, importance. But if you have discovery, development, and delivery, you have a greater chance of acceptance. Greater chance of acceptance. So you have to be careful what type of study you are trying to do. Okay? Again, priority areas, non-communicable disease and communicable disease, because ICMR will have this, or DBT will have that we need studies on cardiovascular disease, we need on stroke, we need on chronic renal disease, we need on hemoglobinopathies. Then those problems will come accordingly to Northeast India, some specific problems, and then you have to concentrate on those areas only. This part, problem statement and rationale of the study. So this, I'll tell you, carries 20 marks each. 20 marks. 40 marks you will get from here. in your project, if you submit it to ICMR, sometimes if your summary methodology is poor, you may not get marks out here. Okay, so you have to remember problem statement. I'm telling you an example. Okay, TB elimination is one area that this present government is targeting by 2030. So you are going to implement something else. Suppose we all know that latent TB is around 22% of the whole population of India. That will come to 300 million cases. Is it possible to eliminate all those cases? Within next 5 years, this 300 million, that is the population of United States of America. Is it possible within 5 years to treat all those? It's near to impossible. Until and unless we have something else. So you may suggest how to detect latent TB. You may say that IGRA is there, but IGRA cannot be applied in the field, interferon gamma assay, release assay. Then you wanted to develop another assay that can be used in the field. That can be used in the field, you can implement and see if a rapid like to detect latent TB. And you can do a trial out there so that those things are there. Suppose we are using anti-tubercular drugs and 20% people are having adverse and they don't want to take the drug and they will miss it. What happens then? Which next drugs you are going to give? If the first line drugs you are having liver toxicity, then they are not treated. Then we have this reserve drug for drug resistance, that is bedaquiline. So bedaquiline has got a long half-life. Okay? So if this drug is given like a depot once a month, because people don't like to take every day. So if a clinical trial is done of once a month dose of bedaquiline with others, so these are some of the studies people can devise from Northeast India. We can give on and on example, but this has to have some impact nationally. So this part has been described, PICO, population, patients, intervention, comparison, control, outcome. So lot of things, your idea of having an hypothesis, and if it falls under the PICO. Does it falls under the PICO? Generally, most of the studies falls under this category, PICO. This is the part, most important part of the research methodology. And one thing is that a lot of the projects get rejected because of the study design. And now our focus of the research methodology was basically study design. Most of the speakers out here talk about the study design. And then another important part was sample size calculation. Sometimes you can say, okay, I have taken a software like EP-Info and calculate the sample size. For proportionate, you use that is Z square P minus Q divided by D square. So that is the formula. But is it applicable to everywhere? Is it applicable to everywhere? No, it is not applicable. Suppose you have a sample size is it applicable to large, maybe a population survey like PPS sampling? So how many of you have done a PPS sampling methodology? Anyone? PPS sampling? Probability to proportional to size, PPS. Has anybody done? Okay, so this is very important. When you do a cluster sampling, randomization of sample, it's very simple. It's not a complex thing, PPS sampling. Suppose for Northeast India, you wanted to survey some diseases and prevalence of diseases in Northeast India. How much sample you have to take? Is it multi-stage? How randomization is there? even for Dibrugarh district, suppose Dibrugarh district has a population of 14 lakhs, according to the survey of maybe 2011 or something like that. And how you are going, suppose there are 700 villages and 24 wards in Dibrugarh district. How you are going to design a study? It's simple. You take the data from the population of all the villages and population of all the wards, and you arrange it in an Excel by ascending order from the lowest to the highest. Okay, so every villages will have maybe 10 number, then it will increase. Then you have to calculate the sample size first based on your proportional study, that is you are taking 4p minus q, pq by d square. Then again one problem comes up. What is the expected prevalence? What is the expected prevalence? If it is between 10 to 90%, you can take 50% prevalence. Why? Anybody, why? If your expected prevalence is between 10 to 90%, why do we take 50%? Anybody? Yes? It will give the highest sample size. 50% prevalence will give you the highest sample size. If you don't know, if you have no idea about the sample size, it will give you a highest sample size at 50% prevalence. Between 10 to 90% prevalence, expected prevalence. Suppose we are getting a small, you are doing a small 384, you will get at 95% confidence interval. Okay? But below 10%, will it give, suppose your prevalence, expected prevalence is 5%. Will it give the same? Will you use the 4pq by d square formula or different formula? So here, can I use this one for a minute? Okay, this will make you clear. Just one example I'm giving you. This is an X and Y curve. This is, I'm just writing a W. This point is 50%. This point is 10% expected prevalence. This is 90%. This is 90%. And this at 95% confidence interval, this is 384. If you are not using a design effect you can take at 1.5 or you can take at 2. Okay? So what you are getting is that at 50%, if you know the prevalence is between 10 to 90%, this is the sample size everybody takes. By this formula, the formula is sample size is equal to 4pq by d square. D is the precision. What is the precision? It's 0.05. Precision is 0.05. If you take the precision out here and if you use this formula, your sample size will go down like this. Go down like this. That will not be accurate. So you have to change this formula to, you have to divide this and square this. Okay? So that is the change in formula, where if you have a prevalence less than 10% or more than 10%. Okay? So coming to the PPS sampling. So PPS sampling, the first thing you have to do is collect the data, the survey data, put it in your Excel, put it in ascending order, all the villages and the words. And then you have to calculate the sample size like this one. And after calculating, you will need to know, suppose your sample size you are taking at 5% and at a precision of 5%, you are getting a sample size of 1200 based on your design effect. Okay, design effect. Now from Dibrugarh district, you have to select 1200 samples. Where do you select? From which villages? From which ward? Then you have to know a random start, RS. What we call is RS. RS, this is called the random start. What is the random start? It's a number between 1 to 1200. Because your sample size was between 1 to 1200, your random start will be between this number, between 1 to 1200. Then you need to calculate another part that is called sample interval, SI, sample interval. So at what sample interval you are going to do the first village and the next village, then the next village. So how to calculate the sample interval? The sample interval is calculated very easily. It is based on how many clusters you are taking divided by your total sample size. So you will get the sample interval. Suppose you have a total population of 14 lakhs, and what will be the sample interval? Okay, so that you can calculate from your total sample size and how many clusters you are taking. Suppose you are taking 60 clusters, and how many patients are there in each cluster. So there is some way that you have to have between 10 to 30 individual, 10 to 30 individual per cluster. Okay. So if you have 10 to 30 individual per cluster, and then you are taking you will be able to calculate your sample interval. Suppose your random start was 700, then again your sampling interval will be maybe because you have to divide how many clusters are there, 60 clusters, and you have to divide it by 60, and at what interval, maybe 5,000, 6,000 samples, you will get one cluster. So you have to take that cluster. That ultimately, your sampling unit is there. The term we use is called PSU. This is called the primary sampling unit. So you have to remember only these three things. So your primary sampling unit is there, your sample interval is there, and your random start is there. With this, you can calculate your PPS sampling. Okay, so anyhow, we do have another actually research method or data analysis we'll be having within next 2 months. So there we will be doing this type of thing to calculate this sample size, PPS, and other data analysis. So here I'll not go further than this now. Can you go back to the slides? So the methodology, what we have, suppose you have got 3 objectives. The 3 objectives may have study design, different study design. So for every methodology, you have to put your study design. Study area, sample size, primary, secondary outcome measures, and design of statistical analysis. This is one of the important part. Nobody gives importance to this part, design of statistical analysis. What type of data? Everyone says that we will take a chi-square value, also association study. Okay. Yesterday it was discussed the difference between odds ratio and relative risks. Then what is the adjusted odds ratio? Anybody? Adjusted odds ratio? Huh? Yes, when we do a regression analysis of the factors or association, we will come, suppose we are doing a binary regression or a multiple regression analysis, we'll come with some odds ratio that are different from your cross-tabulation odds ratio. What is odds ratio? A, B, C, D is there. A into D divided by B into C. That is the odds ratio that we generally calculate. Okay. And this can be adjusted by binary regression or multiple regression. So I'll just have another question that I'll just move forward. If you have one dependent variable, okay, and maybe others are independent variable, what type of design you will have for analysis? Will it be cross-tabulation? Will it be multivariate analysis? Or will it be multiple regression analysis? If you have one dependent variable, are you going to do multivariate analysis? Anyone? So the concept I will make you clear right here. It's very easy. Suppose you are studying rheumatoid arthritis and you are collecting data maybe for rheumatoid factor, maybe a C-reactive protein, maybe other immunoglobulins or biomarkers, then you have got multiple dependent variables. Your dependent variables are multiple, not one. You have C-reactive protein, you have a rheumatoid factor, and you are combining this and you are doing a multiple variate analysis to see the association. And here you can do a multiple variate analysis, not a multiple regression. So there are two types of data. One is the categorical data, another is the continuous data. Variables may be continuous. Suppose blood sugar. Blood sugar is a categorical or continuous data. Suppose for diabetes, you are collecting fasting blood sugar. Okay, is it a categorical data dependent variable or is it a continuous variable? It is a continuous variable, isn't it? Continuous and quantitative data. It is not positive, negative. It's not a positive. Categorical will be either positive or negative, like hepatitis B positive or B negative, or HIV positive or HIV negative. That is categorical. Okay. Continuous data, you have a continuous data. So I think that is clear, but to refresh for some of the confusion. Then another part important is what type of analysis you are going to do. Is your data parametric data or non-parametric data? So anybody, parametric data, difference between parametric? Yes, it is non-parametric and parametric. Normal distribution. If your data has a normal distribution, the bell-shaped curve, it's a parametric. If it is not normally distributed, it is non-parametric. And data you have to take. For continuous variable, you have to analyze like t-test, the student t-test or the paired t-test. Okay? For categorical data, you can use a chi-square, Pearson's chi-square test or a Fisher's t-test. Okay? So there, what test statistical analysis you are doing, it will determine your data. So so design of statistical analysis is one of the very important thing, and you have to give some importance to that part while writing. So another important part is this implementation strategy and milestone chart. Gantt chart, we call it a Gantt chart. And if you have theirs, because this also carries some marks. I'll show you the marks actually. Can you make out? Implement bad. Generally, those proposals that get over 70% mark, they are usually accepted. That may differ, but generally those scoring above 70, those scoring above 70 has a greater chance of acceptance. So you have to remember if you are writing these things properly, because methodology carries your highest mark. So what this is, CERT, this is called the Gantt CERT. Implementation strategy over. Suppose you have a project of 18 months duration and you are if you are not putting this CERT in your project proposal, your 15 marks will go off. Remember that you have to put this CERT. If you don't put, your 15 marks will go. Remember that. And we like to have contingency plan. Suppose your manpower was not there on time, your equipment didn't come, you need to have a contingency plan that you are going to, if you do not have equipment that you are going to do for first few months in a different institute, that will be your contingency plan. You cannot keep, okay, equipment didn't come for first year, I will start in the next year. It is not like that. You have committed to complete the project in 1.5 years, and you are not being able to give a contingency plan. Suppose your equipment didn't come within 4 months. Your project got delayed. So you do not have an excuse that, okay, my equipment didn't come, my reagent didn't come, so I'll not be able to complete it in 18 months. So generally, the funding agency doesn't accept that. They will see that you must have a contingency plan for that also. Okay? So I'll not go into details. This you have to go on yourself. Data collection tools. What important data collection tools we have? Generally, we use the Excel, or we use some specific tools like SPSS. Or we can use some EP info tools like there are a lot of Open EP and all. Again, study design, this has been described by everyone. I'll not go, but this is a revision only. Descriptive versus analytical, observation versus experimental. So this has been given multiple times, so you please remember this. And these slides will be given to you. So which type of study is it? And we have already taken a class on it. Again, cross-sectional study, I'll not go into detail. T-test, ANOVA, these are for continuous variable. Chi-square association, these are for categorical variables. Okay? Then there is another called ANCOVA. Then suppose you are experimenting with one, suppose we had a mushroom poisoning outbreak. Some cases came to Assam Medical College, and there were a lot of mortalities. There were around 60 patients came in Assam Medical College, and we did one thing. We calculated liver enzymes at entry, and all the liver enzymes were raised, and we wanted to develop a algorithm at the baseline. SGPT, SGOT, then other enzymes were there, bleeding time. Then we wanted to see the ROC curve, the receiver operator curve, and the Kaplan-Meier curve. So we wanted to develop at the age, the gender. So what are the causes? Then you can develop an algorithm based on the receiver operator curve, that what will be the base algorithm, maybe SGPT or SGOT or combination of all that. Then you can do another thing that is called a principal component analysis, PCA. The weightage that we get is called the principal component analysis. So I'll not go into detail these things. So one important study I'll just like to mention that is given by everyone, one of the largest study in the world is the Framingham heart study. Framingham. Framingham is a town in Massachusetts, US. So it is the longest duration of a cohort study. It started in 1948. Around 5,000 people from the town has been studied, and now it's the third generation of the people. Earlier we didn't know if smoking or atherosclerosis or cholesterol or diet or exercise are related to heart disease. So from that study, we came to know that exercise, diet, all are related to cardiovascular disease, stroke, and heart attack. So that is one of the biggest and the longest running cohort study, the Framingham Heart Study that started in 1948. Okay. So ultimately, I'll not go into details. You have to remember some tips. Align with funding priorities. That is number one. If your funding priorities is based on cancer, some disease or breast cancer or gallbladder cancer, you have to align with the funding priorities. Then keep it concise and evidence-based. That is another part. Review before submission. Don't wait for the last day. Last day submission is not proper. You cannot review it properly. You have to show it to your peers. You improve it. You discuss it with your peers. Then you submit. So there are a lot of things. For here, I will point out your studies should be based, design should be based on your objectives. And is it feasible or not in your institute? If it is not feasible, are you having a collaboration or not? Where it will be feasible. So already ethical consideration has been taken up, 2 different lectures. I'll not go into the ethical consideration. Common mistakes to avoid: vague objectives, inappropriate design, ignoring sample size, and over-ambitious timelines. So please avoid these things. These are the things to be avoided. Okay? So to have a good research project, you have to have in mind that you are not putting an objective vague. I have to read, the reviewer has to read 3 times the objective to understand what you are going to put forward. If the reviewer doesn't understand your objective, then your project may not be accepted. Sometimes what happens, a very experienced person from IIT, they have submitted so many projects, they have a lot of projects, then they will just put very carelessly the objective, the method, just 1-2 line. But a new reviewer will see, he will not see his prior capability that he is from IIT, because these are blinded. When the reviewer gets, it is blinded. Is he from IIT, is he from AIMS, or is it what? They may be blinded, but for each project, you have to give your concentration on your methodology, and your objectives should be clear. Okay, so already we are very, very late, maybe 1 hour behind schedule, but I'll just go within 2 minutes. Report writing is not the problem. You have completed the project. The last thing is to submit the report so that on time. On-time submission means before 3 months you have to prepare the report. By the end of the project, you have to submit it. And if you don't submit, the next chance of your getting another funding gets diminished. If you are taking 1 year, after 1 year you are submitting your report, or you are not submitting the UC, SOE, your next funding you may not get. So while writing the project, report writing, this is the flowchart. Project title, unique idea, principally, all those things are given in the ICMR website. How you are going to dispose the equipment, what is your product outcome, what product you are getting, is it a patent, is it publication, how many publication, are you getting some PhD, how many PhD you have produced from your project. So all those things comes in your final report. So you have to design your project in such that you get some PhD output, maybe patent, you may not have patent, but even Subarna sir has said, publication is a product. If you have a good publication, high impact publication, it is called a product. Lot of citation, and somebody may have a patent from that. Always you do not need to do a patent. The person who invented polio vaccines, Dr. Salk, he didn't patent it. He didn't. Lot of people don't patent everything because this is for the population. So ultimately, nowadays what we are getting is everything should be patented. Okay, but you need to have publication, if you have good publication. So Samarian Pandit sir had said about this beginning, manuscript writing, all qualitative data, but I disagreed for one, but he's not out here. I said it was mixed model study. Why? Why? Because I usually see first of all if the impact factor is good or not while submitting. Is it above 5 or below 5? This is quantitative data. Again, we see, I usually see if the publication is, approval is within 2 months, 3 months, or it takes years, 60 days, 120 days, more than 180 days. So these are the criteria I use generally from ICMR we use. These are quantitative data. So both qualitative and quantitative data, when you do a research, that's an example, you have a mixed model. Anybody who knows about quasi-experimental study? Anyone? Yes? Huh?
It's experimental study, but we are not doing the randomization.
Part is correct. I'm just telling you, if it is a pre-test, post-test, pre-test, post-test is a quasi-experimental. Suppose before starting this workshop, if we had a pre-test, you got a score of, average score of 50% or 60%. And after that, and now we are conducting another overall, you are anonymous. We are not taking a male or female or like or ladies or gents. This is not randomized, okay, or not selective. This is quasi-experimental, post-test, and we are measuring the outcome. From 60%, you are scoring 80% or 90% average score. So this becomes your quasi-experimental. Just I'm giving you an example like that. So this flowchart is for DBT. They ask for summary report, and then they will ask for comprehensive report also. This is for DST. Again for DBT it is similar. Section A, they will ask for this type of reports. They will also want a comprehensive report, and they will also ask for details of grant utilization. Okay? Manpower, staffing, details, recurring financial requirement, all those will be asked. So this is the format of report writing. I'll not go into detail. These are available in the website. These are available in the website. So deviation made from original objectives. So this has to be approved by the reviewers. Suppose you are giving and you are asking for a deviation, that this drug is not, suppose you are taking artemisinin and you wanted to see if artemisinin has got a anti-cancer activity in cancer cell line, like breast cancer cell line, and you wanted to add another drug, but it was not in your objective, then that is a deviation from your study design or study. Then you have to put forward to the reviewer in the first year that you wanted to add this drug, and if they approve it, then there is some deviation and you have to put it in the final report that you have made a deviation. So your data result has to be appropriate tables and graphs, like if you are doing a multiple regression table or adjusted odds ratio or your confidence interval, the data you are presenting always, if you are providing a descriptive analysis or association, always makes you are putting both the numerator and denominator data and the 95 confidence interval in your data. That is what reviewers looks for, if the confidence interval is or they are not. You are putting an odds ratio maybe 2.5 and you are not putting any confidence interval. What is the confidence? Confidence interval may be very large, from 1.5 to maybe 15. So your study is weak, or it may be very close and strengthened. So that confidence interval should be there in your results. So these things are again in the summary sheet and key findings. So ultimately key points should be there and bulleted if they can be used for public health intervention, if your project results in public health intervention. So this is very important. Your key points should be highlighted, and you should have a 300 words abstract of your find, because these are published. 300, because your report and abstract should be there. Summary report, conclusion, and key highlights, and your abstract should be there. So with this, I think I will conclude, because I'm taking a lot of your time, but I thought this was essential. To cover this part, we could have got into more detail, but due to lack of time, I'm just skipping some of the points. But this is very important, and this data you will be getting in the website, how to write your report. And this is one of the important. If you have given the report very clean and adequately, and you have published 4 or 5 publications, and you have patented, your chances of getting future projects and large projects increases. So with this, I thank all of you for your patience hearing. And if there is any questions related to this part, I'll be happy to take. Yes?
So while reviewing the project, you said that novelty carries 0 to 15 marks. So 0 to 15, how do you, anyone will classify that? And another thing is, rationally is 0 to 20 marks, where it's written of 500 words. So how do we do that so that we get?
See, when we give a rationale and a possible your impact and novelty. Novelty, you know that this type of study has not been done in the country, and if it is not done internationally, you will get 15 marks. But it decreases slowly if it is done by multiple, but there is a gap, still there is a gap. We'll still consider it as a novel if you have some gaps that you are doing to do, others have not done, and that information is going to help somewhere. Suppose somebody is getting a nanotechnology, or suppose somebody is finding a new therapy like Poltech or a nanotechnology based cancer therapy and all, and that has not been done in India. So we will try to see that novelty, okay, the person or the investigator will get between 12 to 15 marks. It is novel, and if the impact is high, suppose he is doing artificial intelligence or machine learning project, and if this machine learning will replace and benefit the society, and that has not been done earlier, so novelty is there, and it will have a huge impact, possible impact, you will get good marks. And if it has lot of studies has been done and already lot of things has been done, your novelty and your possible impact decreases. Okay? So this way, how? Suppose I'm giving an example, just an example, toxoplasmosis gondii, just came to my mind. You all know the prevalence is very high about Toxoplasma gondii. It is in Assam, it is around 35%. In Arunachal, it is around 56% of prevalence of Toxoplasma gondii. What does it cause? It can cause congenital malformation in the newborn. Okay, that will be rare. Another important thing is that in person, it can cause a decrease in reaction time. Okay, if I study the reaction time that I have collected from this room, blood sample 2 ml from each of you, 80% are there. I did a IgG analysis. I divided it randomly into 2 groups and put an app out there to calculate your reaction time. You have to click the color. If your color is red, then you have to click it and your reaction time comes up. 0.2 millisecond or 20 millisecond or 150 milliseconds, like that. Then I divided between 2 groups what is is there any association between toxoplasma positive and toxoplasma negative with your reaction time? So this is a simple study. But is there any implication of that? Is there border implication? That may have an implication in sports medicine. A cricketer like Virat Kohli has got a very good reaction time. Okay, so he may be toxoplasma negative. Those who are having toxoplasma positive may have a slow reaction time and he's a bad batsman or his reaction time is less. So in sports medicine it may be benefit, or not only sports medicine, maybe lot of accidents happen. You have to put the brake, you could not put the brake in time. If that data is known, if you are positive, your reaction time, then you go for treatment. The person goes for treatment. Okay? So chronic toxoplasmosis, so everything has got some importance. So how you put forward your matter, that matters. You can put forward that it has got national importance, it has got international importance, it has got implication in sports medicine, it has got importance in accident. There are 150,000 or more than occurs in India, fatal cases of accidents. If you know this reaction times improves with treatment, then you can reduce the fatality of accidents. So this is one hypothesis. I'm not saying that this is true. I'm just saying that hypothesis. So that way you can see for each and every project what is the importance, and what is the implication. Okay, any other?
Sir, I had a question.
Yeah.
Sir, what would be the best practices in determining the expected prevalence for a disease?
Expected prevalence of a disease. So there are two factors, as I have told you. If there is a previous from your region, from your geographical region, if there is any previous study, and suppose one is, as some example has been given, one is 23%, another is 3%, which one will you take? The 3% or the 23%? You are supposed to take the 3% because 3% will have a higher sample size. But if you use this formula, 4pq by d square, you are going to have a lesser sample size at 3%. You have to divide the d square by 2. Then only you will get a greater. That means precision, from 5% precision, you have to go to 1% precision. Okay? Then there is a study design. If your study design is a multi-stage or PPS sampling, then you have to put a design effect. And design effect generally we take from 1.5 to 2. Suppose you you do not have any prevalence data. We do not have. Then you take 50%. 50% you take as the prevalence, that will give you a 384 samples. Then design effect you take at 2. This will give you around maybe 800 samples, 750, 800. From 800 samples, you can, if it is a multi-stage, then again you have to double. Suppose you are doing it in wards and villages, or even different. Then again multi-staging, you have to double that. So that way the sample size increases. Why do we need a sample size? Because the sample size with limited number is a snapshot. Here in this room we have 100 people. Randomly I take sample from 10 people. I don't take sample from all the 100. It will cost me more. From taking 10 samples, I'll get the data of all the 100 people. The accuracy will be 5%. So for a population of India, 140 crores, do we need to test 140 crore population? No. We will test maybe a 1 lakh population or 30,000 population. But randomization has to be proper. So here comes the cluster sampling or PPS sampling. If you do a PPS sampling properly, your sample size of 30,000 for the whole country will be adequate.
Considering an outbreak where there is no prior knowledge on the prevalence, so how should we begin with?
Again, that is one thing, if it is a rare disease or if it is outbreak. Suppose outbreak, we do not do prevalence study in outbreaks, first of all. We take what is the meaning of the outbreak? Outbreak is that from previous infection you have more than double. Suppose in the month of October you have fever cases rising 2 or 3 times the previous year. Then you will be saying that we have an unknown fever outbreak in that area. I went to Longding in 2013. In the month of December, there was 110 deaths. Within a span of 1 month. Dr. Anirudh also accompanied me out there. And the news came, fever of unknown origin, within 1 month, 110 people died. We were oblivious. It is malaria negative. It is because in Arunachal they will think about malaria. Then we took only some kits of leptospira. Okay, these leptospira kits are not very sensitive. This lateral flow, they came negative. We collected 30 samples, and those patients were given ceftriaxone, IV ceftriaxone injection, and they are not responding. That was the clue to us. That was the clue. Because ceftriaxone IV injection was not benefiting the population. Then we thought the disease may be not responding to ceftriaxone. It is malaria negative. Then what it should be? Maybe lepto or scrub. Because lepto and scrub doesn't respond to penicillin type of drugs. So immediately these 30 samples, I went from, because it was an urgent situation. They stayed back. I came back with the sample, tested in the lab. All 30 samples came positive for scrub typhus. Immediately I rang up, start doxycycline for all the villages. Prophylaxis. Then last year, one outbreak occurred in Goalpara district. 11 with jaundice, 11 deaths come up within a period of 2 months. The team from Guwahati Medical College went out there. They declared it is an outbreak of hepatitis A. Okay, so we know that hepatitis A outbreak does not cause that much of mortality. Okay, 1 death. Okay, out of 5,000 population, 1 death we suspect. 11 deaths in a population of just 4 villages. 60% were infected, and we have 11 deaths. I thought it's not hepatitis A. We sent our team. Immediately I said, collect urine, collect blood, bring it out here. We tested for hepatitis A, and we tested for another, that was leptospira. Okay, the infection was co-infection of hepatitis A also there, but leptospira was positive in more samples, and leptospira came positive in PCR. And we detected one of the agent called leptospira wolfii for the first time in Northeast India. That was circulating in Goalpara. That was last year. So outbreak, you have to keep an open mind what was there. And regarding your question is the prevalence, we do not see the prevalence in outbreak. Like incidents, our thing, if it is a rare disease or something, we know prevalence is what, it's a cross-sectional. It may come up. We said positivity rate. What is the positivity rate of this pandemic in the month of January or in the month of February or in the month of this? Suppose COVID positivity rate is 4% last year. Now COVID positivity rate is 10%. It is increased due to this NB 1.8.1 because it is more infectious. It is more infectious. So positivity rate has increased. So it is not a prevalence. We said it's a positivity rate has increased. So outbreak, we do not see the prevalence in outbreak. Okay? Yes, sir. Any question? So thank you, everyone. Sir, if you have.
Thank you for a wonderful talk. Just to add him, what I mean to say, when we call outbreak, it is basically X bar plus 2SD. X bar means, you know, the average of the last 3 years, 5 years, whatever data is available, plus 2SD. Only in that case we call outbreak. And otherwise focal rise. If you don't have any data, so in that case any focal rise can be taken as outbreak, but the epidemic curve should be built up. Otherwise there is a focal rise only. Outbreak is a much bigger word. X bar plus 2SD. That is called the outbreak basically. So now we come to an end. We could have taken some feedback but as I talk to each individual table, but what I find that it's more or less satisfactory. And you have any additional comment to add on, ma'am? Anything from any participant you want to talk? I mean, what was your expectation? It was met or not? Because let me tell you, I've joined for 2 days only. I've taken charge. I hope to collaborate with you all. Yeah.
For this, can we get one representative from every state?
Just
a short feedback.
A short feedback. We did not prepare any feedback form. So let's start with the home state, Assam.
Thank you very much for this opportunity, sir. And I think in the introductory part, we were asked to keep our comments about our appreciation towards the end. So I think I reserved that for the end. I think it's a good opportunity for us because being in health research, we had been able to refresh a lot of things and also learn a lot of new things, especially the last. Especially what we had just learned about how to write the project and all, which was very, very helpful for us. And also some things that in between we had talked about, like the concept of meta-analysis, which we thought maybe this can be taken up later. And also another aspect that since ICMR has these capacity building opportunities, another thing that can maybe be taken up in future is workshop for peer reviewers as well. How peer reviewers work. So we had, after our listening to Professor Tharyan, so we had discussed and he said maybe this can also be taken up. Okay. Okay. That would be very helpful, sir. That would be very good for us also, sir. Thank you. And of course, I have to talk about the hospitality and wonderful food and the support from each of the RC NER region members. Thank you all for the opportunity, and it had been a really very good experience. Thank you.
So, taking a cue from ma'am, let me tell you, I'll share in the group a paper that has come up in Lancet. It was written by one of the ICMR institutes. So we can take that as a model and say, all the participants, this group I'll share with you. We can read and all of us can prepare a manuscript based on like that, taking that as a model. Health indicators, social determinants, everything. It's a systematic review that can, it has been never tried for the northeastern states. So that can have a huge impact. So that will be a mammoth work. So I'll send you all in the group. I'll post that, and maybe we'll fix a timeline, maybe 2 months. I will divide each one of the work. Then we can come up with a kind of systematic review. So you have to identify from each of one of you who can do it. I mean, systematic review, who knows, basically, that can be very helpful. Thank you.
Next, can we have from Arunachal?
Yes, this workshop was wonderful with the content made out in such a way that like...
How's the food?
Food? It was great. It is great. Yeah, the workshop was enriching, in fact.
Whatever good workshop is, if the food and stay is not good, then it is not good.
Yeah, I think this is a very good location. I think RMRC is very advantageous.
AC was working?
Yes, yes. Good. Yeah. So, apart from like the good hospitality and I think RMRC is fortunate to have such location. In the whole state, we might not have such location. In fact, Assam will have always advantage of having such good location for conducting workshops or like. But for us, like if we had to conduct this, I think we would be looking for like kind of hotels or to take care of the accommodation, take care of the conveyance, vehicles.
I mean, take home knowledge and interactions are good for you?
Yes, yes, of course. In fact, like all the, in fact, I had contact with some of the resource
Networking,
persons,
that
and
is
like
one of the, if you look at the objectives, it is one of the major objectives of networking.
yes.
In today's world, without network, you cannot proceed.
Of course, we were not able to interact with many other people, but at least one or two whom we felt like we should interact, we had interacted even with the participants. Them, I think even one or two, like the link that we connected here would be useful. And all the teams that we came from Arunachal is like one from basic science, one from clinical, one from paraclinical and public health. So it was a good combination, and like of course streams, the research is yet to come up. We have never like done research in like of course I'm also a member of the institute ethical committee, but and of course we read that we were told to go through the ethical guidelines.
Yeah,
but
you liked Dr. Patgiri's bioethics, I mean research ethics lecture?
Yes.
It was good,
So,
no?
so the sessions were of course we keep reading the bioethics guidelines, but then always we miss certain things, and of course whatever was told, like it has brightened us again. Like so, it was helpful. In fact, very nice, very good work, sir.
Meghalaya.
Thank you very much. It was a wonderfully organized program. Right from the beginning, when we were getting information about the logistics, I think Dr. P.K. Bora did a wonderful job, and all the updates were coming about the arrangements. So that was wonderful to you and your entire team. These sessions were so leader has to be behind, so push the so the sessions were also quite enthusiastic, and they were quite nice, but I believe that today it was too much stretched out, because I think 2 days things is covered in one day. I think it could have been spread. Yeah, so today we had a very long day, and then I think some of the things could have been taken over in 2 days, that I feel. And then initially, as the compressor said, that we should have some ice-breaking session, which is very important when we are meeting from all the 8 northeastern states. Initial first session after the inaugural should be an ice-breaking session where we interact with each other, know each other, what we are doing, how we are doing. So that will definitely help.
Good point. When we'll do the next workshop, we'll put
and that could happen over a field trip. So if you can keep that a part of half-day field trip include it, so that brings in a lot of energy in the group.
We were discussing that a half day anyway.
Yeah.
So next
And the other thing that I would like, so that you said about systematic review, that is very important. The second part is also about the data analysis. Once you have the data, then how you are going ahead and analyzing it, what tools, techniques. So that is also an important part which you can take up in the future workshops.
Right, and we can collaborate on that. We have
Yeah.
got fantastic statistician with us. So
And overall, it was a wonderful experience, sir, and the way it was arranged, the comfort which was taken. So, sir, kudos to you and your team. Thank you so much.
Thank you, sir. Sikkim.
somebody from that Sikkim, they have joined us.
First of all, thank you so much. I think from the beginning, since we were nominated, the coordination has been so well, and we have had such a comfortable stay. From the beginning, I think till tomorrow we'll have a comfortable stay here. And now looking at the workshop, I think most of it has already been mentioned. But in these 2 days, we have had a concise overview of research methodology. So most of it has already been touched. And at least, and we got a lot of tips from each session, we could gather some tips, how to do, what are the nuances, everything we could get. So I think in these 2 days, though it was very short, but we could take back a lot. And considering Sikkim, no, we always say that there are 7 sisters and 1 brother. Brother is the Sikkim. Though we share our many things common with Northeast, but Sikkim has its own difficulties and its own limitations. And like us, Sikkim Manipal being a private institute, many times the funding and all are mostly directed towards the government institutes. So we would, like if private organizations, private colleges are also included more, then we would have a better opportunity. So if ICMR can look into it, then Sikkim Manipal will be really benefited. Thank you.
Thank you so much. Our brother Sikkim has this time sent all ladies team. So I think it is a continuation of Operation Sindur. Anyway, thank you so much. Now moving on to Mizoram.
Yeah, one more thing that Dr. Lakshminarayan mentioned, the extramural grant that Dr. Barkaguti talked about. There are 3 types of, one is the CAR, which is more than 17 crores, and one is the large grant, and there is an intermediate grant, 2 to 7 crore, a small grant up to 2 crore. That is only meant for non-ICMR institute. For the last 2 years, if you look at the ICMR website, it is the major funder of biomedical research in India. Most important funder and for the private institutes. And non-ICMR institute, be it government or private institutes. So 30th September is the last date for, no, this extramural grant date this year has gone. So we'll have a series of workshops as Dr. Barkaguti very beautifully presented in details. Maybe we can have another workshop on this for the extramural grant. Please avail that. And if required, we'll handle it, right? Because this is the new ICMR has come up. There's a huge paradigm shift, huge funding for the non-ICMR institutes. In fact, we are not getting the fund. We are getting fund from intramural thing. And not only that, you rightly mentioned, Northeast has got a separate funding. So please avail that. And there is a huge opportunity and huge problem and area to you can address to, be it public health, be it epidemiology, be it clinical, be it basic science, right? There is a huge opportunity. Please try to avail it. Thank you.
Thank you, sir. Mizoram.
Greetings from Mizoram. Upon arrival, when I first saw the list of speakers, I was amazed. I was very excited as we were going to hear from all respected, well-known pioneer researchers, which we have all adorned. So as expected, it turned out that not only we learned about research methodology, we learned a lot from what our pioneers, our seniors have experienced in their journey. So this 2 days journey had been very fruitful, and I had learned a lot myself. Thank you.
Thank you so much. Now moving on, Nagaland.
This has been a very wonderful experience. Right from the time we got the invitation, we were all very excited. Why? Because while filling our APAR forms, what we requested was we wanted a workshop on research methodology. And when we got the invitation from P.K. Bora sir, we were very excited because we wanted to learn. Because this, you know, the topic that has been included here was something that we were lacking. There was a lacunae in our research areas, and you know, coming here, all the topics were very nicely covered, and I believe, like, you know, we are going back very enriched, and we will be able to contribute to our institute also. And since ours is a very new institute, we are looking for collaboration, and you know, we have learned from our seniors, from the speakers as well, how to go about and about networking and all those. So we're going back really equipped. And logistics was wonderful, sir. And the stay was wonderful. Just one feedback, sir. We missed this topic scope of health research in Northeast. So if we have a PPT, if you could share it to us, that would be very good, because we would also like to know the scopes. And yeah, thank you so much, sir. That was the session that was going to be held today, point number 5.
Mahanta sir's session. She was referring to Mahanta sir's session, sir. Okay, thank you so much, Nagaland. And now, Manipur. No, sorry, Tripura. Tripura has left. Okay, Manipur.
I thank the organizer. In one line, what I would like to say this is more than what I expected, what I got. Actually, I always prefer to be a student rather than to be a teacher. So it was wonderful. 2 days I spent really wonderful sessions, and all the workshop and lectures and all these things were wonderfully designed, nicely arranged, and from my side, there is no complaints regarding fooding and staying, etc. Now, thanks to the resource personnels who took the pain for these 2 days, about these 2 days, to explain so many hard things in a very lucid way. And one thing actually impressed me when other resource persons were discussing the problems. So other resource persons and the audience also asked the questions. Means this is so wonderful. So it is not, you see how ego is not that I'm a resource person, I shouldn't ask the question. So this is the very encouraging. You should come out of this shell. If you don't come out of this shell, you can't learn. This is the first thing of learning. And another thing is that it was also a wonderful opportunity to meet my many old friends over here at this stage, I can see. And it also gave me the opportunity to make new friends also. So thank you to the organizers. And regarding, because it was a very compact program, I think it would have been better if another session, if it could have arranged that way that I assume everybody is working, everybody has a research problem, but you know, all the luminaries and experts are there who knows so many things, so very well on the statistical designing and these things, and I am pretty sure somebody actually might be lacking these things. So it was a wonderful opportunity for everybody. So had there been one session where everybody could explain their work, what they're lacking, where they're facing the problem in designing, and if they got the answer for their own problems, it would have been better, and it was a good opportunity for them. But I know this is a very busy schedule, but in future, if you can arrange this kind of things, so who can explain, who can get the answer for their own problems, specific problems, so it would have been better for them. So with these things, again, thanking the organizers for organizing such a wonderful program and inviting me over here. I really thank from the core of my heart, and thank you all. Thank you.
Thank you so much, sir. Now, Manipur.
Thank you. First of all, I'd like to thank the organizer for giving this platform and for inviting us especially. And then, you know, the situation in Manipur is not good. At the time when we are about to come, we don't have the internet, and it's hard to get the message in time, but still somehow we manage. And for the warm welcome, we really thanks to the organizer for giving us a platform, such an opportunity. Well, coming here, we come completely blank, and we are returning back with the full packets of the knowledge. And for that, I really appreciate and thanks the organizers. About the food, about the accommodation, it's up to the par, and then everybody says that it is beyond expectation. And about the contents of the workshop, say different participants has says that things can be improvised. So I would suggest that I just feel that it's instead of 2 days, if we do for 3 days, like then various topic can be covered, like the meta-analysis, a review part, and then the analytical part and all those things. And the one thing I just like to know, I like to not complain, just suggest that if we got an opportunity to visit the ICMR NER, that would be a great experience for us, so that we every coming here have the idea of what are the facilities that ICMR has, so that in the near future when we want to conduct such kind of studies, and what kind of help or what kind of extension we can seek from the ICMR, that will be a better opportunity there. So beside that, everything is up to the par and very well organized, and we are really thankful from the side of the Manipur, and thank to the ICMR Northeast region. Thank you.
Thank you so much. Those of you who are having afternoon flight tomorrow, you may detour to our campus. It is near to the Dibrugarh airport, so you may kindly visit. You would be most welcome to visit our labs and our facilities. And further, I would like to announce that we are going to conduct this kind of research methodology workshop in every state in the next 5 years. We have got, in project mode, we have got approval and sanction, in fact, from the DHR. HRD call for Northeast, for specifically for Northeast funding, we have got, in RMRC, we have already got sanction for this. So over the next 5 years, we will be coming to every state and organizing this kind of workshop. Perhaps maybe for a more junior level or young researcher. And I know some of you are HODs and seasoned researcher. So some of you, we would need your help and support in conducting, as well as some of you can be resource person in that kind of program that we are going to conduct. So I would look forward to your help and support in that. And we could not yet formally ended the previous session, in the sense, though everything is done. I would like to thank the resource person, Borkokati sir, for the session on proposal writing and report writing. I hope the practical guide on proposal writing on our ICMR format will be helpful for the participants in submitting proposals to ICMR. And sir has also given us a final exam with 11 questions. And I am happy to announce that all of us has passed with flying colors. And the one who got those gifts are the topper list, okay? So normally top 10 was announced, but sir is lenient enough to announce 11, top 11 who got the prizes instantly. So thank you so much, sir. We would like to hand over a small gift as a token of our love and appreciation to our resource person as well. So I'd like to request Dr. Borkakati sir to kindly come forward. And I would like to request Lakshmi Narayan sir to kindly hand over the memento. I would like to request Dr. P.K. Borasa to kindly come forward as well, as we would like to hand over a memento to him. So I would request our DDZ sir to kindly hand over the gift to Borasa as well. Thank you, sir. So we are at the final end of our program. So since we are already late, kindly bear with us. We would now call for the vote of thanks, and after the vote of thanks, I would also request Dr. Subarna sir to kindly highlight his institute in 5 minutes, because he is working on traditional medicine and integrative health, and Northeast has a huge scope of work in these areas. So sir will present the work that is going on in his institute in NITM, National Institute of Traditional Medicine in Belgavi, so that as some of the participants said, possible collaboration can be thought in the future with the SIRS Institute as well. So with this, I would like to request Bora sir to. Yes, a quick remark from Assam team.
From all the participants, I like to thank you, because from yesterday you comparing the whole the episode or whole the program in so nicely. It's amazing and fabulous. Thank you.
Thank you so much, sir. Thank you so much, sir. Thank you, everyone. Before I invite the vote of thanks, I would like to invite Mr. S.N. Babu, sir, the Administrative Officer of ICMR RMRC Dibrugarh. He has a few announcements to make, so I would like to give the time to AO, sir.
Good evening to all. Respected our Director Suman sir, respected Subbanara sir, and respected Dr. Lakshminarayan sir. Thank you very much for all our participated in this meeting. As a good platform from ICMR to you as a scientific and research, this is a good opportunity for you and us. Always Northeast, golden face and golden heart. I am coming from South India. So more than 1 year I am working in Administrative Office of RMC Dibrugarh. So like very much for Northeast people. Once again in admin part, once again, and thanks to B.K. Bora sir, organizing for this meeting and arranging for vehicle Dr. Timbu sir, and our PhD student, administrative staff, and account staff. They are all more than 1 week to work for this meeting regarding arranging of vehicle, hotel booking, and everything. And 2 days, accounts people and admin people work for your preparing of TADA and everything. Don't worry, sir. Your TADA will within week create in your respected accounts, bank account. I assure to you. And one more, my humble request to all our senior Dr. Lakshman sir near retired this week. So I humble request to all, please stand of his service, orient of service in ICMR to claps to on standing and claps to Lakshmanan sir. Please. It's a wonderful service in ICMR. Not only ICMR, all 28 institute, he has to participate and advise to all institute. And special thanks to our sir. Sir, our anchoring is very efficient and lovable, sir. Our pronunciation is everything pakka. Thank you very much. And specially invitee, also thank you very much. I pray to God you safely go into your home. Behalf of our RMS ICMR Dibulgar. Thank you very much.
Thank you so much, Babu sir. Now I would like to invite Dr. P.K. Bora sir to kindly deliver his vote of thanks.
Respected dignitaries, esteemed speakers, dear participants, my colleagues, a very good evening to you all. So as we come to the end of this enriching two-day workshop on capacity building in research methodology for health professionals and researchers from Northeast regions for undertaking quality health research for diseases of national and regional priority, it is my honor and privilege to deliver the vote of thanks on behalf of ICMR RMRC NE Dibukar. For me, it is really a difficult task. This workshop was funded by DSR under PM Abhim Ayushman Bharat Health Infrastructure Mission and was proposed with the objectives of framing hypotheses and research questions, methodological parts including sample size calculations and sampling, importance of ethics in biomedical research, data analysis using statistical software, report and manuscript writings. Based on these objectives, we accommodated the expert of national and international report, and we presume that majority of the objectives are achieved with these workshops. I think that this workshop will be like eye-opener for the new researchers, and it will be like reorientation for the senior level researchers because they already have many experiences in doing research, but it will guide us further to do a quality health research. First and foremost, I extend our deepest gratitude to Dr. Rajiv Behel sir, Secretary DSR and DG ICMR New Delhi for allowing us to organize this event and delivering his insightful address on ICMR's vision for Northeast India, as well as highlighting the need for practicing to get the best outcome of these research workshops. Sir, your continued support and leadership will inspire us for undertaking good quality research that truly serves regional and national priorities. Dr. Sanghamita Pati Madam, ADG, Additional Director General ICMR New Delhi, who could not attend this workshop because of her busy schedule, but he supported us a lot throughout from the development phase to the execution of these research methodology workshops. Your support will certainly encourage us for undertaking big endeavors in future. Monisha Saxena Madam, we also thankfully acknowledge the support received from Monisha Saxena Madam, Senior Deputy Director General Administrations, for extending her support in various aspects for conducting the research methodology workshops. Dr. R.S. Dhaliwal sir, who is the chief of NCD and who is looking after our centers. We sincerely thank Dhaliwal sir for guiding and conducting these research methodology workshops. We are indebted to Dr. Lakshmi Narayan sir, DGG admin, who was instrumental in approving and releasing the fund. Sir, it is because of you we could select this venue, this beautiful venue, because you identified, and that's why we have selected this beautiful venue to conduct these workshops. We also have come to know that you are retiring in this month only, so we wish you all a happy post-retirement life. Thank you so much, sir, once again. Dr. Dharmesh Lal, who is one of my best friends, he supported us a lot during the conceptualization of the projects and also pursuing the approval at ICMR headquarters. We are also blessed and guided by three respected directors, namely Dr. Natarajan sir, actually who was there at the time of conceptualization of these proposals. And in fact, Lakshmi Narayan sir asked Natarajan sir that you should submit one project for research methodology workshop under PM of him. Accordingly, Natarajan sir guided me, and we have developed that proposal and submitted to ICMR headquarters. And we are proud to disclose that without any change in the proposal, without change in the budget, Lakshmi Narayan sir approved that funding. So that is why we are here. Dr. Subarna Roy sir, he is the director ICMR NITM Belagavi, but he was taking the additional charge of our institute. He had guided throughout the period for conducting, where to conduct, how to go ahead, all administrative and logistic support. Dr. Suman Kanungo sir, who is our current director and who joined on 10th of this month, he was also very instrumental in guiding and helping to conduct this research methodology workshop. Thank you, our respected directors, sirs. Thank you so much. I am deeply grateful to Dr. Pratap Tharian, retired professor of psychiatry, adjunct professor, clinical epidemiology unit, CMC Vellore, whose decades of experience in evidence-based medicine reminded us why health decisions must always be anchored with reliable data collection, adopting rigorous methodology. We had the opportunity to listen to the lecture by Dr. Sanjay Javekar sir, a leading voice from KM Hospital and Research Center, and also the professor in Health Science KM Hospital Research Center, Pune. In fact, sir is also one of the mentors. Other mentors are Dr. Biswajit Kumar sir and Dr. R.M. Pandey sir. Under their mentorship, we could establish health and demographic surveillance system in Dibrugarh district. That health and demographic surveillance systems include the population of the village and 20 tea gardens, covering around 1,06,769 populations. We had the privilege of learning from Dr. R.M. Pandey sir, retired professor, Department of Statistics, AIIMS, New Delhi. With grace and precision, he unpacked the complexities of study, design, sampling, sample size, reminding us that the foundation of good research lies in formulation of a good methodology. Thank you so much, sir. A special thank to Dr. Biswajit Kumar, who is the founder and chief scientist of Community Empowerment Cell, who shared his journey in implementation research and also highlighted how it was important in the perspective of policy implementations. In the same spirit, we would also like to thank Dr. Suborna Roy sir, Director ICMR NITM Belagavi, who demystified the art of publishing. His sessions on scientific writing, which focus on integrity, impact, and finding one's voice in academic space, was deeply enlightening. On the second day, Nolin Mehta sir, who is the director, NEGRIMS, Shillong, whose thoughtful reflections on ethics and role of institutional review boards underscored the immense responsibility we carry when researching on human lives. He agreed to join this workshop even after his busy schedule. We are grateful to you, sir. We also had the privilege of listening Dr. Prem Kumar Moni sir's lectures, who is the Vice Dean and Professor, Division of Epidemiology and Biostatistics, Population Health, St. John's Medical College and Research Institute, Bangalore, who distilled the fundamentals of research methodology into clear, digestible insight, reminding us that simplicity and scientific rigor are not mutually exclusive. Thank you so much, sir. Dr. Samran Panda, who is the Chief Editor of IJMR and former ADG, ICMR, walked us through the nuances of manuscript writing. With editorial wisdom and candor, he showed us what makes a research paper not just publishable, but purposeful. We are indebted to you, sir. We also thank Dr. Saurabh Jyoti Patgiri, my colleagues, and scientists, ICMR RMRC Dibrugarh, for his clear explanation of ICMR's publication ethics. His sessions ensured better understanding and the need for conscious and responsible compilation of any manuscript. Dr. Shiraz Ahmed Khan, sir, scientist G, and my senior colleague from ICMR RMRC Dibrugarh, who brought the light the often invisible challenges of conducting research in difficult terrains. His sessions was a powerful call to resilience, creativity, and empathy in fieldworks. Thank you so much, sir. Dr. Biswajit Barkakoti, my colleague from ICMR RMRC, NE, closed the loop with a focused sessions on writing good proposals and report writing, reminding us that research will be only impactful when we communicate it clearly and convincingly. My sincere thanks to all the esteemed participants, from young researchers to senior professionals, who came all the way from different NE states to Dibrugarh to attend this workshop, making it a grand success. I assume that it was a learning experience for all of us and a rare opportunity to have all the experts of national and international effort in one platform. This is the opportunity to identify your mentor to initiate and carry out good quality research. I also request you to provide your feedback. As you know, there is no dearth of money. For a good occasion, there is no dearth of money. But there is dearth of provision of relaxations. So within that limited provisions, we tried our best to provide best services to you. If anything we could not provide, we may be forgiven for that. I sincerely thank all organizing committee members, Siraj Khan sirs, Dr. Saurabh, Dr. Dimpu, Dr. Aniruddha, Dr. Poonam, Dr. Atik, S.N. Babu sir, administrative officers, Mintu Gogoi, accounts officer and his team, all my colleagues, PhD scholars, Ms. Tara and Soindas. They should have a special mention because they are the person who are coordinating throughout the all northeastern states. And David Squads, there are two Davids. One is David Pegu and David Dotto, IT professional. They're constantly working to make it a grand success. Finally, my sincere thanks to the management of Solitary Bliss, Tinsukia, and also Palm Resort for their support in venue arrangement to accommodations. Special thanks to Chef Sandan and his culinary team for arranging the delightful and tasty dishes. Thank you so much. Wish you happy and safe journey. Thank you.
Thank you so much, sir. Now I would like to request Dr. R. Lakshminarayan, sir, for a final feedback, sir.
First of all, sorry from ICMR side for keeping you till 7:30 p.m. And only 2-3 messages I will give you. And that madam, she asked for funding for private institutions. For the last 2 years, private institutions are getting more funding from ICMR. You can see it in ICMR website. Second thing I will tell you, you will be shocked to know that you are not tapping the funds of Northeast allocated by ICMR. Not even 50% we are using Northeast funding. So you have another 50% with you. You can avail these funds. As this person said that he is going to conduct state-wise research methodology program. Every state has got unique way of doing, unique cultural, what Kansar has said. So I have in May only I visited Arunachal. You can do it, sir. You have all the facilities, and research faculty will be from ICMR. So each state you take this opportunity and conduct. We will be very happy to fund you. And I am very happy actually. After my basic training, I started from Shillong, Meghalaya, and at the end of my retirement, I am ending from Northeast Dibrugarh, Assam. So this is my last workshop, and this is a very happy experience to me that I started from Meghalaya and ending it in Durgapur. Thank you all the best.
Thank you so much, sir. And from my side, I would like to thank everyone as well. And I request everyone to kindly bear with me if I intentionally happen to make any mistake or hurt somebody's feeling or intention within the course of these 2 days. While I thank everybody sincerely from the bottom of my heart, I would like to dedicate a special thanks to the Sikkim team, as I have missed out the local language welcome yesterday. So let me try this. ICMR RMR CNE Kotaraf Bata Sikkim ka sabai pratinidhi horule hardik dhanyabad cha. Thank you so much. Have a good night. Safe journey back tomorrow. See you somewhere, sometime. Once again, thank you so much. Excuse me, everybody, can you please just stand on your own place, and we'll have one final group photo. Here. Okay. Okay, let's quickly come to this side. And before we depart, let's have a final photo. And for the participants, dinner is included in your hotel arrangement, so you can order the room service for your dinner. And I request the RMRC team to kindly stay back for a short internal meeting.